unpublished on looking at regulatory networks that are occurring in different cell populations that are important for controlling the plasticity of development to both flooding and water deficit stress. So a quote that inspires me is this one: "In wilderness is the preservation of the world." To me, this has multiple meanings, perhaps not the same meaning as the author, but to me it is that we can look at nature and find inspiration. Just as this water hyacinth shows us the elongation of a petiole as it lives in an aquatic ecosystem, we can look at nature and find examples of strategies that are effective for a plant to grow in a water-prone environment. So this water hyacinth shows the petiole elongation, which is an avoidance strategy, so that it is able to be reproductively competent and have effective photosynthesis above the air-water interface. This river photograph, and this one as well, was taken by me in Peru, showing you the deciduous shrubs on the bank of the river that are bursting with leaf growth as the water level falls. And in this case, shows you these plants that were fully submerged for during the high water season, and they have not lost their leaves, but they're able to endure and maintain this greenery with low photosynthetic competency when it's underwater, and then just maintain and grow when they're desubmerged. This is an avoidance strategy as well as the elongation strategy, and this is perhaps an endurance strategy that we can consider a true tolerance strategy, all shown in nature. If we turn to rice, which is the model organism of my group, what we find is that there's many mechanisms for aeration that allows there to be exchange of gases between the air and regions of the plant that are underwater. So that could be waterlogged root systems, or also the aerial tissue when the plant is partially to completely submerged. Those traits that provide aeration vary in nature, but in a rice plant include the formation of lysigenous parenchyma, which are gas spaces formed within the cortical layer of the root system, and then up into hollow stems and hollow regions of leaves that allow passage of gas exchange of both CO2 and oxygen between the roots and the shoots. And then also as well, there is epidermal lignification and subepidermal, particularly in the exodermis, suberization that can occur to provide and facilitate this aeration. We see these traits also in nature. So if we look at these Cecropia plants also growing in Peru, we can see that there are lenticles and other features of the stem. And when these are cut, you can see that they are very hollow plants to provide that gas exchange. Well, rice is a wonderful crop species to work on. I know it's not a major crop in Mexico, but one of the reasons why it's so wonderful to work on is the genetic diversity. And that genetic diversity has allowed rice to be cultivated in very dynamic ecosystems. The diversity comes from the wild relatives of rice that were used in domestication. And the traits include things like the ability of rice to grow in a delta region where there is a rising flood over the season. These plants maintain some leaf material above the air-water interface and have very long stems that I'll show you on a subsequent slide, as I mentioned the mechanism that allows these deep water rice varieties to have this ability to be reproductively competent in deep water. Most rice in the world is grown in a paddy ecosystem. These are rain-fed regions, typically with low irrigation control, and they can be highly productive because the paddy has the water to provide rice both water and nutrients in the soil during early developmental stages. But Post-establishment, these plants can undergo a partial to full submergence event, and they can also undergo a drought event, and this can be intermittent. So sometimes we see that a paddy will be submerged and then droughted, or oftentimes there's these periods of drought because farmers are dependent upon rain. Also, as well, there are upland, dry or aerobic rice that's grown, and these are irrigated. Collectively, these varied rice varieties that have been cultivated for many years by farmers, these land races, have a lot of genetic diversity that provide opportunities, not only for studying flooding, but drought and many other traits that are relevant for agriculture today and that may have been lost during selective breeding to give us the elite varieties that are widely used. So deep water rice has this ability to escape a rising flood because of internode elongation. This is an underwater escape response, and there's a number of genes that are important for the response. The characterization of these genes has all been done in Moto Ashikari's group in Japan. The first genes that were recognized were these snorkels, and subsequently several other genes. There's some lovely reviews on this topic, and I encourage you to look at it. I just want to compare this to what my group has studied quite extensively, and that is the underwater quiescence response that's seen in varieties that have a gene called sub1A. And this particular gene allows the plants to be quiescent when submerged, by contrast to non-sub1 varieties, which will attempt to elongate, very similar to deep water rice. Sub1A is important, as well as a couple of other genes that I'm going to mention in the next slide. So one of these genes that's relevant to both underwater escape and submergence tolerance is the gene that's called semi-dwarf 1. This is the Green Revolution rice gene that limits GA biosynthesis, which is mutated in sub-1 varieties, but is a functional gene with high activity producing bioactive GAs in this underwater escape deep water rice variety. So in all cases, these genes, the snorkels and sub1A, which encode related ERF transcription factors, are triggered by the submergence event by the accumulation of ethylene. And in the case of deep water rice, ethylene activates the snorkel genes, which then promote responsiveness to GA. This activation of ethylene really de-represses a pathway where GA promotes starch catabolism and growth. And so this pathway is really activated in response to submergence in deep water rice. And by contrast, what we see in rice that has non-sub1 is this pathway promotes growth directly without that bolstering of GA levels and elongation of the internodes underwater. The sub-1 varieties, though, on the other hand, show this quiescent response, and what we've been able to decipher is ethylene upregulates sub-1A, and sub-1A then dampens the GA responsiveness, curbing this growth response. So that's a very brief primer. There's a lot more details there, but I just want to go on to say that this single gene not only provides resilience to submergence, but it also enhances the plant's ability to recover from submergence. And so this pair of plants really contrasts the post-submergence recovery response, where having sub-1A, the plants recover quite quickly and can flower, whereas plants that lack sub-1A have elongated more and are more senescent, and they either are delayed in flowering or die as a consequence of the stress. So this illustrates how identifying a key gene, sub-1A in this case, has enabled the breeding of submergence-tolerant rice. And I want to say that the Sub-1 project was a project that was between 3 institutions, International Rice Research Institute, as well as Pamela Ronald at UC Davis and my group. And it was the International Rice Research Institute that has been responsible for breeding sub 1A into farmer preferred varieties. And at this point, providing a flooding insurance for farmers so that their plants can undergo very prolonged periods of complete submergence and they can have less loss of the yield as a consequence of this. It's been very impactful, and sub 1A has been bred into many other rice varieties that carry genes that are critical for both quality traits as well as other stress resiliences. And so this example is one where we see that finding genes that exist in germplasm can really benefit farmers. And so this is something that is ongoing and very important, and I want to emphasize involves the activities of many people. And so this quote that I show here is also another one I find very inspiring, and that is, "Men work together. I told him from the heart, whether they work together or apart." And the point of this really is that the challenges that we have are too great for an individual to work on. Of course, we all can have our part, but in order for us to really safeguard crop production, we really need to work together. And the sub 1 story is really an example where that has been essential for us to get to where we are today. So I want to go on and talk about some other collaborations that my group has been involved in. These are ones that are quite active and ongoing now. And the first one that I want to talk about was the initial project of a team of people that we call the Plasticity Project. And this is the group of us as of several years ago with the lab leaders, Siobhan Brady, Nilima Sinha, Roger Deal, and myself, as well as the postdocs and graduate students that were involved in the early chapters of the Plasticity Project. Kaisa Kahala, who's now at Utrecht University, Donnelly West, Germain Paluzzi, Mauricio Reynoso, and Marco Bacek, shown here. So I'm going to talk to you about the work that we've done to define regulators of developmental plasticity to stress, looking at multiple species, and taking advantage of the integration of data on chromatin, particularly chromatin accessibility, and mRNA dynamics. And we were motivated to do this because of questions we had of what's going on within the shoot base of plants and in specific domains of the plant. So in the case of sub 1A, what we see is meristems survive. The apical meristem, the primary meristem, or tiller meristems can continue to survive in this quiescent mode and then are really the source of the rejuvenation after desubmergence. And then also we have questions about what goes on in roots where there's a lot of dynamic plasticity that's relevant to varied stress and environmental responses. And this is the lower half of the plant, which is really much more difficult to capture in an environment such as this field environment. And so together we worked on translating technologies that we had developed in both my lab and Roger Deal's lab so that we could look at defined groups of cells to understand the developmental plasticity and define the gene regulatory networks that underlie that plasticity. So Roger Deal at Emory University had developed a method that's called INTACT. So this is the isolation of nuclei tagged in specific cell types. INTACT is really a very simplistic concept. Roger built a chimeric gene that encodes a protein that is associated or anchored in the nuclear envelope. So his initial constructs are associated, and in rice we redeveloped this so that the construct or the protein would be anchored. And then this is associated with a biotinylated portion of the protein that then facilitates the ability of these nuclei that are tagged to be purified using an antibody linked to streptobdivin. So it's a very powerful way to isolate nuclei from cell lysates of tissue that has been flash frozen after growing in different environments. And then this will capture the chromatin and any RNA as well as proteins that are associated or contained within the nucleus. The second technology is the translating ribosome affinity purification, or TRAP technology, that was developed in my lab many years ago. In this, we tagged a single ribosomal protein of the large ribosomal subunit. And this enables us, again, to use biochemical purification and capture, using an antibody conjugated to a bead, those ribosomes that are associated with mRNAs, or really we look at it the other way around, those mRNAs that are in a single to a polyribosomal complex. This gives us a readout of those RNAs that have been initiated or are undergoing translation within cells. And by using promoters that drive the intact or the trap constructs with specific cell types, we can capture these readouts, those which are from the nucleus or from the ribosomes, in specific domains of expression. And so the Plasticity Project team, particularly Kaisa Kahala and Germain Paluzzi and Marco Bacek at this time, developed a toolbox whereby these intact and trap constructs were expressed in different cells. So they use the traditional or very common constitutive or near constitutive 35S promoter, and then ones which would capture actively dividing cells, ones that may be expressed specifically in the cortex or in the quiescent center, the endodermis, or the vasculature as examples. And this was done with tomato, Solanum lysopersicum, the wild species Solanum pinelli, which grows in dryland, elevated mountains in the Andes, Medicago trincatula as a legume model, and then rice. And so with this toolbox, together we have done some very challenging experiments over time, and I'm going to tell you about these adventures that we have had. So the first one was meant to be a very simplistic experiment where we took these 4 species and our trap and intact constructs that were driven by the 35S promoter and asked for whether or not there was conservation of gene regulation specifically in the tips of roots of these plants. And we're interested in this early response to low oxygen stress, which is a component of the response to submergence. And so when seedlings are submerged for a short time, in this case we used a 2-hour time point, within the root tip as well as within other cells of the plant, there is a change in the levels of ethylene, so those levels are concentrated, and then there's also a change in the levels of oxygen. And we'd characterize this response in Arabidopsis quite extensively, and we're interested in, across these species, what sort of response we would have at this very early time point when there's this first response to low oxygen stress. We took advantage of the intact method to look at accessible chromatin using a methodology called attack-seek. And for those who are not familiar with this, it allows us to see regions of chromatin that go from being condensed to accessible. And that increase in accessibility is presumably due to the binding of transcription factors or changes in really the chromatin remodelers so that regions become accessible. The attack-seek method takes advantage of a synthetic or an engineered transposon, which can enter in regions that are accessible and not condensed chromatin regions. And then by using next-gen sequencing, one can find regions that become accessible, and these are often just upstream of transcription start sites, as well as in distal regions that can be accessible, and then often also downstream of genes. So this was a very powerful method that we were able to do using this 35S intact purification. We also used intact purification to look at the nuclear RNA because we wished to see how that readout compared to the traditional bulk RNA readout. And then we also compared these two RNA readouts to the trap-seq readout, so this translatome that we get from capturing RNAs associated with polysomes using trap. And then we went further to also look at ribosome footprints. So we looked very deeply at this pattern of differential gene expression and discovered quite a lot. And I'm going to digest this down to just a few slides for you. So by looking across these different species and recognizing genes which are orthologous or homologous, we're able to recognize gene families that we could then compare dynamics in gene regulation across the species. So it was just under 7,000 families that we were able to look at. And when we looked at our combined data, we were able to see that there were 68 of these families of genes that had one to many members that were upregulated by this 2 hours of submergence or this early low oxygen response. We also looked at these genes much more deeply to consider whether or not the genes were syntenic, so both orthologous and syntenic. And I'm not going to talk to you about the syntenic observations that we have, but just suffice it to say there were some very interesting things. So this 68 families of genes we called the submergence upregulated gene families, or the SERFs. And I'm going to tell you more about the SERFs and how they compare between these species. So by taking advantage of this attack-seek method, we were able to show that the upregulated SERFs have promoters that show a pronounced opening or an increase in accessibility, as shown here by this rise in this purple signal right over the transcription start site, or just 5' to it. So this is the region, sort of a normalized region of coding sequence, showing you this increase in accessibility 5', and then also 3' of these strongly upregulated genes in rice, and then also in Medicago. And so we were able to use the data here showing this accessibility to see that there is this consensus between upregulation in RNA and increase in accessibility. And that can be seen a little bit more clearly if we take our 4 RNA readouts and do a clustering analysis and show, for example, that this cluster is the most highly upregulated genes across all of our readouts, and also showing the most increase in accessibility in the attack-seek data. And I realize this is very tiny, but it's meant to show you how we can see just how there's this consistent opening. And if we look across all of these, those which are highly downregulated show very limited change, or even a downregulation in accessibility in response to this condition. So these regions of accessibility here and across the genome were used to look more deeply at the dynamic genes in order to build for these submergence upregulated genes cis-regulatory networks for the 4 species. And so again, focusing on this group of genes, the SERFs, and the accessible chromatin regions, as well as the presence of motifs that could be recognized using DAP-seq data or methods such as those that are provided by the MEME suite, we're able to identify 4 transcription factor binding sites that were overrepresented amongst these genes and compared in many instances between orthologous genes. And with these, we were able to build networks of association between transcription factors represented here and genes that were in the networks. And I want to show you some of the major findings of this by comparing these circular gene regulatory networks where we've displayed genes as very small symbols on the outside here, connected to transcription factors that are represented by these four the hexagons of the network. And again, these are transcription factors deduced by the cis regulatory motifs and are likely multiple transcription factors within these BHLH, HRPE, MIB, and WRKE motif-associated families. So in rice, many of the genes that were upregulated are associated with the presence of this red motif, which is this HRPE motif. Many genes have more than one motif associated, and it's complicated. But if we look across the species, what we can see quite easily is that medicago, tomato, and Solanum pinelli have less of a prevalence of this HRPE compared to the other motifs shown. And in the case of Solanum pinelli, this HRPE motif is the least prevalent. So I just want to emphasize that if you go to the paper, you can look at these in great detail and see every last one of these genes. There's a lot of data here, and I'm just digesting it for you. So I'm going to focus on this HRPE motif, and I'll tell you a bit more about the exact motif and the transcription factor that's relevant in a moment. But I first want to show a really interesting observation, and that is when we look at the number of motifs in these upregulated genes, so 0 to multiple motifs, and whether or not the motif is within a region that's accessible, so this is a transposase hypersensitive site, versus outside, what we see is that the motifs that are inside accessible region are the ones that show the most pronounced upregulation in response to the stress. And this was seen in both rice and medicago, where we had the attack-seq data. And so this suggests either that accessibility is associated with binding or accessibility precedes binding. We don't actually have a real true understanding of which comes first or not. But this motif had been previously characterized by a former postdoc of mine, Angelika Mostrov, in collaboration with my group, and it was shown to bind a group of ethylene-responsive transcription factors in this subclass 7. And it turns out that the sub-1 and the snorkels that I talked about earlier are group 7 ERFs. And with Angelika's help, we were able to connect the association of binding to this motif with the upregulation of a core group of genes that are the hypoxia-responsive genes of Arabidopsis. And so we're now seeing that this motif is conserved across species and putatively associated with the binding of group 7 ERFs. These transcription factors are extraordinary and important in terms of plants' responses to low oxygen stress. So as oxygen levels decline, these ERFs are stabilized. And in Arabidopsis, there's 5 group 7 ERFs. 3 of them are constitutively expressed. And so during normal aerobic development, these proteins are turned over by a pathway that's called an integron pathway. And I'm not going to go into the details of this pathway, except to say that it is an oxygen-dependent or nitric oxide-dependent degradation pathway. When oxygen levels decline, and as that decline gets to below 10% oxygen within a cell, what we see is that the levels of these ERFs increases. So these constitutively synthesized and translated proteins accumulate, enter the nucleus and activate the regulation of genes that are the hypoxia responsive genes. So this is data from Arabidopsis. We know the genes that are upregulated are these anaerobic metabolism genes, and then several genes that are important in this pathway. One of them includes a negative regulator that's called plant cysteine oxidase, and others of these important genes either positively or negatively regulate the pathway. And so I just want to say that sub1A is an ERF7. It has the characteristics that would make it a target of this pathway, but it's unusual in that it is not stabilized by the same pathway. So that's just a little aside. But when we look at the networks that we discover with this in these studies, what we find is that there's really strong conservation in both the presence of these genes and their regulation across the species. And so to highlight this, I just want to say that we've been able to show that there's commonalities in the hypoxia response across these species, and we see dynamics in the expansion and contraction of these families and variation in the motifs that regulate them. And so just to summarize, this key low oxygen signaling and response mechanisms and the hypoxia response genes are highly conserved but vary in their cis-regulatory features and the degree to which they are enforced across plant species. So I want to turn to some of the more recent work that we've been doing on root plasticity, which asks a question about how plants' root systems respond developmentally to environmental cues. And what we're really interested here is in considering relationships between water extremes. And so I want to do this a little bit quickly because I can see I'm running out of time. But again, we can look at variations in nature, and we can also address the question, which I think is really important, and that is whether or not some of the traits that are relevant for drought are also relevant for submergence or waterlogging. And there really are two that are quite evident. So parenchyma are relevant to gas exchange under waterlogging conditions and are also important during drought to facilitate the growth of long, thin roots to access water deep in the soil. The second one is the formation of a suberin barrier on the exodermis, in the exodermal cell layer of the root. And under waterlogging, this ensures that gas exchange can actually take place near the root tip where cells are actively dividing and growing, as opposed to escaping laterally outside. So it's a barrier to low oxygen. And in terms of water deficit stress, this suberin barrier is important to ensure that water is taken up and doesn't escape via a diffusion barrier. So work that we've done has been to focus on how these different root responses to submergence, waterlogging, and water deficit, as well as the recovery from these responses, is dynamic in different cell types of root systems. And given the time, and I'm kind of surprised I got so far behind in this talk, I really want to jump ahead, or maybe I'll just show you just a very, very little bit of this work. But it was done by Mauricio Reynoso, German Paluzzi, and Alex Borowski. And what they were able to do was to collect a number of trap-seq and intact-seq data sets for different cell populations across the different, really 9 different environments that were evaluated. And with that, to use some of the same types of pipelines that we used previously that ultimately defined, in this case, cell population and conditional networks that associate transcription factors with genes. And so this includes co-expression networks that leveraged chromatin accessibility data and also inference analyses that allowed us to highlight and recognize hierarchies between transcription factors. And so with these analyses, we're able to find things such as a core response to water deficit stress. And I'll just highlight that to say that with the analysis, we're able to recognize some of the known ABA responsive networks and associations between transcription factors in those networks. And so this dynamic network that video that I'm showing you here shows that under control, water deficit, and drought conditions, that based on the analysis, there's really a hierarchical interaction between these transcription factors. So these ones which are known to bind an ABA responsive element and others which were recognized within our core response data set. And then additionally, in our studies, we've been able to recognize genes that were associated by expression specifically in this barrier cell layer using promoters driving constructs in this endo-exodermal domain, those genes which are associated with suberin biosynthesis. And just in the interest of time, I'm going to skip this and just go on to this last slide, which says, just summarizes this ability of ours to take advantage of these cell population analyses to define response networks that give us an idea of relationships between transcription factors, which in some cases are apparently very broadly conserved across plant species, and in other cases may be more specific to monocots or even rice, and identify dynamic networks. And at this point, some of the key questions that we have are, what is actually triggering this network? And a paper that was published this week in New Phytologist says that this suberin network in rice that is activated under waterlogging stress is ABA responsive, and that's also what we've been able to deduce, as well as others, that it's ABA responsive in terms of water deficit stress. And so going back to this idea that there could be common mechanisms that are important for both waterlogging and a water deficit in a key aspect of root developmental plasticity, leaves us with this perspective that our type of analyses are allowing us to recognize these key features. And so I just want to conclude with saying that this talk has highlighted some conservation and flexibility of gene regulatory networks that are relevant in terms of plastic response in roots. These networks are highly dynamic and reversible and can be within specific cell populations and clearly control a number of resiliency traits by diving deeply into chromatin accessibility and taking advantage of the trap type transcript analyses. We're able to identify networks that now, based on motif discovery as well as transcription factor discovery, should facilitate looking both at existing genetic diversity and stimulating breeding studies, and also potential CRISPR engineering studies allow for greater control of these plastic strategies. And just in conclusion, I want to say that everything I talked about today really involved working together. And so I urge you to seek complementarity, identify common solutions, and work together. So thank you very much for your attention, and I hope there's time for a question or two.
Thank you so much, Julia. It was a wonderful talk, really, and very, very interesting. I hope we will have you again sometime soon. But now go to the questions. I think we'll have time just for one quick question. And the first one will be from Dr. Luis Cárdenas. Luis, you can go now. You can unmute yourself. Luis? Let's let's see. Now.
Can you hear me now?
Yeah, now.
Yeah, okay, thank you, sorry. That was a wonderful talk, Julia. I just have a very quick question. So regarding the hypoxia response with this transcription factor, the ERF7 group, so do the response is very similar in the meristematic region and the cortical cells, for instance, or do you expect to see differences? How the plant adapt to these hypoxia conditions?
So that's a very intuitive question. Thank you very much, Luis. So something I didn't mention is that meristems are conditionally or constitutively hypoxic. And so one of the reasons why there is this highly conserved pathway does indeed have something to do with the role of hypoxia there as defining a niche within meristems. And so, so we do see that there is this hypoxia response superimposed within the meristematic cells. We know this is the case because we do see within those cells the upregulation of response to the stress. And so I think that there's really a sort of a reinforcement of a response that's already there within the meristem, that's at really just a much higher magnitude within the meristem that we also see across cells. And so it's really quite older work that was done by Angelica Mustafa and Arabidopsis that shows this the most clearly. And we see this as well generally in these other species, yes.
Okay, thank you.
So I'm sorry, but we have to go to follow up with the next one. And thank you so much, Julia.
All right, bye-bye.
Bye. Okay, so let's go to our next talk that will be presented by Professor José Dineni, and also it's my honor to introduce him. He will talk about discovering innovations in stress response through comparative genomics. Thank you so much for accepting this invitation, José. Go ahead.
Of course. Thank you, Alejandra, for the invitation. It's great to be able to speak to this audience, and congratulations on assembling a really fantastic conference. So today I'll be talking to you about largely unpublished work where we are exploring the diversity of resilience, stress resilience mechanisms using comparative genomics, focusing our work on the Brassicaceae family. Okay, so we've all gotten kind of a lesson in resilience over the last year and a half plus. And you might have seen a number of these sort of snapshots from Zoom meetings from different speakers or presenters. And this is ours, I think probably a month into the pandemic. And this is my group. Now, we've all tried to be resilient across this time, but we're all humans, and you can see just a couple months later, sort of the change in mood. I think I've changed my hair color about 5 times during the pandemic. And, we're all just a little bit, tired of the changes to our lives that have been occurring. But, when we think about resilience and how we can be more resilient, perhaps a good place to look for resilience mechanisms is from plants. And this is a photo that I often show in my presentations. It's a fig tree that has germinated on the rooftop of a temple in Cambodia. And I'm quite certain that this fig tree seed didn't imagine it would be germinating in such a precarious position. But due to the ability of plants to perceive environmental cues, to regulate their growth and development, and to acclimate to different availability of resources, this fig tree was able to grow and germinate and ultimately thrive and provide a home to many other plants and provide us this beautiful picture as well. So we're interested in these naturally occurring stress resilient mechanisms, and our work is guided to discover them and understand their potential for application. So in particular, we're interested in understanding root-based mechanisms that allow plants to be resilient to stress. We're also exploring engineering strategies to tune these responses to develop more resilient crops. And we're also interested in building better science-society partnerships that support and share the fruits of innovation, because ultimately most of our work is funded by the taxpayer, and we need to provide good evidence this work is ultimately going to help average people live a better life. Okay, but the focus of my work today that I'll present is on exploring these resilience mechanisms across a diverse set of species. Now, I think the past 20 years in plant biology has been really focused on model systems. Arabidopsis, of course, being a premier model organism to understand molecular genetic mechanisms of signaling and stress response, as well as our old friend maize, which has been a fantastic genetic and now molecular genetic system for 100 years. But the problem with these model systems is that they're not particularly stress resilient. And if we want to identify different ways in which plants can tolerate stressful environmental conditions, which will be important, as Julia mentioned, for developing crops that can be resilient to the effects of climate change, I think we need to look more broadly. And so I'm really excited and hopeful that the next 20 years is really going to see a blossoming of our ability to use non-model plants in our studies. Now, Arabidopsis, being a fairly stress-intolerant plant, is actually in a plant family where there is a great diversity of environmental tolerance mechanisms. So here's just a subclade of species within the Brassicaceae family that have had their genomes sequenced. And we can see members of this plant family that are halophytes, so plants that are able to grow under high saline conditions. For example, Shrinkiella parvula, which grows along the coast of a hypersaline lake like Tuz in Turkey. We also have plants that are tolerant to low temperatures, to heavy metals. Importantly, however, we also have plant species that are particularly useful for climate change resilient agriculture. For example, oilseed crops that are going to be an important source of bioenergy are also present within this plant family. So hopefully the discoveries that we can make on these stress tolerant species will be more easily applied to these close relatives that are useful for bioenergy. So the exploration of this plant family and the innovations that this plant family brings to our understanding of stress resilience was initiated. This project was initiated by a really talented former graduate student in the lab, Ying Sun. She's now a postdoc at Salk Institute. And she wanted to develop comparative genomic approaches that could be used to begin to compare the stress tolerance mechanisms across these species. This was in collaboration and consultation with Dong-Ha Oh, who is a research scientist at Louisiana State University and our bioinformatics guru. And if you want to learn more about the project, we have a preprint of the work now in bioRxiv, and with any luck, hopefully we'll be seeing it out in short time. Okay, so there are a lot of species in the Brassicaceae family, that's for sure. We're not going to study all of them. How do we identify those that are going to be the best starting point for our exploration of stress tolerance variation? We chose a panel of 4 species in particular that had similar genome sizes and also similar numbers of predicted coding sequences within those genomes. And what that allows us to do is to be able to identify 1-to-1-to-1-to-1 orthologs of genes across these different species. And that really facilitates comparative genomics, because now we can be confident we're comparing the same ortholog across all of these different species. In this panel of 4 species, we have, of course, our old friend Arabidopsis thaliana. And this is essentially an outgroup for 3 species. Sicymbrium ereo, which is also a non-salt tolerant species, and then 2 salt tolerant species that are thought to have evolved salt tolerance independently. We have Shrinkiella parvula and Eutrema salsiginea. Now, what do we focus on to try to understand the variation in stress tolerance? Well, some of the challenges come from making these cross-species comparisons in that these different species, of course, have different tolerances to stress. And what might induce a stress response in one species might kill another species. And so we don't want to be studying the transcriptional profiles of dead plants. We want to understand their differences in signaling capacity and response. And so we decided to focus on a signaling molecule, a hormone, that acts as a proxy and an important mediator of environmental stress responses. And this is abscisic acid, or ABA. So ABA is a stress hormone that's induced under a variety of different water-associated stresses. Cold, of course, limits water availability because of the freezing of water into ice. ABA levels increase under these different stresses and mediate a number of different changes in growth and physiology that, to a large extent, limit the ability of that plant to continue to grow or assimilate carbon dioxide. These changes in physiology are a mechanism that allows that plant to tolerate or even avoid the harmful environmental conditions that induce ABA. So we're using ABA treatment across these different species as a proxy for abiotic stress. And this avoids some of those difficulties in comparing stress responses, as I mentioned. But an important caveat is that we have a limited understanding as to what environmental contexts might actually trigger ABA in these different species. And so that's an important next step for our future work. Okay, well, we're root biologists, and we started looking at different responses to ABA in roots. So it's been very well characterized that in Arabidopsis, if you treat roots with ABA, you see this reduction in root growth at different concentrations. And we could also observe the same response in Sicymbrium ereo. So no big surprises there. However, when we looked at the halophyte Shrinkiella parvula, we saw a very different response. So even at between 1 and 10 micromolar ABA, we have this very strong growth induction that is occurring in response to ABA. And this response is very similar all the way down to about 50 nanomolar ABA. So it is a robust and strong response to ABA that induces growth that is substantially different than the modest growth induction that we see in other species at low concentrations. Now, is this a halophyte specific type of response? It appears not, because Eutrema salsiginea, which is also a halophyte, has a response to ABA that is actually fairly similar to Arabidopsis and Sicymbrium ereo. Okay, well, we want to understand the mechanism by which this ABA is regulating growth, and growth of the root can be measured in 2 different ways. So continuous growth of the root is dependent upon cell divisions that are occurring within the root tip stem cell region, which is called the meristem. And also root length is contributed by the final cell length, or the amount of elongation, cell elongation, that is occurring in cells that leave the meristem. And so either of these 2 mechanisms may contribute to this change in the way that ABA regulates growth in Shrinkiella parvula. So we looked at this more carefully. We made confocal images of root tips across the different species and measured cell lengths along that longitudinal axis. And what you can see here is Arabidopsis thaliana. On the x-axis, we have the different cell positions along the length of the root, and then we have the length of those cells. And what you'll see is that under controlled conditions here in gray, we have a lot of these small cells. These are cells of the meristem, and then the cells exit the meristem and start elongating, and then ultimately reach a final cell length. When roots are treated with ABA, that meristem decreases in size, but the final cell length is about the same. So this indicates that in Arabidopsis, the primary mechanism by which ABA is regulating growth is by reducing the size of the meristem. This is similar in Cocymbrium aereo and Eutrema salsaginium, where the biggest effect we see is in the change in meristem size, not necessarily in the change in final cell length. This is, of course, different in Shrinkiella parvula, where we see some effects on meristem size, but the biggest effect here is in final cell length. So the final cell length of parvula is much greater than it is under control conditions, and this explains why ABA is able to accelerate growth in this species. Okay, so that provides a developmental mechanism. Now we want to search for a molecular mechanism to explain these differences. And so the ABA pathway has been characterized now in depth, and our understanding of ABA signal transduction is quite mature, thanks to a number of people, in addition to individuals like Sean Cutler, who co-discovered the ABA class of ABA receptors, the PIL-PIR RCARs. Without going into too much detail, these receptors are able to bind to ABA and ultimately lead to the phosphorylation of a specific type of transcription factor called an ABA response element binding factor, or ABF. And this ABF, when phosphorylated, is then able to bind to the ABA response element and activate ABA-dependent gene expression. So we thought this could be a really important point in the pathway to look at. And we wanted to know whether the differences in ABA response that we are observing between species could be explained by differences in the genes that were being targeted by the ABFs in each of the different species. But how can we generate an ABF gene regulatory network across these different species? Well, fortunately for us, really pioneering work from Joe Ecker's lab by Ronan O'Malley and Carol Huang established the DAP-seq method. And DAP-seq is essentially an in vitro method that allows one to interrogate the pieces of DNA or the genomic regions that are bound by a particular transcription factor. So DAP-seq begins with the in vitro transcription translation of a transcription factor of interest. This is then bound to beads and fragmented genomic DNA, and the DNA that is bound is then sequenced. And so because all of this is done in vitro, and because the transcription factors are sourced from sequence, we can do all of this from non-model systems so long as we're able to get genomic DNA from that species. And so this of course enables transcription factor DNA interactions with native genomic DNA. It allows those interactions to be reconstituted in vitro, and it has a good correspondence with ChIP-seq data, as I'll also show later on. And so this facilitates now an ability to create these gene regulatory networks in species that are difficult to transform. So many of the approaches that Julia Bailey-Serres, for example, illustrated, which provide a beautiful insight into cross-species comparisons, are only really possible in plants that can be transformed. Okay, so we found that when we searched the genomes of our 4 species, that there were 4 ABFs in each of the species. So each species had an ABF1, an ABF4, an ABF2, and an ABF3. And we cloned all of these coding sequences and used them in DAP-seq to interrogate how the different isoforms of ABFs are able to interact with their native genomes. But because this is in vitro, we can do something that is really almost impossible in an in vivo context, which is we can swap the genomes and test how sequence differences across transcription factors might affect their ability to interact with the same genome. And so Ying called this swap DAP, where now we can interrogate all the different transcription factors of, say, isoforms of ABF4s across the different species on the same Arabidopsis genome. And this allows us to then interrogate whether there are coding sequence differences in those transcription factors that might contribute to the gene regulatory network differences. Okay, so we did all of these experiments. And what we largely found is that the different transcription factors across these different species bind essentially to the same sequences. So their position weight matrices obtained either by DAP-seq or swap-DAP are very similar, and they have a large overlap in the genes that they target. There are some differences, but to a large extent, they behave as similar transcription factors in the context of ABA response. Okay, so what we want to use this data for is to identify genes where the wiring, where the connection to the ABFs has changed. And so I'll show one example here and then broaden that out. So this is what the DAP-seq data actually looks like. We see, for example, the ability of the Arabidopsis ABFs to bind to the upstream region of this gene, which encodes a DNAJ heat shock protein. We see conservation of that binding in Cimbrium aereo and also in Eutrema salciginium, but this binding is lost in the 5' region of this gene. And through swap-DAP, we can see that it's not because the Shrinkula parvula ABFs are not able to bind to this sequence. They're able to bind to that sequence if we put it in the context of the Arabidopsis genome using swap-DAP. Does this change in binding have any regulatory consequence? Well, right now, all we can do is correlations, but we see a very nice correlation where only in Shrinkula parvula, where that binding is lost, do we see a loss of ABA induction by RNA-seq. And when we go down to the sequence level, we can see that Shrinkula parvula has a change in from a C to a T in a conserved ABRE sequence. So presumably this is the sequence basis for this difference in ABF binding. Okay, well, that's one gene. Can we broaden that out to looking at sort of genome scale comparisons? And so this was an analysis that Dongha developed, and he was interested in understanding this difference in binding between orthologous genes, and he calls this presence-absence variation. So if in one species an ABF can bind, and in another species ABFs are not binding to the ortholog of that gene, what sequence differences might explain that? Well, there could be structural variation, meaning that that binding site simply doesn't exist, or maybe the promoter has been rearranged to an extent that it's no longer possible to see where that binding sequence is present. There might be a SNP, or a single nucleotide based change in the core ABRE sequence that could explain the difference in ABF binding. Or perhaps there might be instances where we can't explain why the ABFs are no longer able to bind because there is a conserved sequence and the promoter architecture is conserved. So we wanted to kind of bin different possibilities into these different categories to see what was most prevalent. So first, we categorized genes into different groupings. So we looked at orthologous pairs of genes and looked to see whether there was an ABF binding in both, and that's here in blue, only in one species, so this presence-absence variation, which is in pink, or there is no ABF binding in either gene. And what we can see is that if we look at overall promoter similarity, that there's a much greater overall promoter similarity between promoters of orthologous genes that are able to be bound by ABFs. And so this suggests that not only is the ABF binding site important, but overall promoter architecture may also be important for facilitating ABF binding. We also then categorized these different variations across genes, and we looked at, sorry, someone was knocking on my door. So in context where one ortholog has a binding event and one ortholog doesn't, what sequence differences might explain that? We saw that the highest fraction of genes varied because of structural variation. So the promoter sequences were rearranged. A close number of genes, we saw a CRE modified. So the actual binding site had a SNP in it that may have affected binding. And a small fraction had a conserved CRE. So this shows that structural variation and SNP variation may be a prevalent way in which the gene regulatory network is rewired. Okay, so taking our data, we curated a core ABA gene regulatory network and looked at how ABFs were interacting with the promoters of a number of different genes across different biological functions. And this network was based on a gene regulatory network for ABA that was developed by Liang Song in Joe Ecker's lab. Now that network was based on ChIP-seq, and we find that our network overlaps in terms of the connections by about 89%. So very similar results were obtained whether you use ChIP-seq or DAP-seq. Now in this network, we don't see a lot of changes. Only 10 connections here are changed in parvula. And of course, that's the species we really want to understand how that rewiring led to that growth induction. And so it doesn't seem that the core network is going to be the most important place where that variation is occurring. So we also generated curated networks for ethylene and auxin and looked at how many changes within that network were Shrinkula parvula specific. And so that's shown here in this graph. So we see different percentages of the nodes vary in Shrinkula parvula, and that may be based on a number of different criteria used to build the networks. But what's I think most interesting is that for the auxin network, we have a much higher percentage of connections that change only in Shrinkula parvula. And remember, that's the only species where we saw this growth induction by ABA. So we focused on auxin, and this network shows all of the connections that have changed in parvula. What I'd like you to focus on is this subpart of that network that regulates auxin biosynthesis. So TAA1 and YACA2 are two genes that encode enzymes important for the last steps of auxin biosynthesis. We see that in Shrinkula parvula, the ability of ABFs to bind to the promoters of these genes is lost, but it's present in all the other species. And this leads potentially to a change in gene regulation where we see, YACA2 and TAA1 show a loss of ABA induction as a consequence, whereas we see a strong induction in Arabidopsis. Okay, so how can we put all of this together into a potential model? Well, in Arabidopsis, we see that ABA, of course, leads to the induction of ABF function that then, based on our gene regulatory network, directly controls auxin biosynthesis, and we would hypothesize then that auxin biosynthesis may inhibit growth. And that's consistent with the effects of auxin treatment, for example, on root growth. In parvula, this ABA would still presumably activate the ABFs through phosphorylation, but these ABFs are no longer able to bind to the promoters of these auxin biosynthesis genes and no longer able to lead to this growth inhibition. Do we have any evidence for that? Well, in Arabidopsis, we can look at mutants that are defective in auxin biosynthesis. For example, this WAY8 allele is essentially a mutant allele of the TAA1 gene. And whereas wild-type Arabidopsis, the growth is inhibited by ABA treatment, in a WAY8 mutant, this growth inhibition is strongly reduced in WAY8. We can also use a drug, ELK-KYN, which inhibits this same class of enzyme. And we also see that this treatment leads to a reduction in the ability of ABA to repress growth. We can ask the question the other way: can we make Shrinkula parvula more like Arabidopsis? Well, here what we can do is we can do co-treatment with ABA and auxin. And what we see is that with ABA alone, we see this nice growth induction in Shrinkula parvula, but if we supplement with auxin in these conditions, we can inhibit that ability of ABA to promote growth. So that backs this, this hypothetical pathway, but there's still an important part that we haven't discovered. What is it that allows growth to be accelerated in Shrinkula parvula? And so we're actively trying to discover this pathway. In order to do that, we're expanding out our universe of species. So really talented postdoc Prashant Ramachandran is looking at ABA responses and growth across a large number of species, and we see tremendous variation in how ABA regulates growth. And we're also generating the ABF GRNs across these across 11 sequence species in the breast KC family. We're collaborating with John Schiefelbein, Maheshi Desanayake, and Song Lee in order to generate single-cell RNA sequencing data sets for these 4 species, and that is allowing us to then explore cell type-specific innovations. And we're performing anatomical analyses of all of these different species, looking at stains such as Nile red, which is a lipophilic stain that stains suberin and other hydrophobic molecules. And we see interesting variation in the pattern of these hydrophobic molecules across our species of interest. So to summarize, we've seen that ADA has diverse effects on root growth across the Brassicaceae family. We've seen that DAP-seq is a really useful and reliable tool for cross-species analysis of GRNs. We see that changes in the way that auxin biosynthesis is regulated in Shrinkula parvula likely underlie the differences in growth regulation by ADA. And through our collaborations and anatomical analyses, we're hoping to discover new cell type specific innovations that differentiate these species and may have potential for application. So I've thanked the folks along the way, but I really like to thank my group as a whole. This was a recent pumpkin festival where we dressed up as characters from Sesame Street. And so this work was brought to you by the letters HHMI, NSF, and DOE. Thank you very much for your attention, and hopefully I'll have time for at least one question.
Thank you so much, Jose. It was so interesting talk, very intrepid approach. I think the swapping approach, that's very nice. So let's see if we have questions. I don't think so. Okay, so we are on time, so we'll finish here. Thank you so much for accepting our invitation, and I hope to see you soon. Thank you
Absolutely.
so much.
Thank you.
Thank you.
Thanks, Alejandra. Our next speaker is Professor Verona Montgomery. She's coming from Michigan State University, and she's going to talk about lessons about and from plants, plant color vision, and developmental acclimatization. Please go ahead, Verona.
Thank you. It's a great pleasure to be invited to give a talk today. I am going to tell you today a little bit about some recent studies in our labs to learn about how plants are responding to light, how their color vision works, and also some lessons that I and others in collective community have learned from them. So in the lab, we study primarily on the plant side Arabidopsis, which undergoes, like most plants, the phenomenon described here or depicted here of photomorphogenesis. And this is a process by which plants that are growing in the dark have a very different appearance than plants that are growing in the light. And this is behavioral adaptation that's largely driven by the energy budget that the plants have in these two conditions. So these plants are growing on the same media and have access to mostly the same environment. But the primary difference is that all things being almost equal in terms of what water, nutrient profile, temperature, humidity, and other environmental factors are present, the plants shown here have been growing in the dark, and in the dark, energy goes into rapid elongation of the stem or the hypocotyl to try to project the plants into light. In the presence of light, plants have the critical access to light that they need to drive photosynthesis and the production of sugars. And in this case, energy instead goes into other organ development. So in the light, plants actively inhibit elongating the hypocotyl, and instead energy goes into developing the leaves, which function as the equivalent of solar panels for harvesting light, and in the presence of simple compounds like carbon dioxide and water, are used to drive photosynthesis to make sugars. Also, light promotes root development to promote uptake of nutrients and water from the growth medium. And so this behavioral adaptation driven by the energy budget in response to light is known as photomorphogenesis. And we and others have been interested in the process for years to understand specific light absorbing proteins or photoreceptors that are responsible for these changes that we see in growth in the dark relative to light. And we know that there are multiple classes of photoreceptors that are used by plants to absorb throughout the visible spectrum that allow them to detect the comprehensive light spectrum, both light quality or color that's available, as well as light intensity. And the output of these photoreceptors collectively then drives various aspects of plant growth and development. So three major classes of photoreceptors are depicted here: the phytochromes that absorb in the red far red region, two classes of blue UVA absorbing proteins, the cryptochromes and phototropins, as well as the most recently characterized at the molecular level photoreceptor UVB receptors, UVR8. And as I stated, all together, these function to give plants a comprehensive view of the light spectrum. And that drives different aspects of plant growth and development, from the emergence of the seedling from the seed and germination to the process of de-etiolation, which is that change that was depicted on the prior slide that we see for plants grown in the light versus those grown in the dark, all the way to the shift from vegetative to floral growth, as well as ultimately senescence, as well as a number of growth aspects. In my laboratory for years, we've been very interested in the role of phytochromes in these light-dependent growth processes. Phytochromes have two major components. There is a protein component, the apophytochrome, that is encoded by nuclear genes. And then the chromophore, or the light-absorbing moiety that's a part of phytochromes, is encoded, it's synthesized from two committed enzymatic steps, where a circular tetraparole heme is cleaved by a heme oxygenase to make biliverdin, and then that gets reduced to make the chromophore. This chromophore binds to a chromophore binding pocket on the apophytochrome, which has the activity to then drive covalent attachment of this chromophore to a conserved cysteine residue to create the holophytochrome, shown here in the dimer states in which they function, both homo and heterodimers, in the red light of ground state. And so we've been interested in the function of these in planta, and we're very interested in how these function as a family, but also how they function across time and space. So we do know that plants have families of phytochrome. Arabidopsis, which we study, has 5 phytochromes, 5 nuclear encoded genes, A, B, C, D, and E, whom they all use the same chromophore, phytochromobilin. And this has been interesting because there are 2 classes of genetic studies, genetic mutants that have been identified that have helped us understand the role of this family in plant photomorphogenesis. So you can imagine having mutations in any of the 5 nuclear genes. As depicted here, if phi C is mutated, you isolate a mutant, and the characterization of that mutant and the differences in phenotype relative to the parental wild type gives you an indication of what that particular phytochrome family member contributes. There are mutants in all 5 of the phytochrome. The other class of mutants that have been identified are chromophore deficient mutants. And so there are mutations in either the gene encoding one of the heme oxygenases or the gene encoding the biliverdin to phytochromobilin, that phytochromobilin synthase. And in these cases, chromophore is missing. As all 5 of the phytochromes use the same chromophore, you have global phytochrome deficiencies in these particular plants. And this is important for the approach that we've taken to understand a subclass of phytochrome responses. So we have been very interested in my lab in tissue and organ-specific phytochrome responses. And that is the idea that light has different impacts on plant growth and development depending upon the organ which is exposed to light. So as compared to dark-grown seedlings, where there is limited development of the cotyledon, and most of the growth and development is in the hypocotyl, in the light, you have active promotion of growth in the cotyledon as well as the development of the roots. So light promotes growth and development in the cotyledons and the roots. Compared to the dark, however, there's an active inhibition of growth in the hypocotyl for light. So light can either promote growth in one organ or tissue or inhibit growth. And we've been very interested in how a single signal is perceived by a family of photoreceptors and either promotes growth or limits growth depending upon the tissue to which light is exposed. Because there are 5 phytochromes, one hypothesis might be that there's one phytochrome in the hypocotylar stem that inhibits growth and distinct family members that promote growth in the leaves, cotyledons, and roots. But what you find when you look at plants and ask where the phytochromes are either expressed in terms of where the messenger RNA is accumulating or where the proteins are accumulating, in both cases going from low levels of protein or messenger RNA in blue or high levels in pink to red, either solid bars for messenger RNA or striated bars for protein. What you find, and although this is a busy image, all you need to see is that if you look at what's happening in the shoot, the hypocotyl roots, there's no one phytochrome that's in the hypocotyl that would be associated with inhibition of growth and others in the shoot, but you find some level of all 5 in the shoot, hypocotyl, and root. And what this indicates is that whereas we know that the phytochromes are controlling these tissue-specific growth aspects of either promoting growth in one organ or inhibiting it, that means that there has to be something that's happening different of downstream of the phytochrome. So tissue-specific phytochrome signal transduction pathways that are promoting growth in the shoot, and a distinct pathway using different effectors that would then inhibit growth in the hypocotyl. And one of the questions we've been asking is how we can get at understanding what those distinct tissue-specific or organ-specific phytochrome-dependent pathways are. And so we've taken an approach of doing transcriptomics to ask if we look at, for example, comparing seedlings that are grown in the dark where there's active promotion of growth in the hypocotyl versus those in the light where there's an inhibition. If we isolate messenger RNAs from that, can we see the genes that are differentially regulated in those? And then identify genes of interest, target those for genetic studies. But if we really want to get at understanding how the phytochromes are controlling these organ-specific processes, the approach we've taken is to ask if we can metabolically control synthesis of the chromophore in vivo. If we can ablate accumulation or synthesis of the chromophore in particular cells, we can create a local phytochrome deficiency, because as I indicated, all 5 phytochromes use the same chromophore. And so we have indeed taken the approach of expressing a gene in plants that targets the key precursor of chromophore synthesis, biliverdin, and inactivates that or reduces its accumulation, thereby creating a local phytochrome deficiency. We then ask if we can remove the phytochrome in particular cells, can we create these tissue-specific disruptions of phytochrome accumulation? And then ask how we've disrupted particular signal transduction pathways. So we've done this in a number of organs, for example, expressing the gene in cotyledons or leaf tissue to inactivate phytochrome accumulation in these tissues and ask how do we disrupt the signal pathways that are needed to promote growth in that organ. Likewise, we have inactivated it in other tissues, in the hypocotyl, in the root, very specific tissues such as stomata and other kinds of specific organ ablation of phytochrome accumulation. We then can ask, how do we disrupt these signal transduction pathways through using omics approaches such as transcriptomics or proteomics? And in cases where we have inactivated chromophore accumulation, here I'm showing you a line where we have expressed a reductase of the biliverdin to remove that key precursor for phytochrome chromophore synthesis. And if we do that in all cells and all tissues using the 35S promoter, these are both seedlings grown in white light, compared to wild type that has the short hypocotyl where it's being inhibited from elongation, but we're promoting leaf formation and root development. In these lines where we remove the chromophore, you get something that looks very similar to a dark grown seedling, where there is no inhibition of the hypocotyl, so there's active growth of the hypocotyl, and limited development of cotyledons and promotion of leaves, as well as the root development that is characteristic of growing in the light. So we were able to show that this approach works. This looked very much like a natural chromophore deficient mutant. And then we moved on to using tissue-specific promoters as well as enhancer trap approaches for inactivating chromophore synthesis in the cotyledons or other organs. Once we have lines that we then have a disruption in the active phytochrome profile, we can use seedlings that are grown in the same light condition and ask if we disrupt the chromophore synthesis, are we then able to look at messenger RNA accumulation in either a particular organ such as a hypocotyl and ask how we've disrupted that a localized inhibition of growth in the hypocotyl versus the promotion of growth in cotyledons. And so we have done that, identify genes that are differentially regulated when we target a pool of phytochromes, and then to target those genes of interest for further approaches, mostly through isolating tDNA lines from the Arabidopsis stock center and asking if we have identified a gene that we can then place in an organ or tissue-specific phytochrome-dependent regulation of a particular growth response. Here I'm showing you 3 lines that when we looked at if we inactivated phytochromes in cotyledons, we were asking, do we disrupt genes that are important for local cotyledon growth or for communicating with the hypocotyl in terms of inhibition of hypocotyl elongation? And so here are just a sample of genes. I'm only showing you a few. We have we isolated hundreds of genes, have characterized many of them, and I'm just giving you a sample of some of the types of results we get. Here we identified 3 genes where if we looked in wild type where the gene was expressed, these are cotyledon-enriched genes. And so the messenger RNAs using qRT-PCR are differentially accumulating in RNA from the cotyledon versus the hypocotyl or root for all 3 of these genes. And when we remove phytochromes from the cotyledons, these genes are downregulated. So we've identified genes that are upregulated specifically in the cotyledon when phytochromes are activated. And this was a previously uncharacterized gene, and 2 others of these are genes that we have some indication of what their function may be. The CYP-K20 has a role in calcium calmodulin signaling, which has been implicated in phytochrome signaling before. And then we recognize the GCN5 transcriptional-like regulator, which means that the phytochromes are controlling cascades of gene expression. When we isolated mutants in each of these genes to ask if they may have roles in specific aspects of the plant growth and development, what we identified doing some analysis of what the seedlings look like. And in this case, I'm showing you a single allele, although we had multiple alleles for each gene. And in each of these panels, you're looking at 3 wild-type seedlings grown in far red light versus mutant seedlings that are grown in continuous far red light. And for each of these genes, what we identified is that when the gene is mutated, the phytochrome inhibition of hypocotyl elongation was disrupted. So each of these mutants had significantly longer hypocotyls than wild type. And while these are having smaller effects than if you knock out the phytochrome itself, here we're seeing a 10 to 15% increase relative to what we would see as a 100% increase perhaps in a phytochrome mutant. We're identifying that multiple genes are functioning downstream of the phytochromes, expressed in the cotyledon, and then involved in a previously known interorgan signal to inhibit hypocotyl elongation. We likewise found other genes. One interesting gene that we identified that was downregulated when we removed phytochromes from the cotyledons and green tissue specifically was that there was a sigma factor, a nuclear encoded sigma factor that was downregulated. And so this particular sigma factor had been previously implicated as having a role in greening. It was isolated in a yellow-green screen. And we know that these sigma factors are encoded in the nuclear genome. They're then imported into the plastid, where they work with the plastid RNA polymerase to tune plastid gene expression. So this was presumed to be part of a light-dependent induction, phytochrome associated with this, of light induction of plastid development. And here we identified specifically that phytochromes were functioning to control this sigma factor. When we looked at seedlings at the bottom, these are dark grown seedlings, 3 again for wild type, and we have 3 sigma 2 mutants, 2 strong mutants, sigma 2-2 and 2-4, which have basically no accumulation of the sigma 2 messenger RNA, and a weak allele that still has 70% of wild type levels. When we're going up toward the top of the slide, for seedlings that were grown in blue light, we saw that the previously identified chlorophyll deficiency in sigma 2 was apparent. But we noticed something specific, that the sigma 2 factor appeared not only to affect plastid development, but also had a role in phytochrome-dependent photomorphogenesis. Because in red continuous light, in addition to the yellow-green phenotype that we see, clearly you see elongated hypocotyls for the strong sigma 2 mutants. And likewise, we did see elongated hypocotyls in far red light. We presume that this was specific to phytochrome because in blue light, where the cryptochromes are active, there's only the yellow-green phenotype, but not an impact on hypocotyl elongation or the cotyledon size and root development. So here we identified that a previously identified factor having a role in greening is regulated by phytochromes in mesophyll cells, presumably, because that's where we've knocked out their accumulation, but also because it's affecting photomorphogenesis. And so we started to be able to tease apart some of these tissue and organ-specific phytochrome signaling pathways that have critical roles in plant growth and development. So all of these studies from our lab and other labs have been really teasing apart what happens in the dark that allows the plants to focus on elongation. So when the photoreceptors are not activated, there are a set of signaling intermediates and signaling pathways that promote the elongation of the hypocotyl and inhibit really progressive development of leaves and root tissues. And several photoreceptors are functioning in the light. We focus on the phytochromes. Different signaling intermediates are impacted, a different set of signaling pathways. And what my lab has really been layering onto the work that other labs are also doing is to really add to this idea that these seedlings are perceiving light, changes in the environment, the gene networks are being modulated, and there's an outcome. But we have also been looking at what are the tissue-specific pathways that lead to promotion of growth or inhibition of growth specifically. So I wanted to pivot in the last few minutes just to share not only what we have been learning about how these seedlings are functioning in their environment to perceive light and tune their growth and development very specifically across tissues and organs by modulating their molecular networks to promote the appropriate outcome, but also the ways in which we have looked at the similar types of differences in photomorphogenesis to ask questions about what we can learn from this, not just at the scientific level, but also about being scientists in community. And so I've been very interested in how we can continue to fill out these signaling networks to contribute to what we know about this, but also what we can learn from this. And I have been really promoted to think about the ways in which we have learned a lot about the critical importance of the environment on growth and development of seedlings that otherwise start at the same place in terms of their aptitude, and how the external environment and the ability to perceive what's going on around you is as critical as being able to adapt your internal networks to have the appropriate outcome. And asking if we can learn things about this, about how we position ourselves to be supported, but also serve to support students and colleagues. And so this very same image has served very much as a motivator for me to understand about plants, as well as to understand how we support communities that are learning about plants. And so one of the things that I have really taken as a different perspective is that too frequently in community, we focus on individual ability and individual deficit. And we don't take the understandings that we have about the critical interface between individuals and the environment into the ways in which we engage and support each other in community. And I've really been motivated over the years by the ways in which, as humans, our expectations that plants should grow really changes the ways in which we engage with plants. And so humans, by and large, not only those who study plants like many of us here today, but the human population, by and large, believe that plants should grow. That's the default expectation. And if we have plants in our environment that are not growing in the pattern that we anticipate, the questions we start to ask ourselves is whether this organism has everything that it needs. Most humans would look at this plant and say that it may need water. If you live in a very hot and humid environment, you may say that the temperature is not optimal. Likewise, people who grow plants in their homes or gardeners, certainly those of us who are plant biologists, look at plants such as this and start to ask questions about what's missing in the environment that this plant doesn't have access, likely to the nutrients that it needs to green. We don't look at such a plant and say it's incapable of greening. We likewise don't look at this plant and say that it should work harder at soaking up the water from the soil. We ask if someone has prepared it by transferring water from a faucet to its soil. And so when plants are not faring well, we ask questions about what's missing in the external environment, as well as asking questions about the caretaker and whether the caretaker has the skills to interpret what's going on and to mediate what's missing in terms of the critical factors. And so this has really promoted me to think about how that should stimulate us to think differently about mentoring and leadership, and what specific mentoring lessons we could learn from our understanding of the importance of the external environment, as well as plant caretaker abilities for promoting our expectation of success for plants. And so I have really drawn specific lessons that I thought we could learn from this. I'll share just one with you today, but it is this idea that when plants depart from our default expectation that they should grow and be healthy, we probe the environment first and extensively to ask if something is missing, as well as probing our caretaking abilities. When we talk about students and colleagues, when they are struggling, whether it's struggling to get through classes, struggling to get grants funded, struggling to get papers published, we often assume that there's a personal or individual deficit. And I think that our engagement in community would be well informed by expecting growth for each other, expecting the possibility of success, and where there's a departure from that, asking questions at least equally about what the individual may need to strengthen about themselves, but also asking questions about whether everything needed to support success is available in an environment, and or if it's there, but there is not a steward that's made the individual aware that the critical factors are available to them. So I think we need to ask questions first and systematically about how the environment interfaces with the individual to see if success can be promoted. And so what we generally do with individuals is very different from what we do with biological organisms. And often we start by judging the outcome. We ask whether students are routinely performing well in classes. We ask whether colleagues are routinely publishing high impact papers and success with grant proposals or other such expectations. And based on the outcome, if they are routinely performing as we expect, we judge them as high achievers. We say that they're geniuses. If people are routinely struggling, we often attribute that to an individual deficit, their inability to cut it in an environment without asking whether everything is there needed externally to support them. Frequently, when and if we decide that individuals do need advising or mentoring, we often do that through an assimilation-based lens of telling them what they need to do to fix themselves to demonstrate success. And I have simply argued that we know when we work with biological organisms that success or lack thereof is about them being in an environment that has everything they need and their ability to perceive that and modulate their networks accordingly to lead towards outcomes. And I think that we would be well informed to move away from these deficit-based engagements with individuals to using a similar model that parallels what we know happens in nature over and over. And that's to understand that individuals are in an environment. There are either the factors they need there to support them or not, but what is our role as mentors, leaders, as peers to help them perceive what's needed and to modulate the networks. And in this case, the networks are networks of support, networks of development that lead then towards outcomes. And so I really learned all of these things from the plant biology knowledge with which I sit, but not leaving that knowledge in the papers that I publish or on the bench, but asking what knowledge can we take from that to also inform the ways in which we position ourselves to be helpful in community. So I've written about this in other lessons in a recent book that came out earlier this year in April, Lessons from Plants. And so I think that I will stop here and just thank my group, thank my colleagues, thank the funders, NSF, the Department of Energy, and MSU Foundation, and be happy to take questions if there is time left.
Thanks, Brenda. That's a very nice talk. So we have time for some questions. So, anyone for questions?
Felipe, they are asking if they can repeat the name of the book again.
The book is Lessons from Plants from Harvard University Press.
Thank you. Okay. If we don't have questions,
there is a comment by Alma Piñeiro, if you want to read it from the comments.
Okay.
I see. Yeah, so there is a comment here that says we might be biased by the survival of the fittest metaphor to perceive this idea that we should be supporting each other. That's certainly one of our challenges. I think one of the challenges is that it's hard for us to both recognize that admitting that the environment may not be optimal for everyone doesn't take away from the fact that we were successful as individuals. And I think it is hard for us to challenge that. It feels like if we're saying that we had unfair advantages or unfair privileges, that maybe we didn't earn the success that we have. And I think we can have both and. We can recognize that we had to work very hard to have success, and yet the current environment may be requiring some to work much more than others. And so I think that there is this struggle of both acknowledging the work that we've done to get where we are without falling into the trap of presuming that the environment is perfect for everyone. And that's a real challenge to get people to change because it hits against our own questions about how we got to where we are, how we got to be successful. And I think we have to give ourselves grace that we worked hard, but there are changes that are still needed to expand the environment for those who have access and ability to succeed.
Yeah, Veronda, there is a question from the chat. She's from Nayeli March. And she's asking, do you think that only light and also another environmental cues affect organs differentially?
Yeah, so certainly there is evidence from people who study certain nutrients, temperature, that there are certainly organ-specific effects in response to other environmental cues, very similar to light. I think there's a hand up. I don't know if Ulysses, do you have your hand up?
Ulysses?
Yes, Felipe. Yeah, Veronda, thank you for your talk. It was very, very nice. Made me think about a lot about what the last things that you were saying. And particularly, I was thinking that perhaps we should change the way we approach plant biology, because as you were saying, we have to look at the heterogeneity of the people who we are working with, but also thinking about what Julia was speaking at the beginning on natural variation, because we are normally very interested on highly homogeneous lines, homozygote lines, but perhaps we should study traits in a more realistic way, taking into account that there is natural variation, right? So I don't know if you have any take on that, Veronda.
Yeah, so I think, yes, I think that, certainly we are primed to study homogeneity for the reasons that we get reproducibility. I think the most powerful analogy that I've had about this is actually not from the lessons from plants, but from the lessons from microbes, and how, I happen to be a part-time microbiologist, but microbiologists in particular are very fascinated with bacteria that we know exist in nature, but we don't know the right growth media yet to get them to be cultivated in our laboratory spaces. And so often I have made the analogy that many of our educational and science environments are the equivalent of LB. They are really built to support a relatively narrow range of individuals. And I think one of the things we have to do is to ask, how do we have a range of flexibility in terms of expectation, but also the environments of support that are there, as opposed to kind of fixed ideas about what leads to success, as well as what counts towards success. So I agree, we have to have to appreciate natural variation, but also understand what it means then to have microenvironments where those individuals that come with this range of different natural contexts will survive. There was another question in the chat, and then I think we have to move on, but there was a question about...
We are very tight with the time.
Okay, awesome.
Sorry,
okay.
Thank you very much.
That's okay. Thank you so much. I appreciate the opportunity. Thank you.
Thank you. So our next speaker is Professor Jorge Nieto from the Institute of Biology, UNAM, and he's going to talk about analysis of mesocotyl length variation in maize by combined QTL transcriptomic and genomic-wide association studies. Go ahead.
Hi.
Okay.
Okay, let me share my screen. Can you see that?
Yeah.
Okay, well, thank you very much, Felipe. And I thank all the organizers of this wonderful meeting for giving me the honor to share with you what we have learned in over many years of work in my lab on mesocotyls in maize. So let me first tell you why we are studying the mesocotyl growth in maize. Well, we would like to learn more about the transition from scotomorphogenesis to photomorphogenesis. Scotomorphogenesis are the strategies that plants take developmentally and growth-wise after germination in the dark below the soil, so that they may spend either one day or actually a few weeks growing under the soil until they reach the light. Once they reach the light, they reprogram their growth and their developmental patterns with the so-called photomorphogenesis pathway. But before they do that, they have to spend a lot of energy and strategies to really develop to reach light through the scotomorphogenesis pathway. We also are interested in this because we would like to learn more about intercalary meristem activity and arrest, because the meristem that directs the growth of the mesocotyl while growing in the dark has a very actually short half-life. It's just a few, at the most, weeks. It doesn't last for the whole life cycle of the plant as like the apical meristems of the roots or the shoot. We would like also to learn about seedling emergence and plant establishment in the soil, which is a very important stage in the life cycle of a plant. We would like to understand also the interaction of genetics with agricultural management. As you will see, the mesocotyl is highly involved in plastic responses and and selective pressures for its genetic variation that controls its growth due to agricultural management. And we would like to study the evolution of seedling development in grasses, focusing in part on mesocotyl and basically on the whole scotomorphogenesis pathways that plants undergo. So let me change my slide. Okay. All right. So what is the mesocotyl? The mesocotyl is the basal part of the shoot. What you are looking at here is a one week old seedling of maize that has been grown in the dark. And the basal part of the shoot, we are not seeing at all the roots. We are looking at from the kernel all the way up the shoot. So the basal part of the shoot is the mesocotyl, which is an organ that connects the coleoptile node, which is up here, with the scutellar node, which is right here in the caryopsis, and you don't see it. Above the coleoptile node, you see the coleoptile, which contains the primary leaves of the seedling, and they still haven't broken the coleoptile. They haven't reached the light yet. And you see just below the coleoptile node, the intercalary meristem that is responsible for mesocotyl growth. So mesocotyl growth requires, of course, the cell proliferation or cell division here in the meristem, and also the cell elongation. So the role of the mesocotyl is very important because the mesocotyl pushes up the coleoptile until it reaches the light and emerges from the soil. A lot of work has been done on which factors control mesocotyl elongation in different grasses. And this is just a summary of decades of work of many labs. We know that CO2 promotes mesocotyl elongation, while light, either red light through phytochrome B or UVB light, inhibit mesocotyl growth, as well as heat and low water potential. There are also internal factors that regulate mesocotyl elongation, mostly plant growth regulators, which inhibit mesocotyl growth, such as ABA, nitric oxide, and strigolactones. On the contrary, ethylene, auxins, brasinosteroids, and gibberellins promote mesocotyl elongation in the dark. Now, before the plant reaches, let me remove, sorry, because I have a problem here with, well, before the plant reaches the light, this gene LACY1 promotes auxin transport basipetally from the coleoptular tip towards the mesocotyl, and that increases promotes mesocotyl growth. And once the coleoptular tip reaches the light or emerges from the soil, light activates phytochrome B, which in turn inhibits LACY1, and this auxin flow stops, preventing further growth of the mesocotyl. So the mesocotyl importance is, of course, prior to seedling emergence. You don't want the mesocotyl to grow further once the plant reaches the light. Okay, what is the relationship between mesocotyls and agriculture? And this is very important. About more than 100 years ago, Collins made a very important discovery. He was a great botanist. He was traveling all the time in many places of the world, mostly the US Southwest and Mexico and Central America. But while he was visiting Arizona and New Mexico at the turn of the century, he realized that the Navajo and the Hopi people in these areas were able to cultivate corn basically in the middle of the desert. And he wondered how is that they do that? And what he found after spending several months with them and doing a very nice ethnobotanical work, he found out that the people there used lands that have reasonable good humidity under the topsoil. Even though there was not much rain, the humidity was there, and they knew where they could find this type of lands. And they were also doing deep planting of their seed. And we're talking about 10 to 40 centimeters depth. And most importantly, their maize varieties displayed, of course, deep planting resistance. And what he found was that what was more remarkable between their maize varieties and a normal maize variety was that the mesocotyls were growing very long, such that the coleoptile reached or emerged from the soil much easier than a normal variety, which actually most maize varieties cannot emerge from the soil at these depths. So, okay, this is the kind of landscapes where this type of agriculture takes place. On the left, you see a view of the Arizona desert at the Hopi Reservation. And on the right, you see in Mexico, there is many places where this type of planting technique is used traditionally to grow corn in the absence of rains or prior to the rainfall periods, like the Mixteca Alta region in Oaxaca, which actually is a very dry region and very with a very high natural erosion rates, one of the highest in the world. So people really take care of the land to prevent the natural erosions of the place. They create soil, they create or they procure that the humidity is maintained in their soils. But they do the deep planting technique to take advantage of this humidity in the soil and to start grow their maize prior to the rainfalls. On the left side, you see this Hopi maize growing in Arizona, right there in the absence of any water outside, or this seedling emerging in the Mixteca Alta region, again in the dry period of the year. In Mexico and many places in the highlands, the starting planting dates are 1, 2, or up to 3 months before the rainfalls. In my lab, we have been studying a lot of these agricultural management practices, collecting native seeds from different parts of the country, and collecting all the management both of deep planting, how they take care of the soil, planting dates, etc., so that for each accession, we have a lot of data about how they do their agriculture. In the lab, we can assay the scotomorphogenic growth of each accession under controlled conditions. We use chambers where there is no light and where we can grow for up to a week our populations of maize, and we can control the planting depth. And afterwards, we can measure all the organs of the seedling to see the growth patterns and developmental patterns that they carry under growth in the dark. On the left side, you can see this picture of how behaves a deep planting variety from Oaxaca that shows these very long mesocotyls. Here you see the level of the soil in our assay in this bar, the red bar. And in comparison, you can see how modern maize varieties like hybrid corns have very short mesocotyls, and some of them have a very hard time or they never emerge from these assays, which actually are not very deep. And when they emerge, they do it differently. Basically, they can, because of the short mesocotyl, they grow their primary leaves and they start the growth of the internodes of the plumule, so that they emerge through the action of the growth of the leaves, not the coleoptile as deep planting varieties do. This is a very inefficient way of emerging, and actually in real life, very few plants can emerge through this type of emerging pathway. We have assayed many accessions of land races across Mexico, including the Hopi maize, And in our assays, here we are showing the average of a population of seedlings from each of these different accessions. In blue, you can see the average length of the mesocotyl for each accession. Red is the coleoptile size, and green is the size of the emerged leaves that break the coleoptile. And what you can see here is that there are several varieties that have this very extreme capacity to grow very large mesocotyls, like the Hopi maize, this chalqueño variety from Oaxaca, another chalqueño from the state of Mexico, this cónico from Tlaxcala, and this palomero toluqueño. So just remember, the palomero toluqueño is one of also of the champions of mesocotyl elongation. I'm going to go back to palomero later on in the talk. We can also, after analyzing these all growth patterns of all these different organs under scotomorphogenesis and knowing how these accessions were handled in their place of origin, we can actually separate these growth patterns very clearly in two large groups. On the left side, all the dots and lines which are marked red, orange, and green belong to deep planting accessions. These are individual seedlings. We are talking about thousands of seedlings that we have analyzed. And on the right side, you see the blue and light blue accessions or seedlings that belong to accessions where they carry shallow planting. So this type of analysis tells us that management practices, in this case, planting depth, can really have an impact on how, on the genetics of scotomorphogenesis growth. And also we can use this type of assays or analysis to really predict or recommend which kind of varieties are more able to survive or to be adapted for deep planting. We have also tested the wild ancestors of maize, both the Teosyntes as CMAs parviclumens and the highland Teosyntes CMAs mexicana, as well as sister genus, which is also an endemic genus of Mexico, which is Tripsacum, different species of Tripsacum. We have tested almost all of the species present in Mexico. And what you can see here is that the capacity of exaggerated growth in the dark of the mesocotyl is present in the wild type accessions, including even some species of Tripsacum. So basically this long mesocotyl trait is an ancestral trait. It was not really developed by men. It was just kept in the population, and in some areas of the world, they keep these traits under selection. So now we wanted to understand how deep planting management influences the physiology and yield of maize under cultivation. We also wanted to understand what's the genetic basis of deep sowing resistance. To answer the first question, what we did was some experiments under controlled conditions in experimental agricultural stations. We were honored to have the opportunity to work with people at CIMIC. They gave us a lot of areas to do our experiments, and we used some hybrids developed by CIMIC. So the idea was to separate the genetics from management, and for doing that, we just took several hybrids, and by using one kind of hybrid, you can make sure that basically the genetics of all the individuals of that population is basically the same. So the only differences that we tested were planting depth, and we evaluated this under irrigation or under drought. And at the end of after harvest, we measured different parameters, and we compared what was the influence of planting depth. So basically, we observed that the architecture of the stem and the root system is more complex when the planted at really high planting depths. At midday, the leaf temperatures were always lower in those hybrids that were planted very deeply, and leaf senescence was delayed very much in those that were planted deeply. There was also an increase in flowering time, both in male and female flowers. So together, all these phenotypic observations indicate that plants that are planted deeply suffer less stress. Basically, they are avoiding stress. We also observed that the weight, length, and width of the cup was higher. The number of grains per cup is higher. The weight per 100 kernels was higher in those that were planted deeply and under drought stress. And most importantly, the yield in tons per hectare of these hybrids increased by with planting depth, even in the presence of drought. So basically, you see a shallow planting depths. You see here the effect of drought, from very high yields with irrigation to very low yields after drought. But as you increase planting depth, the increase of yield is very notorious, to the point where this was a drought of at least 2.5, almost 3 months of drought. And the yield was similar to those that were irrigated. What is the problem with this? Well, that most modern varieties, and even many land races, which are not managed this way traditionally, don't have good resistance to deep planting. And you can see here this comparison. This is a maize from Oaxaca, which is handled all the time with deep planting, which has a very high emergence after planting at 34 centimeters, whereas these hybrids, which is a typical result with modern maize hybrids, they have a very poor emergence after deep planting. We have tested so far around 500 different hybrids, and except one, all of them are really poor. So there is a lot of room for improvement, deep planting resistant in modern maize varieties, to take advantage of this fabulous sustainable agricultural practice that by itself increases yield without much genetics, other than, of course, the genetics involved in controlling mesocotyl growth during scotomorphogenesis. Now we wanted to understand also the expression of genes in long mesocotyls versus short mesocotyls, to really understand what sort of functions are happening in these large mesocotyl varieties. We also perform a genetic analysis to get QTLs related to mesocotyl length using 2 biparental recombinant populations, or we call DTP and LP. And we also perform genome-wide association studies to get genes associated to mesocotyl length variations in a collection of inbred lines developed by CIMMYT, the DTMA collection, and also in a collection of maize land races that we have gathered ourselves, and from which we have all the agronomic data behind those accessions. Now, how is the expression in a maize variety, a traditional maize variety called Palomero toluqueño from Mexico, which has a very long mesocotyl in the dark, versus B73, which is an inbred line from Iowa, which contains or develops very short mesocotyls in the dark? Well, basically, the maize transcriptome basically uses the whole maize genome. It expresses the whole maize genome. But the differential expression between these two varieties indicates that almost 5,000 genes are up in Palomero toluqueño relative to B73 in the mesocotyl, whereas 3,300 and so genes are downregulated in Palomero versus relative to B73. The ontology terms of the genes that or the most common ontology terms for this big large amount of, sorry, there is a lot of announcements here that may bother me reading my screen. We have ontology terms related to carbohydrate metabolic processes in the upregulated genes in Palomero toluqueño. The second most common category relates to RNA modification, and there several categories relate to cell wall related processes like hemicellulose metabolic processes, hyaloglucan metabolic processes, cell wall biogenesis, etc., etc. Whereas downregulated genes in Palomero-toluqueño are mostly related to lipid metabolic processes, sulfur compound metabolic processes, and a lot of activities that relate basically to inhibitors of protease activity, which are strongly downregulated. Also, it's very interesting to note that the response to biotic stress is really diminished in Palomero-toluqueño, which makes sense because in general, all living organisms, when they are actively growing, they tend to have a very low basal response to biotic and abiotic stress. So it makes sense, this data. When we look at specific pathways and see how many genes are up or down regulated, this is the cell cycle pathway. And interestingly, in Palomero-toluqueño, there is a lot of genes basically relating to the S phase, to the G2 phase, and the M phases of the cell cycle, which are upregulated. For example, we see origin of replication complex genes, the mini chromosome complex genes. We see two genes related to DNA damage checkpoint control, and also genes involved in the promotion of anaphase. Actually, some which are part of the anaphase promoter complex, like CDC20, CDH1, and also the separase, ESP1, and RAD21. We see a lot of changes in the ribosomal proteins, both large and small subunits. Most of the changes are upregulated, although there are also a lot of downregulated proteins, but the majority of them are upregulated. The spliceosome, a lot of elements of the spliceosome are upregulated, as you can see here. And circadian rhythm pathway, it's very interesting. Most of the genes, or many genes of this pathway, are upregulated. For instance, the phytochrome B, the CRY1, which is the blue light receptor, Gigantia, time of CAV expression of TOC1 gene, PRR7 are upregulated. And interestingly, Hy5, which is one of the master regulators of promoting photomorphogenesis, is extremely downregulated, which makes sense because at this point, the plant is still growing in the dark, and the last thing it wants to do is to trigger photomorphogenesis. Now, the genetics. To do that, we perform QTL analysis using these two different recombinant populations, which were made out of using two different parents, the TMA227, the TMA245, and they were crossed against the same inbred line and genotype by GBS and phenotype. So the idea was to get these recombination maps out of these populations. And to do that, our collaborator at CIMMYT, Shukai Shang, and my student Leopoldo Vasquez had to develop a method that allows for high density recombination maps, which normally when people do QTLs, they have just a few markers that cover the whole genome, and the basically precision of their mapping, it's not the best. So I think we have really made a great contribution creating this high recombination map, which more or less increases the number of markers by 100-fold relative to a standard QTL mapping. After making the association of these markers, mapping them to the maize chromosomes, we found 3 QTLs that mapped previously found QTLs that influenced the mesocotal growth that were described by 2 different labs in China. but we found 10 new QTLs on chromosomes 1, 2, 4, and 10. Now, the association studies. First, we started with a collection of inbred lines from CIMMYT. It's more appropriate to sometimes use inbred lines to do
2 minutes left.
Association mapping. Sorry, 3 minutes?
2 minutes. 2 minutes
Okay,
left.
so, okay, well, we did the GWS was a study in this inbred line collection. We found and confirmed that actually this is a multigenic trait. We found a lot of SNPs with good probability of association to mesocotyl elongation. Most of the gene products encoded by these genes are cytoplasmic. A lot of them are membrane associated, but there is a large number of them which are nuclear. And the most common molecular function of these associated genes is to be transcriptional regulators. Hydrolase activity is also a common activity. These are examples, specific examples of some of the regulated genes or associated genes to mesocotyl elongation. Phytosulfocan, which is a growth regulator in plants. These are heptapeptides, which are sulfated after processing the hollow protein. And this sulfation requires, among other things, adenylyl sulfide kinase, which generates PAPs, which is a precursor of sulfation, which itself is also an associated gene. So it's very interesting. We found this transmembrane superfamily member, which is involved in rhamnogalacturon deposition, 2 different enzymes, which are remodeling, cell wall remodeling enzymes. This transcription factor that regulates HI5, LACY1 family protein member, a lacy family protein member, not LACY1. They control gravitropism, the asymmetric distribution of auxin and light regulation, an ammonium transporter that is negatively regulated by HI5, and some components of transduction pathways like phosphatidyl phosphate kinase, which control embryonic cell identity, microRNAs like this mRNA A396, which controls plant embryo development, which is interesting because mesocotyl is formed during embryo development. We have done the same with our land races, GWS, and we found additional SNPs that map differently, although 2 of them matched 2 of the SNPs that we found in the DTMAs. So to end my talk, I would like to end saying that modern hybrids are about heat and water stress when deep planted in the soil, which if it has good humidity. Stem and root architecture are more robust under water stress when plants are, the seed is deep planted. Grain yield in modern hybrids improves under drought when deep planted. Modern hybrids require large improvements for good seedling emergence after deep sowing. And transcriptomic QTL and WAS analysis reveal candidate genes involved in mesocotyl length variation during scotomorphogenesis. Candidate genes should be useful for understanding the mechanisms of mesocotyl growth variation, meristem maintenance, scotomorphogenesis, evolution of mesocotyl development, and maize domestication. And to end, I would like to ask all the members of my lab, current and former, which contributed to this work. This is also the work of many collaborators outside of my lab at the Institute of Biology or somewhere else at UNAM, Gladys Casab, longtime collaborator at also contributed to this work. Thank you very much.
Thank you, Jorge, for your interesting talk. I think that there is time for one question, and I saw one in the chat from Luis Cardenas. Luis, could you do yourself the question, please? Are you there, Luis? Okay, I'm going to read it. Do we know if the mesocotyl of the Hopi and Toluqueño plants are stronger in order to emerge from deep planting? if you found some genes that could be related with cell wall remodeling or strengthening cell wall, I mean, if there are genes related with cell wall in your analysis, if you found them. Jorge? Hello? I think that we lost Jorge. I think that we lost Jorge. So we are going now to a break, and we will have to be in the next session at 11:45. Please be on sharp. Thank you. Our next speaker is Professor Caspar Chatter from Kew Gardens and Instituto of Biotechnology, UNAM. He's going to talk about epidermal patterning factor signaling and ligum nodulation. Go ahead, Caspar.
Thanks a lot, Felipe. And thank you to you and all the organizers for such an amazing event. I've been really enjoying the talks yesterday and today. And I wish I could make the later ones, but I have to go to bed before it all finishes. But without further ado, I'd like to talk about the epidermal patterning factor story that I'm going to mention today. And really, this is a story of two plant organs or tissues or what you like, stomata and nodules. And as most of you are aware, stomata are key factors in global hydrological systems, the water cycle, and nodules are key systems for the nitrogen cycles of the planet. So my team in the Royal Botanic Gardens Kew in the UK, we're focused on crops and global change and what we can do in terms of accessing plant traits to improve crop responses and resilience to the changes in climate. So, why is that not working? So this is a big team effort, and a lot of the data, most of the data that I'm going to present is ongoing work and largely done by teams in Mexico as well as the University of Sheffield in the UK. So these are projects that we've been doing for several years. I started them off in the lab of Professor Alejandra Covarrubias and continued them in collaboration with her, with Judy Gray, and with Osvaldo Valdez Lopez, who will be, I believe, talking tomorrow. So we've had lots of funding in terms of European Commission funding, UK funding, and Mexican funding. I'm very grateful that these sources can help us continue our work. So, as I said before, we've got a big problem that we are trying to address, and that is the climate emergency and what it means for our food security, or should I say food insecurity. And we need to develop new ways to improve sustainable intensification of climate resilient farming, producing more in less land for better use of resources and reduced impact on the natural environment. And legumes play a major role in this because they are a major global food source. They're a cheap, affordable source of high quality protein, carbohydrates, and other nutrition, but they're also very sensitive to the impacts of climate change. And CIAT in Colombia has modeled this very well in terms of the impact of heat and drought stress in Mexico if we do nothing about the improving the Mexican bean germplasm and the consequent reductions in future bean yields. So we really need to do something about this. And there's a lot of hope because legumes are special. They have this ability to fix nitrogen with rhizobia in the soil. And therefore, in many instances, leaf nitrogen is therefore not limiting photosynthesis like it is often in other plant systems. So carbon assimilation is not proportional to leaf nitrogen in so much in legumes as it is in crops, which means that we could reduce stomatal conductance, as in reduce the aperture of stomata and therefore increase the water use efficiency of legumes by a lot and still maintain the same level of assimilation of carbon. And that's really interesting to me because it means that we have the opportunity in legumes, perhaps more than other crops, to really make big water use efficiency gains. And that's a large part of my work. So a little bit more on stomata. Well, many of you probably know, but stomata control water uptake and loss by transpiration. And this also has effects on many other processes like nutrient accumulation, CO2 uptake, evaporative cooling, and pathogen responses. And this thermal image here you see is of a moss spore capsule. And you see, even in the moss spore capsule, you have stomata at the base of the capsule, the cooling the moss. And the same goes in flowering plants, such as beans, where you have evaporative cooling from the thousands, millions of stomata on the leaves, and you have this draw up from the soil, from the rhizosphere of the water that then escapes through the stomata. Now, my work's mainly involving stomatal processes, both in short-term and long-term responses. Short-term is aperture, so opening and closing in response to the environment, and long-term is developmental responses, so the number of stomata that you make or the size of the stomata you make. And that's what I'm going to be talking about today. So we know from Arabidopsis that stomatal density is governed by quite key core processes across all flowering plants. And these stomatal density developmental responses are governed by epidermal patterning factors, among other components. And epidermal patterning factors drive the increase or decrease in stomatal density that we see on leaves. And we're able to manipulate this in quite close ways in Arabidopsis, so that we can, using epidermal patterning factors, we can have very low densities or very high densities of stomata. Now, what are they? Well, they're cysteine-rich peptides that are cleaved and secreted by the cell and affect the cells around them and determine cell fate and cell identity. And they do this through as receptor agonists and antagonists, inhibiting stomatal development or inducing stomatal development. So the lab of Julie Gray in Sheffield has worked for many years on this question of EPFs and their role in stomatal development, and how we can harness this to improve water use efficiency, among other traits in plants. So this is just a handful of her papers from various members of her lab, in not only in Arabidopsis, but in wheat, in barley, as well as in rice. And in rice, you see here another thermal image on the right here. And you see that if you have low densities of stomata in rice, you actually have a maintenance of evaporative cooling under drought, because the plants are able to keep water in the soil profile for longer. And that's something that I'm going to refer to again in a bit. But the difference between Arabidopsis and rice compared to legumes is that in legumes you have a much more rich source of EPFs. There's a lot of duplications, and that makes our job a little bit harder to identify which EPF candidates might have a role to play in stomatal development. But nevertheless, we have overexpressed many of these in Arabidopsis, and a master's student, Brianda de la Sancha, in IBT, in Alejandra Covarrubias' lab, she looked at this in Arabidopsis. And we've identified one that we overexpressed also in soybean, an EPF from common bean in soybean, where the effect is you have a reduction in stomatal density. And this reduction in stomatal density also goes on to prevent excess water loss under water deficit conditions. And this is work that's been followed on by Miles Bate-Wellman in Sheffield. So on the left, you see stomatal conductance of these EPF overexpressor soybeans under normal conditions, and then under water deficit, you see that the reduction in stomatal conductance is much higher in the wild type plants compared to the EPF plants, showing that they're able to keep the water within their pots for longer. And just as in Arabidopsis, you also have differences in stomatal size when you overexpress EPFs, and this may or may not have effects on evaporative cooling processes that we're also trying to figure out. So we want to take this to the field and try and understand what's going on, or what will go on, as the CO2 concentrations continue to increase in the atmosphere. And we hope to do this in collaboration with the University of Illinois and Professor Andrew Leakey on the soy face, or soybean free air CO2 enrichment program there. So when you take things out in the field, it gets a lot more complicated, because you're then dealing with heterogeneity of the soil compared to the quite controlled conditions of the lab. And we really want to know what's going to happen in a more kind of agronomically relevant environment, not just above ground, like I mentioned before, but also below ground. So what are below ground traits involved? Now, again, we've mentioned this before, but the legume symbiosis is key here. And what we don't want to do is reduce the CO2 fixation of the plant by reducing stomatal density and then somehow mess with this costly symbiosis by reducing the amount of carbon that goes to the legume's friends in the nodules and therefore prevent nitrogen fixation. That would be the worst result, but it's still possible. Now, do we know if EPFs function in roots or in nodules? And we don't. What the literature is quite sparse for any below ground processes with EPFs, whether that be in Arabidopsis or any other plant. And what we do know is that rhizobial nodulation involves a huge number of other peptides, of which we have RALFs and PEPs and ORFs and ENODs, and in autoregulation of nodulation as well, we have CEPs and CLE peptides. So it's a rich environment, and peptide signaling plays a huge role both above ground and below ground. But so far, no role for EPFs has been shown. Now, what about Arabidopsis? Well, I mentioned there's not a lot in the literature, but papers from Judy Gray's lab have looked at EPFs involved in stomatal signaling to see if there are below ground effects, and there are below ground effects. So EPF1 and EPF2, which are involved in stomatal development, you don't see any expression of those EPFs in the roots or the root hairs, but you do see that if you overexpress an EPF, you have reduction in root mass, and if you delete a couple of these EPFs, you have an increase in root mass, and that suggests that there's a lot of crosstalk, and that's probably not a direct molecular one, but something to do with the hydraulics of the system. You know, you're losing water faster or slower, and therefore you build your plumbing to accommodate that. So we also see if you overexpress an EPF in rice, we have knock-on effects on the parenchyma tissue. And that's, again, very interesting. And we don't know if this is, again, an indirect role like we see in Arabidopsis. Another reason why it could be less direct, and similarly we see this in legumes, is the expression levels of these EPFs are very, very low in nodules, in legume root hairs and roots. And this is just data from phytosome. You see this in soybean and common bean. They're almost traced to none in a lot of these transcripts, again, suggesting they are not doing a lot. So we got the chance to explore this in more detail on a Global Challenges Research Fund project with Julie and with Osvaldo. A year or so ago, we started the project, and as you all know, COVID has played merry hell with all of it. But I've got somewhere, and work from his postdoc, Dr. Litsi Agapado, has been really, really exciting for us to see how this has developed over the past year, even under those conditions. And what Litsi has shown is that we do detect certain EPF peptides and transcripts in the roots, and these EPF peptides respond to rhizobial infection and nodulation. So this is really, really exciting stuff, and it's just very early days for us to probe this. Now, what she also shows is that we can downregulate or overexpress certain EPFs, and we get effects on nodule number, on nodule size, as well as nitrogenase activity. And we are now probing this even further to determine what's behind this. Is this just a case of we're messing with the system, or are we actually getting to a core mechanism of peptide signaling that we can hijack or harness? And what we also see is that we get a larger infected cells and corresponding with these larger nodules. So there's something there, and we are very close to working out what's behind that. Another similar project, again, one that's been like all of them affected by COVID, but we've got some really, really interesting data now. This is a Newton Prize project with the labs of Alejandro Covarrubias and Gina Hernandez in Centro de Ciencias Genómicas, la UNAM. We are looking at common bean water use efficiency, as well as nitrogen fixation phenotypes in a whole population of EMS mutants in common bean. And postdocs Pablo Paleas, Delfina Epen, Alexis Acosta, and Jose Polania over the past year or so have been looking at this population to determine whether they can identify cool mutants or hot mutants amongst them. And they have found a range of both. And when I say cool mutants, I mean mutants that are unable to regulate their water loss compared to the wild type. And when I say hot mutants, I mean those that are unable to lose as much water. So you see in this example here at the top in this thermal image, there is a hot mutant that's quite a bit hotter than its neighbors around it. Perhaps because the stomata are closed, or perhaps there's some other phenotype that we have to identify. And then below you see a much cooler individual compared to its neighbors. And those are the plants that we are now working on with whole genome sequencing to identify the polymorphisms involved, genes involved, and then be able to use that information to improve the water use efficiency and heat and drought responses of these beans. So in summary, I probably raced through this a bit more quickly than I was expecting. We've been enhancing both bean drought resilience and nitrogen fixation processes, or at least starting to understand the processes to be able to do so. We know that EPFs can modulate legume stomatal density through the work on common bean and soybean that we've started, and that by reducing stomatal index and stomatal density in these plants, we can improve their water use efficiency. And now we know that other EPFs modulate legume root nodules, and we don't know how, but we're beginning to find out. And it strongly suggests that we can then use these data and this information to fine-tune targets both above ground and below ground to create drought-resilient nitrogen fixers for a new way of agriculture. and improving food security and water security with these tools. So on that note, I'd like to thank you all again for listening, and happy to try and answer any questions. Thank you.
Thank you, Caspar. We have time for questions. Is there some question?
Hi.
Hi, Luis.
Felipe, can I make a question? Hi, Caspar. Nice talk. I just have a very brief question. So can you spread the EPF factor over the leaves of the Phaseolus vulgaris beans leaves and have an effect on the stomata development? Or do you need to express the peptide? Just in case if you want to see the, yeah.
Yeah, so some early work that I did, I used the synthetic peptide on very young developing leaves and developing pods of beans, and I was able to get some changes in stomatal density. So it's possible with the synthetic peptide or yeah, just the peptide itself. But what I found was you needed quite a lot. So it might just be efficient to get through the leaf.
Okay, so by doing so, do you think that you could affect directly the root architecture or even a nodule phenotype? I'm just thinking about that in Phaseolus vulgaris, we don't have pretty much access for the transgenic stable lines.
Yeah, so it's a really interesting question and one that Osvaldo and I have been working on. And so far we have not got proper information to say that there's a long distance signal. But we know the experiments we want to do that could help us answer that.
Okay. Thank you, Caspar.
Thank you.
We have time for a couple of questions more. Does anyone for questions?
Hi, Pepe.
So if we don't have more questions, we...
Jose Luis Reyes, go ahead.
There is another question? Okay.
Yes, I was trying to unmute myself. Hi, Caspar.
Hi.
Thanks for your talk. Very interesting as always. I remember there are a few versions or a few different genes encoding EPF-like factors. Is it in the future to have experiments with others? Do you think there might be specific effects of using different isoforms?
Yeah, I'm sure that. I mean, the thing that I mentioned is that there are many paralogs in legumes compared to the simple lab rat that's Arabidopsis, and what Litzi's work is showing is that there could be 2 to 4 different ones working in nodules that are what's the word? Probably much more endogenously important than the experiments that we've started. So we're kind of honing in on the main players. Yeah.
Okay. So would there be a point in having combinations as well, or would that makes it too complicated from your reaction?
Yeah, I mean, it's a good question. I think, like I said in the last slide, there's a lot of scope for fine tuning responses, and the fact that there are maybe 2 or 3 up to 4 players could mean that some could be working at this particular stage in infection, nodulation, etc. And turning on or off genes at certain times might be the way to do it.
All right, thank you.
Cheers.
Okay, we have time for one question. Yeah, Aida, go ahead.
Aida?
Yeah, Aida.
I think
Go
you're
ahead,
on
Aida.
mute.
Sorry, did you hear me? No.
No, sorry.
Okay, again, sorry. How much is affected the photosynthesis in your mutants or in your overexpressed plants with this factor?
So for the stomatal development overexpressors in soybean, we don't see any significant difference in photosynthetic parameters. It's negligible if there is.
Okay, good. Thank you.
Thank you.
Thank you very much, Caspar, for your very, very
Thanks a lot.
interesting talk. Thank you.
Thanks for giving me the opportunity to talk. I look forward to the others. See you later.
Bye. Our next speaker is Professor Patricia Leon from Institute of Biotechnology, UNAM, and she's going to talk about the plastic retrograde signals and their impact in plant development. Go ahead, Patty.
Thank you. Let me just try to put my, hope you are seeing my screen right now. It's okay?
Yep.
Okay, well, so I will start, and I will start by thanking the organizers for this splendid meeting under the circumstances. It's great. And also for giving me the opportunity to be here and present and share with you some of the current work of my group. And in this occasion, I will tell you about plastic retrograde signaling and the impact in plant development. And well, just as a very brief reminder to you, I would like to mention that the acquisition of the chloroplast by the plant cell not only provide to these organisms the autotrophic capacity, but also during the evolution, this organelle become the center where a lot of different specialized biochemical processes take place. And this aspect become very in coordination with the appearance to different plastic types that were not present in the original endosymbiont. Thus, it's not surprising at all the fact that they must be, in order that all these pathways work efficiently and are regulated, there must be a close connection and communication between the nucleus and the organelles. Of course, the nucleus regulate most of the developmental and metabolic process of the organelle because, as you know, the majority of the proteins required for these processes are encoded in the nucleus. And that is what it has been known as anterograde regulation. However, it is very well accepted that the organelle sends signals to the nucleus and modulate the gene expression of many genes involved in the plastic functionality, but also, as I'm going to show to you, genes that affect the growth and the development of the plant. And this is what it has been known as the retrograde communication or regulation. Retrograde communication is a quite complex process because it involves many signals and signaling pathways. And they have been divided in those that operate during the in the differentiated organelle as the operational signalers, and those that happen through the biogenesis of the organelle. So in my group, we have been interested trying to understand the signaling pathway and isolate and identify new signals of the of this retrograde communication. And today in this talk, I would like to share with you some of the work that we are doing in two main lines. In the first part, I would like to present to you what are we doing trying to understand the evolution of the biogenic signaling and signaling pathway. And we are doing that by analyzing the present and function of known factors that participate in biogenic control in vascular plants, like model plants, like Arabidopsis or maize, in early terrestrial plant lineages. And early, we heard Dr. Solano's talk regarding the great characteristics that plants such Marchantia polymorpha has with this low redundancy. And especially and particularly important for us is that this lower plant, this basal plant have already different plastid types. So we think that it's a good model system to study and compare the evolution of these pathways. And in the second part of my talk, I would like to mention to you the work that we have done regarding a new signal derived from cis carotenoids that we call ASIAS1, and its impact in the plant development. And the first part of this talk, in order to analyze the diverse elements that participate in biogenic retrograde signaling in evolution, we made an analysis of the putative or the presence of putative orthologs in different plant species. And to our surprise, we observed that many of the factors that we analyze, and I'm showing to you here just a few of them, are present in most of the lineages that we analyze, and in particular in the case of Marchantia polymorpha, that with few exceptions, such PRIN2, they are present. And in all of the cases, they are present as a single copy gene. So today I would like to tell you the story of the analysis of one of these factors, and that is the GLK transcriptional factors. GLK transcriptional factors are two genes in the case of Arabidopsis and several other vascular plants that have been shown to be very important for the chloroplast differentiation. For example, mutations in these are partially redundant, and mutations in these two genes result in defects in the plants, such as green pale phenotypes, and defects in the differentiation of the chloroplast. More importantly for us is that overexpressions of either of these two genes result in ectopic differentiation of chloroplast in tissues that are not usually present chloroplast, such as the root. So our analysis demonstrates that Marchantia polymorpha has one single putative ortholog for the GLK genes. And you can see here, this gene has the classic domains, the DNA binding and protein-protein interaction domains. However, the protein of Marchantia polymorpha is it's bigger than the ones from Arabidopsis. And in fact, it contains an additional domain, a pseudo-response regulatory domain that you can see here, which interestingly is also shared with other members of the GARB family. However, these members, at least in the case of Arabidopsis, are not function or do not function in a biogenic control, but more in biotic stress and cytokinin responses. So we were interested to understand or to try to see whether this putative ortholog has any role in biogenic control. And we did this, or we have been doing this, by isolating mutants or overexpressors of this transcriptional factor. And to our pleasure, we could observe that the phenotypes of the plant that we obtain in Marchantia polymorpha are very similar to the one observed in Arabidopsis. That means mutants of the GLK transcription factor has a pale green phenotype, whereas overexpressors have a more green phenotype with a higher chlorophyll accumulation. More important for us was the analysis of the different tissues in Mercantia, in which we could observe that tissues where no chloroplasts are present normally in the wild-type plants, such as the storage region, we do observe marked green color, as you can see here, that correlates also with autofluorescence of the chlorophyll, suggesting that, in fact, in these tissues, now we are having probably chloroplast or chloroplast type of organelles. Also, we observe important differences in the chloroplast morphology. For example, a strong accumulation of amyloplasts. Thus, by doing this analysis, we have been able, or we have a lot of evidence that the Mercantia polymorpha GLK acts actually already in a retrograde signaling, affecting and modulated the differentiation process of the chloroplasts. And we will continue to analyze this in more detail. So now I'm going to go to the second part of my talk, which involves this new signal that we call AZS1, that is derived from cis carotenoids. And this work started with the identification and characterization of a mutant that we call ZLB5. ZLB5 affects an enzyme involved in the carotenoid biosynthetic pathway, which is the zeta-carotenoid desaturase. However, these mutants, in contrast to other mutants that also affect the same carotenogenic pathway, have a very dramatic phenotype in the leaves, as you can see here, because it contains radial leaves that are very different to the ones observed in the wild type or other mutants in the same pathway. We were able to demonstrate that, in fact, the accumulation of these two linear carotenoids, phytofluin and zeta-carotenoids, were responsible for this leaf phenotype. And then we hypothesized that probably a signal emerging from these carotenoids were causing this leaf phenotype. Carotenoid biosynthesis is a very interesting pathway because it is responsible for the production of many different signals, including very important hormones such as ABA and strigolactones that are involved in many stress responses, and also signals that are retrograde signals such as beta-cyclocitral and mycorrhizin. However, many of these signals derived from the carotenoid pathway are actually derivatives of carotenoids, known as apocarotenoids, which are produced either by light or by enzymatic reactions. In our case, we were able to demonstrate that, in fact, this is also the case for the AZS1, because the enzyme CCD4, the carotenoid cleavage deoxygenase 4, is required for this leaf phenotype, and double mutants without CCD4 and CLB5 have partial recuperation of the leaf morphology. So unfortunately, up to now, we have still not been able to known or characterize the exact structure of AZS1. And these have turned out to be very tough, in particular because the amounts of these carotenoids appear to be very, very limited. So in order to try to have an idea of its function and the signaling pathway involved, we decided to take an analysis, to perform an analysis, knowing a transcriptional profile in the CLB5 mutant. And we analyzed the expression, the gene expression in the CLB mutant, and we compare it not to the wild type because there are many differences, but with a mutant in the same carotenoid biosynthetic pathway in the previous enzymatic step, the PDS, which also do not have carotenoids, but have a very normal leaf appearance. And we observed that, to our surprise, more than 2,000 genes were significantly affected in its expression. Moreover, we found out that many of these genes become more similar to the PDS3 in the double CLB5-CCD4 mutant that, as I mentioned to you, recuperate the leaf phenotype. When we analyze the biological processes that were affected of the genes affected in the mutant, we find out something very interesting, and that is that more than 50% of the nuclear encoded genes for the protein chloroplast ribosomal proteins were severely repressed or downregulated in our CLB5 mutants. And this downregulation becomes suppressed in the double CCD4 CLB5 mutants. We have been able to demonstrate more recently that this decrease in the transcript accumulation results from the repression of the genes, of these genes. As you can see here, the nascent or primary transcript in our CLB5 mutants of many of these chlororibosomal proteins are downregulated as similar to the mature transcript. So from this, we could conclude that ACS1 were downregulated transcriptionally the expression of many of the chlororibosomal proteins, nuclear encoded chlororibosomal proteins. Immediately when we found this, it became to our attention previous studies that have already published that the addition of plastic translation inhibitors such as spectinomycin or lincomycin results in defects of the leaves. And we did close comparisons of the morphology of the leaves in our mutant and in streptomycin-treated plants. And we observed that the phenotypes of these leaves were undistinguishable, so suggesting a common mechanism. As you can see here, both are radial and both have similar deficiencies. However, we find out also that the treatment with the spectinomycin or lincomycin were not able to repress the expression of the nuclear chlororibosomal proteins, as you can see here, which is very similar, or even in some cases, higher than in the wild type, in comparison to what we have previously observed in the CLB5 and another allele, SPC1-2, another allele of the CLB5 mutant. Those suggesting that, in fact, ACS1 was affecting chlororibosomal protein expression, and these defects was affecting the plastic translation in a similar manner to when inhibitors were used by spectinomycin or lincomycin. However, both causing this plastic translation reduction result in leaf in development. So then one thing that we were interested on is to try to understand in more detail the biological consequences of the ACS1 accumulation in the plant development. We realized that maybe this analysis could help us to understand and identify signaling factors involved in this pathway. And We initially, some time ago, already have demonstrated that the leaves in the CLB5 mutant have important effects in the adaxial and abaxial pattern, as you can see here with these reported genes, where the expression are in some cases completely gone, for example, for filamentous flowers. However, more recently, we were interested trying to understand whether these defects in the leaves were actually started early in development. And for that, we also analyzed the shoot apical meristem and the expression of some marker genes. And to our surprise, we observed that in fact, in our mutant that you can see here, we have an important reduction of gene markers. And in this case, I'm showing to you some of them, like Wuchel and Clavata, that are very important for meristem maintenance. This demonstrates then that our mutant, CLB5 mutant, have important effects, not only in the leaves, in the leaf morphology, but also in the meristem homeostasis and gene expression. However, the analysis of the some meristem in our mutants have demonstrated to be quite challenging because of the mutant size and number. So we decide to analyze in a more detail whether other parts of the plants or other regions of the plants were also affected in our CLB5 mutant. And we could demonstrate that in fact, this is the case. We found defects also in the root apical meristem. As you can see here in these slides, we observe clear defects in the morphology of the cell niche and the columella in both CLB5 and SPC1, which is the allele, an allele in the Columbia background. And this is in comparison to the wild type and also to the PDS3 mutant, which is the mutant just above the biosynthetic a step above CLB5. So having demonstrated this aspect, we thought that it could be much easier to analyze the expression profile of different markers of the root apical meristem. And as you can see here in this slide, I showed to you that several markers, including wolf-pied, scarecrow, have similar expression pattern. We do not observe like mis-expression in different cells. However, we certainly observe difference in the level or in the cells that are expressing these markers. We also have shown that the accumulation of starch in the columella is quite deficient in our CLB5 mutants, as well as the accumulation of the auxin levels around the root apical meristem, shown by the DR5 marker and PIN1, as you can see here. This expression profile is consistent to meristem exhaustion in this mutant. Well, now I would like to end this talk just by showing to you where is it a place where natural AZS1 can be produced? That is a question that we have been interested for a long time ago, and it was very tough to be analyzed. But now, knowing these defects in the root meristem, it has been much difficult. And to do that, we made constructs that express from the promoter and the protein of different of the different proteins that are involved in AZS1 biosynthesis. That means the DXS protein, which is involved in the synthesis of the precursors of all carotenoids, the PDS, which is the enzymatic, in previous enzymatic step, and the ZDS. And we could observe how DXS is expressed in all the plastids present in the root apical meristem, as you can see here. However, to our surprise, and this is, I think, quite interesting, is that PDS3 is expressed mainly in the upper part of the quiescent center, as you can, I hope you can see here, and a little bit in the columnella at the end, at the tip of the root. In contrast, ZDS is basically absent, as far as we can say, in around the root meristem niche, and it's present in the tip of the root. This could create gradients of productions and sites of synthesis, of potential synthesis of AZS1. And we think that this result will help us to first identify probably the nature of AZS1, because it's possible that we can detect it in a very specific tissues. And second, we could understand some, or at least explain why we have some defects, for example, in the columnella. And with this, I just want to summarize, and I hope during these minutes, I was able to convince to you that the plastids send a variety of signals. And now we are discovering this new signal derived from linear carotenoids, which by the participation of carotenoid deoxygenases enzyme, produce this new novel carotenoid, one or more, and in which light is important. I'm sorry I didn't have the time to tell you about this. But what is interesting is that we are seeing this dialogue that is starting between the chloroplast to the nucleus, in which AZS1 is produced, affecting the plastid translation proteins, which in turns affect the translation of the chloroplast, which in turns sends another signal yet to be identified that affects again the expression of nuclear genes that are important for the meristem homeostasis in the shoot apical meristem, in the root apical meristem. And also, I didn't have the time, but we have pretty good evidence that that happens in the floral meristem. So today I'm showing to you that these slaves have an important voice to say, and that modulates the development of the plant. And with that, I want to conclude and finish acknowledging, if I can, with the most important slide of my talk, which is acknowledging and thanking the people that have made possible this for me to be here, which is previous participants, Lucero Gutierrez and Odetta Ventaño, current participants, current members of the lab, Elizabeth Cordova, Julio, Lina, Kenny, Ariel, and Leslie, that without them, I couldn't be here. My collaborators, Mario Arteaga from the University of Veracruzana, regarding the Mercantia polymorphia work, Barry Poxon, Ryan McQueen, Sandra Mathoni, and Blake Mayers in the AZS1 work. And of course, the funding agencies, without such, I couldn't make any of what I have told you today. And with that, I'm happy to take and try to answer any question. Thank you.
Thank you, Patty. Is there someone for a question? We have time for a couple of them. Okay, Eleazar. Go ahead, Eleazar.
Can you hear me?
Yeah. Yes.
Okay. Outstanding, Patty. Outstanding.
Thank you, Eleazar.
it's fair to say that the communication between the plastids and the nucleus is not restricted to leaves?
Yes, I don't, if I understand well, you are asking me if they are not restricted to leaves?
Exactly.
Yes, no. What we are thinking is that, in fact, this communication is probably very important in the meristematic regions. And I didn't have the time, but if you can imagine, in the meristematic regions, there are plastids, which are not chloroplasts, of course, but they are probably plastids that are very prompt to signal its developmental stage. It's very well known that it depends on the tissue, and even in the cell layer, it differentiates over different plastid types. So what we are thinking that is far more complex, and that yes, in the different meristem, root meristem, shoot meristem, and also in the flower meristem, this communication might be very important to mention, to regulate some of the fate of the tissues that are going to be originated. Yeah.
I have time for another quick question from Nayeli March.
Hello, Patty. Very nice talk. Thank you. And I wanted to ask you if you think if ACS1 can move between cells, like if you could do some grafting between plants, and maybe that could help to identify the nature of the molecule.
Yes, in fact, that's something that we have been very interested on, because many of the apocarotenoids are very small molecules that have the capacity to travel between cells and in distance. And yes, we are very interested in doing these things. Unfortunately, the mutants without carotenoids have a clear disadvantages. So for a long time, we have been trying to set up a system in which we can modulate the production of ACS1 artificially without having the problem of not having carotenoids, because this mutant, you know, it's lethal. So the handling and the manipulation, it's quite hard. But yes, and that's why it's very interesting that in the root we see this gradient of possible biosynthesis of ACS1. But yes, that's something that we are very, very keen and interested to try to do.
Thank you, Patty, again for your very, very interesting talk.
Thank you. Let me.
So we'll continue with our meeting. And before I present the next speaker, I will recall our audience to please use the option of raise hand in Zoom to ask for questions. That would be easier for us to give you word. So with that, I want to introduce, it's my pleasure to introduce Dr. Marina Gavilanes. She comes from the Faculty of Chemistry from the National University of Mexico, and she will talk about functional diversity on the plasma membrane sphingolipidome. So thank you, Marina, for accepting our invitation, and go on.
Thank you. Thank you so much. And thank you, all you organizers, Alejandra, Felipe, Ulises, Stefan, Mario, and Dan. Thank you so much for this huge effort. And thank you also to the technological team that has made this almost miracle possible. So I will talk about the functional diversity in the plasma membrane sphingolipidome. And here I'm starting with the model of the plasma membrane or any biological membrane that was proposed many years ago, and that has two dimensions: the spatial distribution of lipids and proteins in a bilayer, and then the temporal dimension in which these components are diffusing freely along the membrane in the lateral plane. But this model has been recently refined and tuned in such a way that we now envision these membranes as bilayers with patches composed of specific lipids and proteins that have specific functions, and that these membranes have these patches traveling along the plane with all their charge of proteins and specific lipids for very specific functions. And then it turns out that the plant plasma membrane is a specially patchy membrane. It seems that it is organized in small regions that are confined by the limitation of elements from the fibers of the cytoskeleton and the cell wall. And it seems that this SOPRA organization is specifically planned in order to fulfill the plasma membrane functions. So in this context, knowing the fine composition of membrane is crucial to understand its organization and function, and then taking into account that we can have access to very powerful techniques to know the detailed composition of membranes. It's important to apply all this knowledge to the better understanding of the membranes. And remarkably, sphingolipids constitute a majority of the total lipids of the plasma membrane. And this is something very special that doesn't happen in any other eukaryote plasma membranes. And sphingolipids are a very diverse group that have a polar head and two hydrophobic tails. And according to the polar head, they are classified in ceramides, hydroxyceramides that have small polar heads, and glucosyl ceramides and glycosilyl phosphoceramides that have big hydrophilic heads. And these are the GIPCs components that have huge hydrophilic and voluminous and bulky polar heads. But all of these classes of sphingolipids contain the same two kinds of tails that are hydrophobic, and that these hydrophobic tails in Arabidopsis are formed by fatty acids and long change base. In Arabidopsis, we can find 10 different fatty acids that are different in their length of number of carbons and on the number and position of unsaturations. And Arabidopsis has mainly 4 variants of long change bases that differ in the double bond presence or in the hydroxyl presence. So what I will be talking today is about the detailed composition of this portion of the sphingolipids, the two tails that make the hydrophobic part of sphingolipids. And we had some questions to launch this project about the abundance of a particular sphingolipid class in the plasma membrane could be found, and if the content of individual species of sphingolipid or sphingolipid classes were suggestive of the function of single lipids in the plasma membrane. So our approach was to determine a very fine content of the sphingolipid adsorbent of the plasma membrane and confront it to the sphingolipidomes of the vacuolar membranes and the microsomal membranes and the detergent-resistant membranes, which have been proved to be a good tool to know the lipid and protein components of microdomains in many different membranes. So this is the strategy that we followed, and we used leaves from Arabidopsis thaliana adult plants, and we isolated crude membranes, and from these crude membranes we isolated plasma membranes from with 2 different techniques, and we also purified vacuolar membranes and detergent-resistant membranes. To these 23 replicate membrane preparations, we applied the analysis by HPLC and mass spectrometry that render 84 sphingolipid species in all the different kind of membranes. To these 84 sphingolipid species, we applied an informatic analysis that included multivariate and univariate methods, clustering analysis, and also principal component analysis. This had to be done with informatics because after we end at this point, we have about 2,000 different entries of different values of long chain bases and fatty acids that are composing these sphingolipid species. And here you have general view of the qualitative presence of the 84 sphingolipid species that are composed of 1 fatty acid and 1 long chain base in the GIPC classes, in the glucoceramide class, and the hydroxyceramide class, and in the ceramide class. And as you can see, but with this heat map, the crude membranes contain a little bit of all of the 84 species, but then vacuolar membranes are more enriched in glucosylceramide species and in these other ceramides. The plasma membrane that we analyzed it turned out to be very enriched in GIPCs, and the DRMs in GIPCs and in these other sphingolipid classes. So this is a general view that tells you much about what is the strategy of the plant cell in such a way that the sphingolipid synthesis works, rendering a lot of species, but they are differentially distributed in the different membranes in such a way that they can cooperate to the specialized function of every one of the membranes. Here are the quantitative data about the sphingolipid contents, and you can see that vacuolar membranes and plasma membranes have around the same amount of sphingolipids. But if we unfold these contents, you can see that these contents that were very similar then become different in terms of the amount of every sphingolipid class, both in the vacuolar membrane and in the plasma membrane. And GIPCs, they have about the same. This is in absolute numbers. But in terms of proportions, if we zoom this as a 100%, the percentage of every sphingolipid class is shown here. And you can see how plasma membrane is tremendously enriched in GIPCs that are the ones that have this huge voluminous head. And it contains also 17% of hydroxyceramides and about 12% of glucosylceramides and about 7% of ceramides. And this is important because this makes a profile for the plasma membranes that speaks a lot of, at least suggests a lot of which are the functions that these specialized sphingolipids do in the plasma membrane. And in order to describe the abundance and the diversity of sphingolipid species, the 84 ones that we identified in the 4 membranes, we divided them in species that are in a high abundance or that are in low abundance, depending of every sphingolipid class. And as you can see, the high abundant species are few as compared to the ones that are in low abundance, because they are present in a more in a higher number. Here, and just to give you a sip of the sphingolipidomic study, you can see that GIPCs that were the GIPCs in the plasma membrane that are these 13, these 14, there are 27 sphingolipid species that are in blue. The plasma membrane is always in blue. You can see that they have differential amounts in across the plasma membranes. However, from these 13 GIPCs that are in high abundance, it is not known if they are more abundant in the plasma membrane and are less abundant in other membranes. What Laura Carmona Salazar did was to make a scrutiny of the 84 sphingolipid species and selected with informatic tools which were the species that were mainly abundant in every membrane. I will just give you a small sip of the results that we had, and we are emphasizing the results with the plasma membrane. And the GIPCs that are the ones that are more abundant, turns out that the 27 species, only 8 GIPCs are more selectively abundant in the plasma membrane as compared to the other different membranes. So this is interesting because GIPCs are these sphingolipids with huge heads, very hydrophilic, that are 27 in the plasma membrane. 13 of them are more abundant, and 6 are especially enriched in the plasma membrane, and they will be protruding from the surface of the plasma membrane, and they make this feature suitable for sensing external signals or for recognition of other molecules. And therefore, there are some reports that already have described that GIPCs that attach galacturonans from the cell wall, and other GIPCs that act like a receptor of sodium ions during salinity stress. But at the same time, GIPCs can function as bulk lipids because they are very, very high amounts in the plasma membrane, and they can be contributed to the nanodomain formation or to regulate the membrane fluidity. And sphingolipids, it is known that seem to rigidize the plasma membrane fluidity. So we wanted to determine first the capacity of the membranes that I already showed you to sense the temperature changes, and then later to examine the sphingolipid role in the control of this membrane fluidity. And the strategy that we follow was to isolate the membranes, as in the previous results that I showed you, from the mixture of the crude membranes, the bipolar membranes, and the plasma membranes. And from these ones, remember that they are high in sphingolipids, especially these plasma membrane vesicles. And we expose them to a train of different temperatures that go to the cold to the high temperatures. And then to these membranes, we measure membrane fluidity. And then Dora Luz Cannon Ramirez did these experiments some time ago, and we use fluorescent probes that can sense the mobility of the membrane at different depths of the membrane, like this parinaric acid that senses the fluidity in the intermediate and the middle plane region of the membrane. TMA DPH senses the mobility in the surface, and DPH in the inner or central hydrophobic core of the membrane. And we can follow the mobility of these probes in the measuring the fluorescent polarization of these probes. And what you can see is that high numbers of fluorescent polarization correspond to rigid membranes, and low values of fluorescent polarization correspond to fluid membranes. In these fluid membranes, you can see that the disorder of the acid chains is very high as compared to the rigidity of the chains when there is membrane that is more rigid. And at the same time, you can see that the temperature that we can impose to the membranes that we are heating can produce different states of gradual disorder of the lipid phase, going from the rigid to the very fluid membrane. And this setting of the conditions to measure fluidity is very robust. Dora Luz did a splendid job doing the design in such a way that every curve that you can see here corresponds to about 250 measurements of membrane fluidity. And these are the results that we got. So this is the fluorescent polarization measured with DPH, the TMA DPH, and the parinaric acid. And you will find the curves in blue for the plasma membrane, in green, the bipolar membrane, and in red for the microsomes. And you can see two lines for every type of membrane, and two lines is the recordings when we were heating up the membranes, or the recordings when we were cooling down the membranes. And as you can see that with the hydrophobic probes, they report the membrane fluidity of the fluidity of the plasma membranes as the more fluids, because these are with the lower values, and this is a very different situation with the other membranes. So this means that plasma membranes are per se very fluid membranes. And at the same time, you can see that for every 2 degrees that we increase the temperature, there is a response of fluidity, and this response is fast because every point is taken or recorded every 2 minutes. So this means that the plasma membrane responds with sensitivity and responds with a fast time in order to change the values according to temperature. And you can see here also that the plasma membrane shows curves that change slopes, and these slopes are important because these slopes mean that there are phase transitions from one order state to the other order state of the membrane, and this can be better followed by Arrhenius plots in which these same values are plotted here, and you can see the breaking points that are corresponding to the transition phases that are more clearly shown in the plasma membranes and not in the other membranes. So this is something that suggests that the plasma membrane is very sensitive to the changes of temperature, and furthermore, all these changes are being happening in the cuvette, so there is no input of metabolic energy of the cell to provide this behavior of the plasma membrane. So this is some process that is almost energy free, and that is important because of the reactivity of the plasma membranes of the plants, which are poikilotherms and cannot regulate temperature. So they must sense temperature all the time in a very sensitive and fast reaction. So our conclusion was that the plasma membrane fluidity from Arabidopsis is a good candidate to be a primary sensor of temperature in plants. And but how do specific sphingolipids contribute to membrane fluidity? Since we have the tool of the sphingolipidomics setting and also have the fluidity measurement, our idea was to make a kind of titration of amount of sphingolipids, endogenous sphingolipids in the different plasma membranes of the mutants, and then to see if there was a correlation with the membrane fluidity. And we did the same kind of experiments, but this time we isolated the membranes from the mutants. And then we had the following results. Well, you can see that the sphingolipid composition of the wild type and 3 different mutants in the sphingolipid synthesis. And as you can see, all the mutants observed a major content of sphingolipids as compared to the wild type. And at the same time, the distribution of these total sphingolipids was different depending on the mutant. And there was one mutant that is the SVH1 mutant that is very contrasting with these wild type plasma membrane features. So I will tell you only about this mutant. And here you can see, for example, that this mutant has a very low ratio of trihydroxylated to dehydroxylated long change bases. And at the same time, it observes low ratio of very long chain fatty acids to very long chain fatty acids. And here are the results. These are the long chain bases that lack the hydroxy group in the long chain base. And this mutant has about half of the trihydroxylated long chain bases content. So you can see here that when Dora Luz measured the fluidity of the plasma membranes from the mutant and from the wild-type plants, she observed that in all cases, the mutant had more rigid plasma membranes than the wild-type. So
you have
that
2 minutes,
was,
Marina. 2 minutes left.
Okay, so so this was an indication that this hydroxy group was very important to increase membrane rigidity. And so this says that single ellipses can be negative modulators of the membrane fluidity, and we are working with the rest of the mutants. And these are the people that did all the work. Dora Luz, that had a very nice and longing stay at my lab in her master degree. Gio, that has helping with the analysis, although Dora Luz did all the membrane fluidity measurements. Cello, that did all the membrane preparation, almost all of them. Rosario, that provided the tonoplast vesicles. Becky Cajun, that analyzed all the sphingolipidomes, and Laura Carmona, that was the informatic person that analyzed the sphingolipidomes, but that it was the driving force for this sphingolipidomic project. And Edgar Cajun, because of his generosity in providing the mutants and everything related to sphingolipid analysis. So thank you so much.
Okay, thank you, Marina. Thank you for a nice talk. So let's go to questions. We have a time for just one quick question. No, well, I will ask, oh yeah, Luis Cárdenas, as always. Let's see. So now you can.
Okay.
Okay.
Can you hear me now?
Yes.
Yes.
Okay. Thank you, Marina. That was a very nice talk. I just have a very short question. So could you say that the lipid composition in the plant roots will be very similar to the leaves, for instance? Or do you think that the composition will change?
I don't know. We just have had a look on the mesophyll cells composition. That is the one that I presented, and there is probably some. There will be probably some differences because the regulation of sphingolipid synthesis must be very different in the white tissues and as compared to the photosynthetic tissues.
Yeah, I'm curious because as you said in your presentation, the GIPC, which is the glycosyl inositol phosphatidylceramide, it has been involved in the salinity response. And it's very interesting because this is a lipid that actually is directly binding to the sodium and inducing all the response. So I think that from the point of view that these lipids are like very specific sensors, it's like a completely new role for this kind of lipids. And then I was just wondering about the roots compared to the leaves. And I think that they could be like doing different kind of responses. I don't know what do you think about.
Yeah, I could expect that GIPCs could be also a main component in root plasma membranes. What I would think that would be different are the individual species or different proportions, I would say so. But GIPCs are a must in the plasma membranes. Yes.
Interesting. Thank you, Marina.
Thank you. Thank you, Marina. Thank you, Luisa.
Thank you,
Thank you so much,
Yeah,
Marina,
thanks.
for your talk.
You're welcome.
Yeah, so let's go to the next talk. So it's my pleasure to introduce Dr. Rodrigo Gutierrez. He is from the Universidad Católica de Chile, and he will tell us about plant life at the extreme in the Atacama Desert. Thank you, Rodrigo. Go on.
Hello, can you hear me?
Yeah, everything fine.
All right. Thank you, Alejandra, for the introduction. And I want to thank all the organizers for inviting me to talk about one of the projects that's been going on in the lab for some time now. This is going to be quite a change from the previous talks, but I hope you like some of the data that we have, some of the results. And so, as the title says, it has to do with understanding how plants survive under very extreme conditions, in particular in the Atacama Desert. And the motivation to do this, as you know, human societies are facing an ecological crisis of global proportions, primarily due to the accumulation of greenhouse gases in the atmosphere that is shifting the world's relative balance and increasing global temperatures. And in particular, climate simulations show increased aridity, but especially in the subtropics. And this will affect many countries, such as Chile, for example. But in terms of the global desertification, there is a prediction that will increase from 48% to 65% of the total land surface. So here is Chile, for example. And I just like to emphasize that this is not only a prediction, this is something that is already happening. And in particular, in Chile, I have this example from a lake that's near Santiago, the capital city where we are based, where our university is based. And in the span of 10 years, as you can see from these satellite images, this lake used to be a summer destination for fishing, swimming, boats, and so on. Completely dried out, and this place looks a little bit like this now. So under the current scenario of increased desertification, we think that understanding how life manages to survive and thrive in some of the Earth's most extreme environments is crucial to face the future and promote also social awareness of the dire situation that we face. And we chose the Atacama Desert to do this because, as you may know, it's a unique Earth system. Is between the Andes and the Pacific Ocean, and is considered to be the oldest and driest non-polar desert on Earth. It has experienced semi-arid condition probably for the past 150 million years, and hyper-arid for roughly the past 10-15 years, due to a combination of geologic and atmospheric conditions that we won't go in detail now. In addition to extreme low water availability, there's also extreme daily temperature oscillations, high salinity, particularly downslope, high radiation. In fact, this is considered to be a hotspot for radiation in the planet. And in addition, which was also one of the things that brought me into this environment, the soils are quite poor, very low in nitrogen primarily, but also in some other nutrients that make life difficult here. Despite all these constraints or these harsh conditions, if you know where to look, there is actually a surprising plant diversity. Some of the species that we have found, I'm showing here in these pictures, so you can have a sense of the different lifestyles and habits and types of plants that you can find. And I'm talking about the open land, right? Not on oasis, which is quite a different story. So what we have been doing for now more than 10 years is that we have been studying a transect, which is located, well, here to orient you, this is South America. This is the north of Chile, where the Atacama Desert is located. And we are working on this altitudinal transect that is east of the Atacama Salt Lake, which is shown here. And basically, we go from 2,400 meters above sea level, which is the limit for absolute desert. If you go below this point, there's just no water to sustain macroscopic life. And the upper limit is 4,600 meters above sea level. If you go above these levels, it's just too cold to sustain macroscopic life. So between these two extremes, the absolute desert and the upper limit for plant life, we have some plant life that we have been characterizing along these 22 sites that we have GPS tagged and that we come back every year since 2010. And I think this also has been a great collaboration between a number of groups. Perhaps the most important is Gloria Curusi, with which we have done phylogenomics analysis that I'll mention later in this talk, but also Mauricio Gonzalez and Chilean researchers, for example, for microbiome analysis that I won't have time to talk about. Janet Wolf from New Zealand, where we have done reconstruction of the flora for the past 30,000 years using fossil records, and a number of other people that have contributed in different areas and with which we're developing different projects. This video is just to give you a sense for how we work there. So we take, like as you can see here, liquid nitrogen and all the implements to collect plant material, to do some experiments as well at the different places that we have tagged in the transect. So we have done significant, I would say, significant characterization of the environment. So we have two meteorological stations that we have measured or determined environmental parameters for 4 years of continuous measurements. We take a picture every hour, and this is ongoing. So we have measurements such as diurnal temperature, solar radiation, rainfall, soil water content, and other parameters that I'm not showing here. Perhaps I would like to just highlight that in addition to that plants have low water availability, the rainfall events occur primarily in February in here. So it's very sporadic water where plants have to take advantage of that and complete their life cycle basically and wait for the next event. So it's not only little water, but it's also sporadic that they have to deal with. We have done also characterization of the soil substrate to understand what the plants are looking at in the soil in terms of particle, which is primarily sand across the entire transect, 80% of sand. And here we have different stations, and we have also measured the soluble fraction in the soil in these different stations. And so, for example, we have a pH gradient with alkaline soils towards the bottom of the transect and acidic soils towards the top of the transect. And correlated with this is, for example, metal availability, which tend to be more available at the top of the transect in acidic soils and less available at the bottom of the transect. Here on the top graph, I'm showing nitrogen, phosphorus, and potassium, important macronutrients, and perhaps the most important aspect is that, as I mentioned already, nitrogen is quite limiting, which is well below this red line that's shown here, 100%, which is considered to be the minimum necessary to sustain plant life. So it's quite a limiting amount across this entire transect. So maybe as a summary, so as you go up the mountain, there's slightly more water, there is a decrease in temperature. Also, as I mentioned, the radiation and across the entire transect is low nitrogen availability. So this combination of environmental factors define three ecosystems in Andean Atacama. The high steppe, which is dominated by perennial grasses, as you can see here. The puna, which is dominated by shrubs and subshrubs, and it's the most rich, biodiversity-rich ecosystem. And the prepuna, which is the most arid ecosystem, which is dominated by annual plants and succulents. So one of the first questions that we had was, what is really the biodiversity that we find here along this altitudinal transect? And this is not so easy to answer in extreme environments because many of these species have strategies to basically escape when the environmental parameters get too harsh. So we combine, I'm going to make a long story short, this was already published. We combine a number of experimental strategies, botanical surveys over a 8-year period, which is summarized in this graph. So here are all the different species, which are grouped by annuals, herbaceous perennial, and woody perennials. And combined with these botanical surveys, we did metagenomic sequencing of soil DNA. We did also pollen analysis and soil seed bank, which is basically see what it can germinate from soil samples and then identify the plants that germinate. So altogether, we identify 93 taxa and 78 plant species in these 3 ecosystems. And this is important because it provides a baseline or a photograph of the current biodiversity that's found in these environments and can allow us to compare with measurements either for the future and also from the past. And in particular, we have done the construction, as I mentioned, of the last 30,000 years, which goes into the last glaciation event and allow us to see how the flora change in that climate change event from the last glaciation to the current interglacial period. And I won't have time to talk about that either, but this is already published if somebody is interested to look it up. So what I want to talk about is what we have done with the 32 more relevant species that we identify out of these 93 taxa or 78 plant species that we have found. And these 32 were defined based on 3 criteria: the coverage, so how much of the land they cover, the persistence, so how often we find them when we go to the field, and also the distribution. And as you can see here, these are primarily angiosperms. There is 1 gymnosperm, which is Ephedra americana, but all the other plant species, the main plant species, are angiosperms. And we have different plant families as indicated here. So Poaceae, Polygonaceae, Amarantheaceae, Fabaceae, and so on. They have different photosynthesis types, life forms, and some of them are endemic also, which is also interesting because these are species that are only found here. In this graph in the middle, we have the altitude again from the bottom of the mountain to the top of the mountain, and the color here indicates the persistence, so how often we find these species along this transect in the different locations that we study. Here I have pictures so that you can have a sense for the different type of plants that we have characterized. And I'd like to emphasize the fact that we have a number of Poaceae, for example, Solanaceae also, and Fabaceae, which of course are relatives of plants that are important crops for humans that may be interesting also if we think about later on translating some of the knowledge that we have acquired here to engineering crop resilience or improve crops in some way. And in particular, for example, we have Solanum chilense, which is a relative of Solanum lycopersicum, right, the tomato. So you have a sense for the analysis that we have done. I like to make a pause here and look at this picture. And this is one of the stations that we analyzed. And based on the criteria that I described, the coverage, the persistence, and the distribution, basically what we have done is that we have sequence information for all the species that you can see in this picture. So if you go to this area in any of these stations, everything you can see, basically, we have some sequence information for those species, because all the other ones are either very hard to find, or they are, very, they have very short lifespans, and they come up under very specific environmental conditions and so on. So, but this should be the most important plant species ecologically, or at least we think, because, at least they are here most of the time and in great numbers. So the question that we are trying to answer is, what are the features that these Atacama species have that allow them to survive? And here we collaborated with Gloria Carusi at the Center for Genomics and Systems Biology in New York University to analyze the sequence information we obtained for these plants. We sequenced with Illumina the transcriptome of all these 32 Atacama species. And I have to mention, Trantivarala was very important to do the phylogenomics work that I will describe, and Gil Eschel to do a positive selection analysis that I will also describe in a minute. So basically, the information we have to start were 889,897 total protein coding genes. These are more than 486,000 plant protein coding genes and more than 300,000 new protein coding genes. So it's a huge data set that we had to somehow narrow down to interesting candidates in some way or another. So what basically we did was to compare these 32 Atacama species to what we call sister species. So these are phylogenetically related species to the ones that we find in Atacama, but are species that live in other environments, some of them in mesic environments, but they for sure they're not in Atacama. And we also included 6 model species because this brings annotation information that we can use, to understand what gene families are and what may be doing and so on. We used what's called the big plant pipeline to define orthologs and reconstruct relationships and use these phylogenetic relationships to narrow down candidates that can explain adaptation in Atacama. So the input to this pipeline are these 70 species. This is more than 1.5 million sequences from these 32 species from Atacama, the 32 sister species, and the 6 reference genomes. And so the first part of this pipeline, which is shown in this row, it's basically to build the gene family tree. So it starts with the gene clustering, then alignment using MAFT, and then gene family trees are built using TNT to basically identify orthologs for all these gene families in each species. So with the orthologs, we construct a phylogenetic matrix that has basically all the species in the rows, and in the columns we have the different orthologs for the protein orthologs in all the species, and as indicated here, the amino acids, right? And with this big phylogenetic matrix, we built what's called the total evidence tree. And from this phylogenetic tree, then we can identify the characters that explain the divergence or the separation between, for example, an Atacama species that could be in this branch and a sister species. And from these characters that differentiate these species, then we can go back to the gene families, see what they encode, what kind of biological processes they are involved in, and in this way try to understand the processes, the biological processes that may explain survival in Atacama. So we did a number of analyses, but in the end, we opted for PBS, for positive selection analysis, or DNDS methods that I'll describe, and the results that we obtained from that. So again, some numbers. So we have 32,292 families to build the phylogenetic trees, and I have to emphasize this was a huge computational challenge to build a phylogenetics tree, and also the total evidence tree, which was done by the NYU people, particularly Kranti Barala and others at Gloria Cruz's lab. So we basically obtain a phylogenomic tree as the one indicated here, which matches the taxonomic tree, which is, I guess, is good. And basically, as you can see here from, we have the Atacama species in bold, like Jaraba frigida and Nocella nardoides, and the sister species that are in normal font, such as Stipa grandis, right? And so basically, we're standing in these nodes and trying to understand what separates these Atacama species from the sister species in every case, and see whether there's similar gene families that appear in different branches of the tree, to see perhaps examples of convergent evolution and so on. So again, close to 1.7 million proteins, 15,972 orthologous genes, more than 8.5 million amino acid characters. And just to run, once we had the pipeline optimized, we put it through a supercomputer, we were able to do this analysis in 1.5 months. And here again, Kranti Barala was instrumental to do this, and he's now an assistant professor at Purdue University. So once we had this phylogenomic tree, Gil took that tree and ran different kinds of DNDS analysis, positive selection analysis. We did tree-dependent DNDS modeling using CodeML branch site models, so species-independent adaptation, and also ancestral adaptation. We also did tree-independent DNDS calculation using SNP genes, where we compare different groups. So, and the groups are indicated here. So we compare, for example, the drought adapter to the mesic sister species. We compare also low nitrogen adaptation to high nitrogen adaptation. and so on. So all the comparisons are indicated here. And so from all these analyses, we identified 265 gene families that are under positive selection in the Atacama plant species, which are summarized in this graph. So here on the top, we have the different species. So in this case, the Atacama species are in red, the sister species are in black, and the model species such as tomato, for example, we have here Arabidopsis are in blue, okay? And the plant families are color-coded as in other figures that I showed. So we have Poaceae here is a big group, Astereses also is another big group. And in fact, about a third of the gene families that we found under positive selection are plant family specific, and they are primarily from the Poaceae, and also some of them are from the Astereaceae. So the rest, the 64% or so, are present in 2 or more plant families. So here in this red, blue, and green columns, we have the different evidence from the positive selection analysis that we did. And here I think it's interesting also that we have the expression level for these gene families, which is color-coded. The browner or reddish color indicates very highly expressed genes relative to the transcriptome of the species that from where it was obtained. And as you can see, many of them are highly expressed. So not only under positive selection in terms of the protein sequence, but these are also genes that are very highly expressed. So likely they play a role, and perhaps they may also have some evolutionary regulatory elements as well. Okay. So now to understand what these gene families may be doing, so first we look at PFAM domains. Out of the 265 positively selected genes, we found 177 of them had a PFAM domain, so 67. And what was interesting is that many of the positive selection sites that we identified were inside their domain. So this doesn't prove, but it's evidence that correlates with the fact that this positive selection may have an impact on the protein functions, right? And some of the PFAM domains that we found overrepresented are shown with these blue star, like DNAJ domains, acetyltransferase, zinc fingers, and there's a number of other PFAM domains that have the positive selection sites, some of them inside the domain, others outside, as color-coded here in red and blue. So there's a number of different functions that are affected potentially that are relevant for adaptation. Another way to look at the function that these gene families may be involved in is to look at the gene ontology annotations. So out of the 265 positively selected genes, we found 180 that had a Go term annotation, and they are shown here. And they are grouped to the parent terms to better understand the different functions. So we have an important cluster that relates to response to stimulus. Also, importantly, there's many Go terms associated to metabolic processes. Some of them are overrepresented, and in particular, cellular nitrogen compound by a synthetic process, which probably makes sense considering there's a very limiting nitrogen environment. But there's also things that one might expect, like response to light stimulus, response to reactive oxygen species. As I mentioned, this is an environment with a lot of radiation, response to salt stress as well, and so on. And finally, as a way to try to validate some of the positively selected gene families that we identified, we look at the data from Arabidopsis orthologs to try to see whether any of these genes have been characterized in Arabidopsis, and in fact, we found a number of them that they were. So, particular related to enhanced nutrient use efficiency. So we have a number of positively selected genes associated to this biological process. Some of these genes, like this, for example, WRKI1, has a stress tolerance phenotype that has been described in Arabidopsis. We also have gene families related to optimizing water capture. In this case, several of them have been associated to stress tolerant phenotypes, either by mutants or overexpressors. We have also genes involved in wood hair growth, and we are following up on this in collaboration with Jose Manuel Esteves. Biotic interactions, for example, also flowering time, which also makes sense based on how probably quick they have to respond to water events, to germinate, complete the life cycle, and basically escape, particularly in animals. In photosynthesis, a number of genes involved in photosynthesis or protecting chloroplasts and also improving photosynthesis. And finally, also a number of genes that have to do with coping with high radiation, both related to scavenging gene, reactive oxygen species, DNA repair, and other functions. So overall, we found phenotypic evidence for 59, so about 30% of the positively selected genes, and many of them are also highly expressed in multiple Atacama species, which is indicated with this orange star, as shown here. So these are not only genes that are under positive selection, again, in the protein sequences, but they are also, they have high expression levels in the field, in the Atacama species, and many of them also shown in this blue bar with the red dot have positive selection within the BFAM domains that I described. So with that, so I would like to say that we have used big data set, in this case, an ecosystem level or ecosystem wide approach and integrative bioinformatics and modeling, in this case, phylogenomic modeling to build hypotheses. And now we have so many of them, but we hope that some of these gene families that we have identified in Atacama may be relevant to, as I said at the beginning, to engineer crop resilience or to improve carrying crops to make for a greener future, as shown in this image. So with that, I think I'm out of time. I would like to acknowledge the people in my lab, and in particular, Viviana Arauz, which was instrumental for all the functional analysis of the PSGs that Gil identified. This project was funded initially by the Howard Hughes Medical Institute as an experimental sort of, or I would say pilot experiments. Then we had funding from the Center for Genome Regulation, a program in Chile, and then later on from the Department of Energy in the US. And here I have some pictures from different field trips. So every year we would have different crowds, except for this guy here, Claudio Latorre, which is my friend and colleague from the ecology department. Without him, this project would not have been possible. This is an expert in Atacama, and we've been working together since the beginning of this project. In all the papers that we have from Atacama, Claudio and I have been working on this. So this is 2017, 2018, 19, and so on. Again, I would like to thank the Evonet team, primarily Gloria's lab, Kranti Varala, and Gil Deschel for all the phylogenomic work. There's people also in Cold Spring Harbor, Richard McCombie, involved in sequencing of related species in California desert that were identified by Dennis Stevenson from the New York Botanical Garden. Rob Desa also helped us a lot on the phylogenomic analysis, and Jean-Michel Anea and Heidi Cutler. I didn't have time to talk about that, but they're doing validation of some of the positively selected genes in plants of interest, such as maize and Brachypodium and other species. Again, Claudio Latorre was instrumental to be able to do this project. So he took me to the Atacama Desert in the first place, and I didn't have time to talk at all about Mauricio Gonzalez's work, which involved characterization of the microbiome associated to the Atacama species that we have done, and also the soil microbiome and comparisons between the microbiome in the plant to the microbiome in the soil. But you can find this in the publication that we just got this accepted in PNAS. So we have that information there as well, if you are interested. And with that, I would like to finish, and I'm happy to take questions if there are any. And I leave you with this quote from Raúl Zurita, Chilean poet, related to Atacama. Thank you.
Thank you so much, Rodrigo. So thank you for your fantastic talk. It was really nice and interesting. So let's take advantage that we are going into lunch times, and so we can take some questions if somebody has questions. Luis Cárdenas, do you want to to tell your question, or raise your hand and I can give you, or do you want me to? Okay, so Luis Cardenas says, do we know if those plants growing in the Atacama Desert do mycorrhizal association? And also, I would like to know if there is anything about the microbiome.
Okay.
Yeah.
So for the first part, so the mycorrhizae, yes, for sure. Many of them have. This is not something that we're doing, but I have a former student from my lab that she has now her own lab, and she's characterizing the fungi associated to the Atacama species, not only mycorrhizae, but also other types of fungi. With regards to the microbiome, even if I was not prepared, but I just briefly, so these are some of the results that you can find in the publication. But as I mentioned, we did characterization of the root zone microbiome using 16S ribosomal. So you can see there are differences as compared to the bare soil in terms of the relative abundance of different taxonomic groups. But I think the most interesting part relates to the predicted functions that we find associated to the roots. And in particular, we find, for example, functions related to nitrogen fixation, which we then validated with the NIFH marker, which suggests to us, again, this doesn't prove, but I think it's interesting evidence that points to the fact that nitrogen fixing bacteria that are recruited to the plant root system, it's important to provide the nitrogen, which is, as I mentioned a couple of times, it's quite limiting in this environment. So this is as much as I'll talk about this, but again, I invite you to look at the paper.
Okay, thank you. So let's hear the question of Maggie. You can.
Yes,
Okay.
yes, I am Margarita Collazo.
Okay.
Please, I have one curiosity. In one of the graphs, you present Solanum lycopersicum as present in the Atacama Desert. And this was very curious for me, how it could be present there. This is a cultivated plant. And how can you explain that?
Yeah, sorry, it's not, maybe I misspoke, but it's Solanum chilense, the one that we find in the desert. We use Solanum lycopersicum as a sister species that, or in this case, one of the model species that we included in the analysis to compare. But Solanum lycopersicum, we don't find in Atacama. We find other relatives, like Solanum penelli, not in this area. In other areas, it's also penelli and chilensis in these parts.
Okay, thank you very much.
Yeah, sure, no problem.
Okay, so now the question of Patricia Leon. You can.
Yes, hi, Rodrigo. Very nice talk. And I was wondering, among the positive selective genes, could you tell me, like, there are more putative transcription or regulatory proteins versus biosynthetic pathways, or they are the same, or what is?
No, they're not particularly more. I would say we find probably about the ratio that you would expect based on the abundance of transcription factors in general, like in a plant genome. So maybe 15–20%, something like this, maybe like transcription factors. There are other regulatory proteins like kinases, for example. I don't have the numbers of those, so I don't know. But transcription factors in particular were not something that stood out as, you know, an important category that
right
selection.
Okay, thank you.
Yeah.
Okay, so I'm just going to read two comments that congratulate you. Leticia Peraza says, congratulations, very interesting information and work for the future of agricultural food security. And Ana Lilia Arroyo Becerra says, congratulations, great talk. Are you beginning proteogenomic and super pangenomic studies to identify specific genes from stress resilience?
Yes, right. Thank you. Well, thank you for all the your appreciation of the work. What we're doing now actually is focusing on metabolomics. And we have done extensive metabolomics work in collaboration with the group of Yves Gibbon and Dominique Roland in University of Bordeaux. And there's some interesting results there related to specific, I would say, metabolic signatures that are found along different species from different phylogenetic families that seem to be important for adaptation also in Atacama. And this is something we're currently, is under review, so we hope it will get out soon. This is one of the areas that we are focusing on. The other one is facilitation. And so this is, how different plant species may collaborate, help each other to survive. That's another aspect that we are studying, and we are following up in specific plant families, particularly in the Fabaceae class, because I think they're very important. Also, that we have some potentially nitrogen fixing plants here that may be able to also be drought tolerant and so on.
Okay, so thank you very much, Rodrigo. So we are going out of time, and people wants to eat, I suppose. So thank you very much for accepting the invitation, and I hope to see you soon.
Okay, thank you very much.
Bye.
See you. Bye.
So for everyone, we are going to lunch time, and so let's go back at 2:45 for flash talks. See you soon. Bye.
abstract group, you don't have any problem there. If you take some mistake, please let us know that in order to support the abstract group. Thank you. the first flash talk session this afternoon, please check the abstract book to see if everything is right with your abstract and your names. But if you have some problem there, please contact us as soon as possible, because we have to finish the abstract book, and maybe we have just one day to finish it. Thank you.
Welcome to my talk on the structural characterization of plant respiratory complexes. In order for plants to survive, they must carry out two essential metabolic processes: photosynthesis and respiration. These processes, as you know, provide the plants with the energy and metabolic intermediates they need to live and grow. While we understand the proteins of photosynthesis in great biochemical detail, our knowledge of the proteins of plant respiration has lagged behind. If we want to fully understand the mechanism of plant respiration to be able to, for example, manipulate it to engineer better crops, we need to overcome the challenges and obtain a biochemical and structural understanding of plant respiratory complexes. Respiration in plants, as in all eukaryotic organisms, occurs via an electron transport chain in the inner mitochondrial membrane. This chain is composed of 4 large protein complexes called complex 1 through complex 4 that pump protons for the generation of ATP by ATP synthase. The complexes can exist individually, or they can come together into higher order assemblies called supercomplexes. At the start of my postdoc, we did not have many biochemical or three-dimensional structural details for these complexes in plants. Therefore, the goal of my project was to obtain the high-resolution structures of plant respiratory complexes using cryogenic electron microscopy, or cryo-EM. To overcome the challenges of working with the limited amounts of plant mitochondrial samples, we developed a non-traditional approach to obtain the structures. So instead of purifying each complex separately, we used a biochemically mixed fraction from a sucrose gradient as the cryo-EM sample. Using this strategy, we were able to obtain high-resolution structures for a complex 1 assembly intermediate, shown here, called complex 1 star, as well as complex 3 dimer, complex 4, and the supercomplex 3,4 from one single cryo-EM dataset. These structures allow us to start replacing the cartoons with molecular details, showing us previously unknown features of plant respiration. For example, the structures resolved subunit compositions that had been unclear from genetic and mass spectrometry analysis. For instance, our structure of complex 1 star identified that complex 1 gamma carbonic anhydrous domain, shown here, which is not found in yeast or mammals, is a heterotrimer, and revealed which 3 of the 5 highly similar carbonic anhydrous proteins found in plant actually form part of the domain in this complex. Similarly, our complex 4 structure showed that plant complex 4 has 10 subunits, which is in contrast to other organisms, and that 3 subunits previously thought to be plant-specific, these ones here on the right, are actually homologs of subunits in mammals and yeast. Our structures also showed plant-specific arrangements of the complexes, such as several new structural features in complex 3's catalytic matrix domain, also known as the MPP domain, as well as the plant-specific interface between complex 3 and complex 4 in the supercomplex. In addition to studying the static structures of these complexes, we looked at their movement. Plant complex 3 has complex-wide long-range motions, showed here in these videos, which we hypothesized may be the physical basis for the coupling of complex 3's respiratory and non-respiratory functions in plants. In conclusion, these first high-resolution structures of plant respiratory complexes and the supercomplex identified several new plant-specific features of respiration and set the basis for new functional experiments to better understand the mechanism of respiration in plants. So with that, I would like to thank everyone in the LEDS lab who contributed to this work, as well as our funding sources. And thank you very much to the selection committee. And thank you very much for your attention.
Hi, I'm Jorge Solis, and I'm going to talk to you about the project I did in Carmen Quinto's lab titled "The RALF1, RALF6, FER1 module differentially regulates the optimal number of nodules in common bean roots depending on the nitrogen status of the soil." Nodulation is a process established between legum roots and rhizobia and ends with the formation of a nodule able to fix atmospheric nitrogen. This is a very energetically expensive process for the plant, hence the plant has several pathways that control the total number of nodules formed. The best characterized are autoregulation of nodulation and nitrogen-mediated regulation of nodulation. We wonder what could be the role of family of peptides called RALF peptides and the receptor pheronia during nodulation. We determined that RALF1, RALF6, and FER1 are expressed along all nodulation process under low nitrate conditions that allows the nodulation process but also under high nitrate conditions that inhibits nodulation. We evaluate the effect of silencing and overexpressing RALT1, RALT6, and FER1 during nodulation in low nitrate conditions and observe that silencing RALT6 and FER1, there is a reduction in the nodule number at 21 days and 31 days post-inoculation, and overexpression of RALT1 and FER1 increase the nodule number at these same days. We also observe that there is a correlation between the expression of genes associated to the autoregulation of nodulation and nitrate-mediated regulation of nodulation pathways with the change in nodule numbers in these two conditions. Then we evaluate the effect of high nitrate in these transgenic silencing and overexpressing routes and observe that silencing any of the three genes reduces the effect of nitrate in this inhibition of nodulation, while overexpression of RALT1 and RALT6 make this route more susceptible to the inhibition. We again observe a correlation between some of these genes from the autoregulation of nodulation and nitrogen-mediated regulation of nodulation pathway with the change in the nodule number in these transgenic routes. With these results, we propose that RALT1, RALT6, and FER1 has a negative role on the regulation of the autoregulation of nodulation pathways and the nitrogen-mediated regulation of nodulation pathways under low nitrate conditions, but has a positive role in these pathways under high nitrate conditions. Thank you.
Mine tailings are solid wastes of mining operations, and because of their extreme pH and high concentrations of heavy metals, they are considered toxic. A cost-effective strategy to bioremediate these sites is phytostabilization. Phytostabilization consists of the establishment of a plant canopy that encapsulates these contaminants. Bacteria help in these strategies because of their plant growth promotion and heavy metal cation immobilization potential. That's why we went to Nacosari de Garcia, Sonora, where we can find a mine tailing in the middle of a city in which a community of plants has grown in the wild, and it is dominated by these 4 species. We took root samples from these plants for 16S gene amplicon sequencing. We also took isolates, which we combined in a synthetic community, and then performed a mesocosm experiment in which we selected only the bacteria that could withstand the toxic substrate, that could consume plant-derived nutrients, and also withstand their intra-community interactions. We also performed whole metagenome shotgun sequencing in 1 environmental and in the 2 synthetic communities. When looking at the results, the taxonomic profiles revealed that all samples were dominated by Proteobacteria and Actinobacteria, and the diversity measured with the Shannon Entropy Index was decreased from the environmental to the cultivated communities. When looking at the functional profiles, we saw that the carbohydrate metabolism, membrane transport, and stress response functional classifications were enriched after the mesocosm experiment. Finally, we wanted to look at the raw plant growth promotion potential of the final synthetic community. We saw that the plants inoculated with this community and sterilized substrate showed the highest relative growth rate in comparison to all other treatments. So our conclusions are that mine tailings microbes are microbiomes are dominated by Actinobacteria and Protobacteria. A mesocosm experiment enriched genes related to carbohydrate metabolism and stress response, and the final synthetic community inoculated plants showed higher relative plant growth rates in sterile substrates. This means that it could be useful in a bioremediation strategy in the future.
Carbon concentrating mechanisms impose are key elements for terminal drought resistance in common bean. Common bean is one of the most consumed legumes which is affected by terminal drought. Terminal drought is a drought condition that impacts crops during their reproductive stage and strongly reduce the dry bean production. But resistant cultivars show a more efficient carbon translocation to pods and seeds. So our interests have been to elucidate the molecular mechanisms involved in these processes. For that, our experimental strategy consists in greenhouse terminal drought experiments with two contrasting yield cultivars. Pinto Saltillo considered resistant and Bayo Madero considered sensible. These two cultivars were subjected to terminal drought stress, and at mid-pod filling, we obtained transcriptomic data and physiological parameters of what we call the functional module, which is the continuum of source, leaf, pod, and seeds. Altogether show us a model mechanism. What caught our attention was the higher differential in rich processes of recycled photosynthesis, photorespiration, and CO2 concentration mechanisms in bulk, and were induced by terminal drought in the resistant cultivar. This increase in process increased carbon translocation to seed, and in seed we saw processes as zinc strength and starch synthesis that allowed sucrose gradients and suggests that sucrose is translocated through facilitator transporters. These were more induced in bulk and seeds of the resistant cultivars. Our main message is that terminal drought resistance in common bean can be associated with high photosynthesis by CO2 recycling, high photorespiration, and CO2 concentration mechanisms in bulk.
Hello, my name is Elizabeth, and I'm currently in the 7th semester of the doctorate program from the Universidad Michoacana de San Nicolás de Hidalgo. I'm glad to show you the most outstanding results obtained in collaboration with Dr. Jose Lopez Bucio and co-workers, which title is Micrococcus luteus strain LS570 promotes root branching in Arabidopsis via decreasing apical dominance of the primary root and an enhanced auxin response. The main aim of this work was to find bacteria with plant-promoting traits in Arabidopsis. We obtained an interesting actinobacteria isolated from alkaline soil of root maize crops in Lometón, Tarímbaro, Michoacán. The strain LS570 was closely related with Micrococcus species through the 16S gene sequencing. We were able to capture electronic micrographs of the bacteria, which had around 2 micrometers in diameter, grouped in tetrads. We first investigate the Micrococcus potential to promote plant growth in Arabidopsis seedlings by direct contact, which is a fast and suitable method to distinguish between pathogenic and beneficial effects. In this regard, we found out Micrococcus causes major changes in the primary root system. It helps primary root length, increased lateral root number, density, and length. Overall, increasing root fresh weight. We hypothesized that auxins were involved in these bacterial defects with plants. So we next examined the plant auxin response through transgenic seedlings expressing the auxin-inducible construct, DR5-UIDA. And we observed a strong auxinic expression in the shoot, lateral roots, and the root apical meristem until the 4th day after bacterial interaction. Concluded that Micrococcus triggers morphogenic changes in the root system via auxins. To explain the primary root shortening in inoculating seedlings, we measured the meristematic zone and the cortical cell length, and we found that Micrococcus decreases both the cell mitotic activity by 3-fold and cell elongation by 5-fold. Finally, molecular components of the signaling pathway unveil a new role of ARF7 and ARF19 in root elongation with Micrococcus, which showed resistance in the primary root length repression. Also, solitary root and auxin receptors were crucial for lateral root branching and primary root growth with Micrococcus. Thank you.
Plant species that produce edible fleshy fruits are susceptible to be infected by plant viruses. In this work, we identified more than 2,000 of plant species that produce edible fleshy fruits, the vast majority of them classified within the clade of euticots. Only a tiny fraction of them are consumed by people. However, there are 64 edible fleshy fruits in total that play an important role in the global marketplace. More than 600 viruses infect these 64 edible fleshy fruits. Around 70% of these viruses contain RNA genomes, whereas 30% possess DNA genomes. 15 virus families encompass the majority of these virus species, which are the same, exhibiting a wide host range. An important finding of this work is that euticot species are infected by a higher number of plant viruses than monocots and magnolids.
Virus infection can be local or systemic. The extent of the infection is attributed to several factors. The most important is the plant-virus relationship. The main organelle affected is the chloroplast, and changes in its morphology generate biochemical changes like this. These changes at cellular level are reflected in symptoms causing poor sensory and visual quality of fruit. Agriculture is one of the most important economic activities in the world. 34% of the world population depends on agriculture. Fruit production accounts for 16% of total, and it has been increasing year by year by 1.4%. Fruit production can be affected by different viruses, and the plant infection can be hastened by several factors. Viral disease in crops produce low or null yield, economic losses, and compromise the food security of the population.
Viral diagnosis includes serological and nucleic acid-based assays. In serological assays, the computer-assisted epitope identification has shown to improve antibody production, which is convenient to reduce the costs. Other techniques are based on nucleic acid amplification, many of them by PCR. Microarrays and next-generation sequencing allow the identification of hundreds of viruses at a time, and new viruses and viroids. Biosensors are portable devices to detect viruses in a fast way on-site. The correct disease management involves a combination of strategies, including horticultural practices such as the use of disease-free propagating materials and seeds, nutrition, intercropping, nuclear embryona, and others. Other strategies are vector, biological, and chemical control, thermotherapy, the use of disease-resistant varieties, and quarantine and legislations.
Plant cells are surrounded by a rigid extracellular matrix. During development, cytoskeleton, plasma membrane, and the wall must coordinate in order to allow cell expansion. Here, we investigated the role of class I formants as potential molecular linkers of this cellular structure. So class I formants are transmembrane proteins that possess intracellular domains forming homology that have actin nucleation activity. These proteins also possess extracellular domains that are thought to interact with the cell wall. However, our understanding of the function of these domains is limited, and that is the central question of our study. We focus on two members of the family, formin 5 and formin 3. These are highly expressed in elongating pollen tubes and exhibit this distinct localization pattern, with formin 5 being restricted at the elongating tip and formin 3 localized throughout the pollen tube periphery. So, based on these distinct localization patterns, we hypothesize that the extracellular domain might be implicated in establishing these distinct localization patterns. So here, I'm summarizing the main findings of our study. Through genetic analysis, we determined that the extracellular domain is necessary for protein plasma membrane localization. As seen here, when we eliminate the respective extracellular domains, we see depletion of the plasma membrane localization and increase in intracellular accumulation. So we additionally determined that these domains are necessary for protein function. Through biochemical assays, we were able to detect a post-translational modification in the extracellular domain of formin 3 that resembles arabinogalactan glycoprotein. So we found this evidence for this for the presence of these large arabinogalactan glycans attached to this extracellular domain. In the case of formin 5, we found that it is modified by the addition of this linear chains that are commonly found within the serine-proline repeat motifs in extensins. So additionally, depending on the type of glycan attached to those extracellular domains, we found through FRAP assays that it affects its lateral mobility in the plasma membrane, with formin 3 being the least mobile with these large arabinogalactan sugars. Further genetic studies where we eliminated this arabinosylation modification in pollen tubes show that it is necessary for maintaining formin 5 restricted at the apical plasma membrane. Thus, when we eliminated this modification, we found that it has extended its plasma membrane localization accompanied by actin disorganization. Taken all together, these results indicate that the extracellular domains of these proteins and the post-translational modifications associated to them are important to restrict these proteins to specific plasma membrane domains.
Hi, I'm Constanza, and today I will present the project that we are currently working on in Cuevas Velázquez laboratory. In our laboratory, we are interested in understanding how cells sense the changes in physicochemical properties of the environment and how these changes influence different aspects of cell biology. To shed light in this issue, our research group has developed a genetically encoded fluorescent FRET biosensor called Sensor Expressing Disorder Protein 1, SED1. This biosensor explores the ability and sensitivity of an intrinsically disordered region from Arabidopsis thaliana to reversibly alter its S structure in response to changes in osmotic status, to dynamically track effects of osmotic stress in living cells, transits from an extended conformation to a compact conformation throughout the increase in osmolarity. SED1 is capable of track the effects of osmotic stress in bacteria, plant, and mammalian cells. These findings reveal the potential of using intrinsically disordered regions for generating environmental activated fluorescent molecular tools. So, to take one step further, in my project, we exploit the sensitivity of multiple IDRs from different organisms to build a library of a new environmentally responsive biosensor for using it as a heterologous system. So the library was built using 200 different sequences from diverse organisms. And the library contains IDRs from all different kingdoms of life. And now is being synthesized. So the next step is to screen the 200 biosensor library that is going to be expressed in GIST, and we are going to apply osmotic stress in the screening to obtain new biosensor with novel characteristics in terms of sensitivity and dynamic range that can be used in several organisms. And that's all. Thank you.
Good afternoon. I'm Emmanuel Flores Hernandez. I will present to you my experimental work titled Participation of the Arabidopsis thaliana PUT2 gene encoding a polyamine transporter in the plant defense response against Pseudomonas aeruginosa. In plants, polyamines participate in the establishment and regulation of the defense response through changes in their metabolism, specifically biosynthesis, catabolism, and conjugation, that generate important mediators in the plant defense system. However, it is unknown whether polyamine transport has an implication in the biotic stress response. In the present study, we analyzed the importance of the polyamine uptake transport 2, ATP2 for short, in the interaction between Arabidopsis thaliana and Pseudomonas aeruginosa Pato tomato DC3000. First, using a promoter repertoire line, we found that ATP2 gene expression increased 6 hours after inoculation with Pseudomonas. This result was confirmed through the quantification of the mRNA levels by QRT-PCR. In addition, We evaluate the phenotype of a tDNA insertional mutant line in response to Pseudomonas. We observe a diminution in the bacterial titers in the mutant line in comparison with the wild type 72 hours after inoculation. Finally, the mutant line had an increase in the expression levels of the PR1 at 6 hours after inoculation with Pseudomonas. Both PR1 and ICS are important markers of the salicylic acid pathway involved in the plant response to biotic stress. In conclusion, the results show that ATP2 is an interesting candidate to further evaluate the participation of the polyamine transport in plant defense. Thank you.
Hello everybody, my name is Dulce. Thank you all for staying at the National Plant Biochemistry and Molecular Biology Congress Flash Talks. Today I'm going to present you the project Isolation and Cellular Identity of the Megaspore mother cell in Arabidopsis thaliana that I worked during my master's program here in Simvestab, Irapuato, Mexico. Let me introduce you the Megaspore mother cell, or MMC, that is located in the apical pole of the ovular primordium, in this part. This cell is relevant because it's the initial female germ cell and meiotic precursor of Arabidopsis thaliana. Because this cell is slightly covered by sporophytic cells within the ovular primordium, the MMC is of the difficult isolation. As a consequence, only few genes related to its differentiation and unique identity have been identified. One of the molecular pathways involved in the establishment of the MMC is the RNA-dependent DNA methylation. This pathway is in charge to establish a new methylation marks and to maintain them, in plants principally within the transposable elements. It has been observed that when mutating components of this pathway, more than one ectopic cell reminiscent of MMC differentiate in the apical pole on the ovular primordium. With this background information, we would like to answer two questions. What are the methylation patterns in the MMC and how ectopic cells emerge in the mutant ovules of the RDDM pathway? To address this question, we have established a protocol that allows the isolation of MMC population using specific treatments with different enzymatic digestions. Our results suggest that our protocol can be used to isolate sufficient germ cells from female organs to conduct whole cell-based transcriptome and methylome analysis in Arabidopsis thaliana. We also investigate the possible origin of the ectopic cells in the RDDM mutants using a molecular marker that allows the distinction of meiotic divisions. We evaluate early developed in 5 stages, confirming the absence of mitosis in the MMC and adjacent cells in wild-type ovules. This result suggests that the origin of the ectopic cells is not the result of mitotic division of a previously differentiated MMC. Well, thanks to everyone for listening today, and don't forget to contact me to know more about this beautiful project. Bye.
Thank you, all the Flash Talks contributors. You did a very nice work and presented in a very short time your very, very amazing and interesting work. Thank you all.
Okay, so we'll continue with our next talk. This could be a keynote conference that will be presented by Professor Shumei Chen from the University of California, Riverside. So Shumei, nice to see you.
Yeah.
So, the audience is yours, so start, please.
Yeah, thank you. Yeah, thank you, Alejandra, for organizing this keynote speech. And also, I would like to take this opportunity to thank all the organizers for putting together such a great program. I have been able to attend most of the talks, and I feel that the program is really exciting. So congratulations. And I look forward to more exciting talks.
Thank you so
in the
much.
next 2 days. So let me share screen to begin my talk. So can you see my screen now?
Yeah, it's there.
Okay.
Just
Yeah.
put it in present. Yeah, right.
Great, great. And let me pull up the laser pointer. Great.
Yeah. Good.
So today I would like to discuss a not so, I guess, well-known RNA modification, and that is the capping of RNA with this redox agent, NAD. So you all know that RNA is an information carrier between DNA and protein. But many of us also know that RNA is a lot more than an informational carrier. And that's because a lot of processes actually occur at the level of RNA, and these processes contribute to gene expression. So it turns out that RNA actually serves as the nexus of gene expression regulation. So RNA can be 5' capped or decapped, 3' polyadenylated or deadenylated, and RNA can even be oligouridylated. And all these processes would contribute to gene expression. And you also know that RNA can undergo alternative splicing, which contributes to different forms potentially of proteins. And RNA can also undergo internal covalent modifications, and they also have impacts on gene expression. So all these processes would later lead to effects on the translation, stability, or subcellular localization of the RNA, thus impacting gene expression. So today, the topic is going to be mainly on a non-canonical RNA cap known as the NAD cap. So first, I wanted to discuss the canonical cap, which is the M7G cap. So you all know that the canonical cap is basically a methylguanosine attached to the 5' nucleotide of the RNA via a 5' to 5' triphosphate group. So this M7G cap is added by capping enzymes after transcription initiation, but co-transcriptionally. So usually after 20 to 25 nucleotides have been made by the RNA polymerase, then this capping enzyme adds the cap to the RNA. And once added to the RNA, this cap influences all subsequent processes that this RNA undergoes. For example, in the nucleus, the cap is bound by the cap binding complex. And this CBC then interfaces with splicing, polyadenylation, and nuclear export. And then in the cytoplasm, the cap is bound by EIF4F, and this promotes translation initiation. And the cap also stabilizes RNA and by protecting it from 5' to 3' exonucleases. So the M7G cap is so important to the RNA. So as I discuss alternative caps, I think we should keep in mind what these alternative caps would be doing in terms of RNA metabolism. So in recent years, alternative RNA caps have emerged, and these alternative caps include NAD, NADH, DP-CoA, and FAD. So you can see that these are cellular metabolites. So the fact that these cellular metabolites can also serve as RNA caps suggests that cellular metabolism could interface with RNA metabolism. So today I'm going to talk about the RNA NAD cap, but I wanted to just put it up here, not that although these two caps, NAD and NADH, can be distinguished by certain means, the profiling approaches that I'm going to discuss cannot effectively distinguish these two caps. So I will be referring to these two as the RNA NAD cap. One thing that you want to notice is that all these metabolites are ATP analogs, and that is important when we discuss the deposition of the NAD cap. So because these caps, or because NAD is a ATP analog, it can actually be incorporated onto RNA as a initiating nucleotide during transcription initiation. So it has been shown that some bacterial RNA polymerases, a mitochondrial RNA polymerase, and eukaryotic Pol2 from yeast can use NAD as the initiating nucleotide, as the first nucleotide to synthesize RNA. And presumably that's how NAD is incorporated into RNA as the cap. In fact, structural studies from using a bacterial RNA polymerase together with the DNA template strand and the NAD RNA shows that the NAD moiety, as shown here, is actually quite well coordinated by amino acids in the active center of RNA polymerases. And because NAD is incorporated as the first nucleotide into RNA, it makes sense that in bacteria, promoter sequences influence RNA NAD capping efficiency. This has been extensively studied in bacteria, but whether this is the case in eukaryotes, it's still not known. In human cells, there's also some perhaps implications suggesting that NAD capping may occur post-transcriptionally, but the mechanism is still unknown. So the NAD cap was identified in year 2009 by David Liu's group. So in this particular LCMS study, what they did was to isolate total bacterial RNA, and then they removed the small molecules to retain the macromolecular fraction. Then they subjected the RNA to nuclease P1 digestion, and they used heat-inactivated nuclease P1 as a control. So after digesting the RNA into nucleotides, then they subjected the nucleotides to LCMS analysis, and they were able to identify NAD. And further analysis showed that NAD was actually at the cap, at the very 5' end of the RNA, so it's an RNA cap. So we decided to establish a similar approach, LCMS-MS approach, to test whether Arabidopsis RNAs also have NAD. So we did so using the instruments at the metabolomics core at UC Riverside. And we digested either total RNA or Arabidopsis mRNA with nuclease P1, and we were able to detect NAD in both total RNA and mRNA. And if you look at the amount of NAD in mRNA versus the amount in total RNA, we could conclude that NAD is primarily in messenger RNA. So this approach does not tell us which RNA species harbor the NAD cap. And NAD capture-seq came to the rescue. This technology was established in 2015. And in NAD capture-seq, NAD RNA is first turned into biotin RNA using two reactions. So the first reaction is an enzymatic reaction. The enzyme is ADPRC. So this enzyme converts NAD RNA to an alkyne RNA. And then in the second step, the alkyne RNA reacts with biotin azide to generate biotinylated RNA. And the biotinylated RNA can then be purified and then sequenced. So using this NAD capture-seq, it was shown that bacteria, yeast, and human cells harbor NAD-capped RNAs, which I will refer to as NAD RNAs. And using a similar approach, my lab and Brian Gregory's lab showed that Arabidopsis plants also contain NAD RNAs. So this is the NAD capture-seq pipeline that we used to study Arabidopsis NAD RNAs. I just wanted to point out that the first step, the enzymatic reaction, is really critical, and our negative control would actually be the reaction without the ADPRC enzyme. So the ADPRC minus samples would represent RNAs that are non-specifically attached to the bees. So we subjected inflorescence as well as seedling mRNA, seedling RNAs to NAD capture-seq in replicates. And then we were able to identify thousands of NAD RNAs. So this shows the read counts of the ADPRC plus samples as compared to the ADPRC minus samples, the negative controls. And by comparing these read counts in the multiple replicates, we were able to identify these NAD RNA producing genes from inflorescences and seedlings. And you can see that there is a good overlap between the two types of samples. And we looked into the features of these genes that produce NAD RNAs. So what we did here was to separate genes into 10 groups based on the amount of NAD RNA. So here, ADPRC plus minus ratio is a proxy for the levels of NAD RNAs. So basically, the 10th decile represents the genes that have the most levels, the highest levels of NAD RNAs. And then if you look at these deciles in terms of gene length or intron number, you will see that the genes that produce the highest level of NAD RNAs are shorter and they have fewer introns. And this, by the way, is also the case for human genes. What's going on? Stuck. Oh, I'm really sorry. Let me.
So you cannot there.
I think I cannot. Somehow I cannot move the.
Yeah, you can use the arrow to the right. You see there?
Oh yeah, yeah.
Okay.
Great. So when we looked into the genes that produce these NAD RNAs, we found that they are largely nuclear genes and some are mitochondrial genes. But intriguingly, we did not see any NAD RNAs from chloroplast genes. And when these genes are from the nuclear genome, they can be either protein coding or non-coding. And I should mention here that in humans, small RNAs and small nuclear RNAs can be NAD capped. But our seq did not see them because we had actually very low coverage of the SNO RNAs and snRNAs. But Brian Gregory's lab was able to see NAD capping of some of these RNAs. And from the browser views of the protein coding genes, it was very obvious that these NAD RNAs are properly spliced. As you can see from the gene model, we do see the exons being retained and the introns being removed. So these are the top three are the ADPRC plus samples, and you can see the ADPRC minus samples are very clean here. The NAD RNAs also appear to be polyadenylated. So we looked at this first by RT-qPCR following NAD RNA capture. And we looked at a few genes, and you can see that either, it doesn't matter whether we use the random primer or the oligo-DT primer, we were able to detect NAD RNAs. We also looked at this at the global scale. We first isolated poly-A RNA, and then we subjected the poly-A RNA to NAD capture-seq. And we compared this to the total RNA sample. And here we're plotting the ADPRC plus minus ratio to represent the levels of NAD RNAs. And you can see that there's a very good concordance between poly-A RNA and total RNA. And here I wanted to highlight NAD tag-seq from my collaborator E.G. Xia's lab. So they developed this really innovative approach. They first converted NAD RNA to alkyne RNA, which is the first step, as I talked about before, in NAD capture-seq. But the second step is different. So in the second step, instead of using a biotin azide, they used an azide that's covalently linked to a tag RNA. So afterwards, they would get a chimeric RNA, which is the NAD RNA and the tag RNA covalently linked. Then they subjected this to nanopore single molecule sequencing. So when you sequence using nanopore, then you start from the 3' end of the RNA and you sequence towards the 5' end. And if you reach the tag RNA, then you know that the original RNA was NAD capped. So there are several advantages to NAD tag-seq. One is that you can derive the ratio of NAD RNA versus M7G RNA. So if you focus on a mixture of NAD RNA and M7G RNA, for example, from a particular gene, then after this tagging, the NAD RNA will contain the tag RNA, but the M7G RNA would not. So after you have this mixture, you can do one of two things. First, you could actually purify the NAD RNA using a complementary DNA oligonucleotide and then sequence. The other approach would be to directly sequence this mixture. And if you do that, you can imagine that you could derive the ratio of the NAD RNA and M7G RNA. And when they did this, they estimated the ratio to be about 5%. But I will tell you later that this is likely to be a huge underestimate. Another advantage of NAD tag-seq is that you can actually obtain full-length sequence information because this is single molecule sequencing. So it's very clear that the NAD RNAs are spliced and polyadenylated. And interestingly, the NAD RNAs have shorter 5' UTR. And they have the same 3' UTR as M7G RNAs. But even though they have shorter 5' UTR, the NAD RNAs tend to contain the entire open reading frames. So this led to the question of whether NAD RNAs are translated because they have the entire open reading frame. So this question is not so easily addressable, at least not globally. So we decided to ask whether NAD RNAs are polysome associated, which is easier, more easily addressable. So what we did was to fractionate polysomes, and then we isolate polysomal RNA, and then we subjected polysomal RNA to NAD capture-seq. And at the same time, we also treated total RNA for comparison. As you can see, if you compare ATPRC plus minus read counts from polysomal RNA, we were able to find thousands of NAD RNAs. In fact, if you plot the NAD RNA level of polysomal RNA and total RNA, we actually saw higher levels of NAD RNA in the polysome, suggesting that NAD RNAs are preferably associated with polysomes. So next, we wanted to ask whether these are translating polysomes. So for that, we decided to treat extracts with pyrimycin. Pyrimycin is a inhibitor of translation, so it splits translating ribosomes into ribosomal subunits. You can see from the polysome profile that the treatment worked. So the polysomal fraction is reduced upon pyrimycin treatment, and the non-polysomal fraction is increased. So next, what we did was to isolate RNA from these two fractions, the polysomal and non-polysomal fractions. And then we performed NAD RNA capture and then real-time PCR to look at the levels of NAD RNAs. So you can see here in the mock treatment, the NAD RNAs tend to be enriched in the polysomal fraction as compared to the non-polysomal fraction. But after I'm having the same problem. But after pyrimycin treatment, this enrichment is either not there anymore or it's even reversed. So this suggests that NAD RNAs are associated with translating ribosomes. So next, I would like to discuss our efforts to further develop the methods to profile NAD RNAs. And in particular, I will be talking about a method that we call SPAC NAD-seq. So this is prompted by our realization that NAD capture-seq has some problems, and that's shown here that's shown by this plot, which shows the gene body representation by the reads. For example, if you were to do just regular RNA-seq or NAD capture-seq in the ADPRC minus samples, you would see very even gene body representation of the reads. However, in the ADPRC plus samples, we see this 5' skew in the gene body representation by the reads. And we looked into this, and we looked into the scheme of NAD RNA capture, and we realized that this is most likely due to the presence of copper in these NAD RNA capture reactions. Copper ions are known to cause RNA degradation. So you can imagine if you have a fragmented RNA by copper, and you also purify the RNA using the 5' biotin as a tag. So this would lead to 5' fragments. That's why you see the 5' skew in the outcome. So we decided that we should improve upon the chemistry so that we could eliminate copper to reduce NAD RNA degradation. We thought that this would also allow us to improve the efficiency of NAD RNA capture. So the click chemistry that was used in the original NAD capture-seq is called copper catalyzed azide alkyne cycloaddition reaction, which I will refer to as the CUAC reaction. So here, the azide is biotinylated, and then the alkyne is appended to the RNA using ADPRC. And you end up getting this covalent linkage between the biotin and the RNA. However, the reaction requires copper, which degrades RNA. So we decided to look into another alternative RNA capturing scheme. Oh, it's happening again. So another click chemical reaction called SPAC, strain-promoted azide alkyne cycloaddition, you don't actually need any catalyst, but the alkyne that's used is in a cyclic form. And my understanding is that because of the strain in the rings, the reaction can occur readily without the presence of any catalyst. Because this alkyne is very bulky, we thought that maybe it's not very easy to append the RNA to this alkyne. So we decided to try to see whether we could append the RNA to the azide using ADPRC. And then we would have biotin alkyne to generate biotinylated RNA. So first, to test whether ADPRC could catalyze the reaction between NAD and the azide, we decided to use free NAD. We also used as a control free NAD and the alkyne, for which we know a product would be made. And indeed, we were able to see a very specific product, and the mass signatures of these products are consistent with the nature of these expected products. So this means that ADPRC can indeed help to react NAD and azide. So then we decided to test this approach using Arabidopsis RNA. So here, what we do is to use either the CUAC reaction or the SPAC reaction to turn NAD RNA into biotinylated RNA. And then we run the RNA in the gel and detect the RNA with streptavidin HRP. So the signals that you see here represent biotinylated RNA. So now let's first focus on total RNA. You can see the SPAC reaction give rise to signals that are much larger than in size as compared to the CUAC reaction. So this is consistent with the fact that the CUAC reaction causes RNA degradation. And here you can clearly see the rRNA bands. So the rRNAs are showing up not because they're NAD capped, they're also showing up in the absence of the alkyne or the enzyme. So they must have some internal modifications that allow them to show up. And this is not a problem for mRNAs. So for mRNAs, these negative controls are very clean. And as you can see, for the mRNAs, the spark reaction gave rise to much higher signals as compared to the Kuak reaction, suggesting that the spark reaction is much more efficient. So we were really excited when we saw this result. However, our excitement probably lasted for 5 minutes because we thought that, such high signals, could it be because M7G RNAs were also reacting? And I should mention that NAD capture-seq was initially developed for E. coli RNA or bacterial RNA, but the technology was adapted to eukaryotic RNA without extensive evaluation of whether M7G RNAs also react. So we decided to look into that. So we first used M7G PPPA to represent M7G RNA to see whether this could react with the azide using ADPRC. And we predict that if they were to react, then we would have these two products. We would have M7G being released from this M7G PPPA. And we could detect the M7G using LC-MS-MS. And we were kind of disappointed to see that indeed M7G was detected. And it didn't matter whether we were using the azide or the alkyne in this reaction. So what this means is that the previous NAD capture-seq probably also had some interference from M7G RNAs. But you could also see that these peaks are pretty small because they are very close to the baseline. So this suggests that this reaction was not very efficient. So we decided to look into the relative efficiency between NAD RNA and M7G RNA in terms of being converted to biotin RNA. So we in vitro synthesized NAD RNA and M7G RNAs of different sizes, and then we mixed the two at different ratios, molar ratios, and we subjected them to these reactions. And the signals that you see here represent biotinylated RNA. So you can see that at a higher molar ratio, M7G RNA can indeed be converted to biotin RNA. We suspect that this was also occurring even with the Kuak reaction. It's just that the RNA degradation prevents the confident identification of the M7G RNA signal. So then we thought that maybe we should try to remove M7G RNA. And there is a commercial antibody, anti-M7G antibody, and we wanted to see whether that works. So we mixed the in vitro synthesized NAD RNA and M7G RNA, and then subjected them to either no treatment, treatment with blank bees, or anti-M7G bees. And afterwards, we would convert the RNAs to biotin RNA using the spark reaction. And you can see here in the input, we could see the NAD RNA signal as well as the M7G RNA signal. And this was also the case in MOG depletion. But in M7G depletion, you can see that the flow-through did not have the M7G RNA signal anymore. So this means that the M7G antibody was effective. Next, we wanted to evaluate whether Arabidopsis mRNAs can be treated similarly to remove M7G RNAs. So here, what we did was to either do MOG depletion or M7G depletion. And then afterwards, we take the flow-through or bees, digest the RNA to nucleotides, and then we detect M7G PPPA as a readout for M7G RNA and NAD as a readout for NAD RNA. And you can see here in the M7G depletion, in the flow-through, we could get rid of about 80% of the M7G RNA, and the amount of NAD RNA was not affected. So this was actually quite nice because the M7G RNA is not as reactive as NAD RNA, And the 80% reduction should be more than enough for us to remove any interference from M7G RNAs. So then we combined the new reaction, the SPARC reaction, and the M7G depletion to re-profile NAD RNAs. So what I'm showing you here on the x-axis is RNA-seq abundance, so representing the abundance of RNAs. And then on the y-axis, I'm showing you the NAD-seq relative to mRNA-seq. And you can see these, the green dots or the orange dots here represent the statistically significant NAD RNAs. So you can see that the SPARC NAD reaction actually gave us more NAD RNAs as compared to the CUAC reaction. And the NAD RNA signal intensity is also higher. More importantly, we do not have this 5' skew anymore. So this 5' skew is caused by the CUAC reaction. Now the SPARC reaction, we have a more even distribution for the reads along the gene bodies. And you can see from the browser views here, so the CUAC reaction has this 5' skew, but the SPARC reaction gives rise to very similar read coverage as compared to regular RNA-seq. So using this improved pipeline, we were able to identify NAD RNA genes and as well as GO terms associated with these genes. Some of these GO terms represent response to environmental stimuli. So we think that perhaps one biological function of NAD RNAs is in environmental responses. And I want to mention here that from the previous NAD capture-seq, we also got a lot of response genes, but we also got a GO term photosynthesis. We believe that this is probably not true now. Photosynthetic genes are highly, highly expressed, and the original NAD capture-seq did not incorporate this M7G RNA depletion step. So we think that this GO term is probably because of the M7G RNAs also being converted to biotin RNAs. And my collaborator also used the SPARC reaction to upgrade their NAD TAC-seq to NAD TAC-seq2. And they applied NAD TAC-seq2 to bacterial RNA, which does not have M7G RNAs. So they didn't need to do any M7G RNA depletion. So one conclusion here is that if you compare the stationary phase and exponential phase, you can see that the levels of NAD RNAs for the same genes actually change, suggesting that NAD RNA capping is regulated. Another really interesting point is that for some of the top NAD capped genes, RNAs, you can see that the NAD RNAs can represent more than 50% of the total transcript pool. Remember, I told you that the 5% that was the estimate before, that was a huge underestimate, and this is why. So for some genes, there is a large proportion of NAD capped transcripts. So next, I wanted to briefly discuss what's currently known about the functions of RNA NAD capping. And this is regarding both the molecular functions and the biological functions of this RNA cap. We do not know a whole lot, so I will just very briefly talk about what we know. So in E. coli, the RNA NAD cap is thought to stabilize RNA. So shown here are RNA half-life measurements. Basically, the half-life of the NAD RNA is longer than the non-capped RNA. In a human cell line, though, the NAD cap seems to destabilize the RNA. So here on the left, what they did was to transfect into wild-type cells shown in blue or DXO knockout cells shown in red. a NAD capped RNA. So this RNA was NAD capped and transfected into the two types of cells. And I will talk about DXO later on. So they found that the half-life of the NAD RNA is very short in the control cells, but in DXO knockout cells, their half-life is longer, suggesting that the NAD RNA is destabilized in a DXO-dependent manner. They also looked at the translatability of the NAD RNA. So here they transfected a M7G luciferase RNA and a NAD-capped luciferase RNA. And they were able to find luciferase activity from the M7G RNA, but not the NAD RNA. So the conclusion is that the NAD cap does not support translation. But remember, I was telling you that in Arabidopsis, we found that NAD RNAs are polysome associated. So it seems like now there is this controversy as to whether NAD RNAs are translated. So we thought about this, and we thought that perhaps our observation of polysome association is due to the fact that we did not do any M7G RNA depletion in the NAD capture-seq. And maybe what we thought were NAD RNAs were actually M7G RNAs on the polysomes. That is a possibility. So we looked into this a bit further. And we found that, so what we did here was that we performed RNA-seq with either polysomal RNA or total RNA. And then we compared the levels of the RNA in these two fractions. And we identified these genes represented by blue dots, meaning that these genes are polysome enriched, and the genes in green, which are genes that are polysome depleted. So basically, we separated the genes into the polysome enriched and polysome depleted groups. And then we looked at their NAD RNA levels in the polysomes. So you can see that it doesn't matter whether the genes are polysome enriched or polysome depleted, we always see this NAD RNA presence in the polysomal RNA. So if this signal were introduced by M7G RNAs, we would expect to see this enrichment in the polysome enriched group a lot more than the polysome depleted group, but that's not what we saw. So we think that NAD RNAs are polysome associated, but of course we now want to revisit these results using the newly developed SPARC NAD-seq with M7G RNA depletion. So next, I wanted to touch upon the biological functions of NAD RNA by discussing the enzymes that remove the NAD cap, which sometimes referred to as denadding enzymes. DXO, which I also mentioned in the human cell experiment just now, is one of the NAD RNA decapping enzymes, and it's present in almost all eukaryotes, in plants, in fungi, and in animals. And DXO is able to remove the entire NAD RNA moiety from the RNA. The resulting RNA would be a 5' monophosphate RNA. And in Arabidopsis, there's one DXO homolog, and the mutant has very severe phenotypes. So DXO, it turns out that is not terribly specific to NAD RNA. The mouse and yeast DXO homologs can act on NAD RNA, but also some improperly capped RNAs, as well as capped RNA at a low efficiency. Arabidopsis DXO is actually better. It's more highly specific towards NAD RNA, and this was shown by Joanna Kufo's lab. And actually all DXO homologs also have 5' to 3' exonuclease activity. So presumably Arabidopsis DXO can remove the NAD and also use its exonuclease activity to remove the rest of the NAD RNA. And indeed, my collaborator E.G. Xia's lab showed that DXO can cause NAD RNA degradation in protoplasts. So here they transfected NAD RNA or M7G RNA into either wild type or DXO protoplasts. And they found that the NAD RNA is unstable in wild type protoplasts, but the stability is much better in DXO mutant protoplasts. In Arabidopsis, DXO perhaps also causes RNA degradation. So this was shown by the fact that in DXO mutants, RNAi is actually used as a backup to degrade NAD RNAs. This was observed by E.G. Xia's group, as well as Joanna Kufo and Brian Gregory's labs. In fact, if you combine DXO mutation and the DCL4 mutation, the DCL4 being a major RNAi enzyme, then you get seedling lethality, suggesting that this RNA degradation mediated by DXO is quite important. And one intriguing result is that even though DXO is thought to have this decapping activity in vitro, in vivo, when you make a catalytic mutant DXO, the construct is able to largely rescue the phenotype of the DXO mutant. So it seems like the endogenous function of DXO still needs further investigation. There is a second class of enzymes that can decap NAD RNA, and this is represented by E. coli NUD-C. So this is a Nudix domain protein, and the mechanism of action is slightly different. So NUD-C actually removes the nicotinamide moiety from NAD RNA, but the end result is still a 5' monophosphate RNA. So in Arabidopsis, there are almost 30 Nudix domain proteins, and DCP2 is actually a decapping enzyme for the canonical M7G cap. So the NUD-C ortholog is likely to be NUDT19. So not sure I have the time. I guess I will skip these enzymatic activity assays. I think
you have 3 minutes more.
Yeah, right. Yeah, so we actually were able to show that NUDT19 is indeed able to decap NAD RNA. So then what is the mutant phenotype? So this is a NUDT19 loss of function mutant, and it looks just like wild type. But others have been able to find phenotypes. So this NUDT19 mutants, they seem to be more tolerant to oxidative stresses. So maybe there are functions to NAD RNAs in responses to environments. So to summarize, I've shown you today that NAD RNAs are widespread in the Arabidopsis transcriptome. The RNAs are spliced and polyadenylated and possibly translated. We developed more sensitive and specific methods to profile these RNAs, and we identified 2 decapping enzymes. So I think in the future, we really need to resolve whether NAD RNAs are truly translated and what are the biological functions of NAD RNA capping. I would really like to thank my collaborator, E.G. Xia. We have collaborated all these years to develop different methods in NAD RNA profiling. And I'd like to thank the students and former postdocs and students who contributed to this work. Yeah, thank you for your attention, and I apologize for the technical hiccups.
Thank you so much. It was really, really, really interesting talk. I really like it. So I'm gonna ask our technical team we are gonna take a few minutes for questions, just maybe 3 more minutes, because we have the flash talk session in just next. So if somebody has a question, please raise your hands. Okay, Marina Gavilanes.
Yes, a very fast one. Is it possible that this NAD capping has to be with a kind of metabolic regulation, since the pair NADH and NAD is a very important indicator of the state of the metabolic activity of the cell?
Marina, yeah, exactly. I forgot to mention that the amount of NAD RNA that's generated is usually correlated to the amount of free NAD in the cell. So I think it could reflect the metabolic status of the cell. Yeah, very good point.
Thank you.
Okay, so the next one is Jose Luis Reyes. You can speak now, Pepe.
You're muted, Jose Luis.
Hi, Shumei. Can you hear me now? Yes.
Yes.
Very beautiful talk. It's a very thought-provoking subject, I guess, and I'm sure you're working on this. Can you share perhaps some of your thoughts on what would be the enzyme introducing the NADCAP, and what would be the translation factor that recognize that for translation?
Yeah. So the NADCAP is thought to be incorporated by RNA polymerases during transcription initiation, although there may be a post-transcriptional capping mechanism, but that's not known. In terms of how they can be translated, so we are looking into whether they are bound by translation initiation factors, and we're also collaborating with Karen Browning to use wheat germ extract to look at their translatability. So these experiments are ongoing, and they're not conclusive yet.
All right, thank you.
Yeah.
Okay, so the next question is from Patricia Leon.
Hi, very nice talk. And I have a question. I would like to hear your thoughts. Why the chloroplast RNAs are not?
Yeah, I don't know, but we looked into this a lot. It's just from this experiment, from either the NAD capture-seq or the SPARC NAD-seq, we do not see chloroplast RNAs being NAD-capped. And I think perhaps, in chloroplasts, the 5' ends of RNAs are not really defined by transcription initiation, but rather they are post-transcriptionally processed. Could that be a reason? You probably are more knowledgeable in this than I am. Yeah.
It's really interesting, and we may talk later, but it's very interesting.
Yeah. Thank you.
So the next one is Svetanka Sesi. Go ahead.
Hi, can you hear me?
Yes.
Thank you, Shumei, for so interesting and great talk, and I enjoyed it a lot. I was a little bit like asking the same like Jose Luis Reyes, but my question is whether the proportion of M7G capped and NAD capped RNA can be affected in some way post-transcriptionally. That means whether the decapping enzyme for the M7G cap could act on an mRNA and then exchange for NAD if the condition.
I see. Yeah, that we don't know. Yeah, whether you could decap an M7G RNA and then add a NAD cap, that we don't know. Yeah, but you can definitely remove the NAD cap. So I guess the ratio could be modulated by NAD RNA decapping.
Okay. And can I ask another question? Sorry.
This quick question.
The polysome bound NAD capped RNAs. So whether do you think that they could be like maybe changing the translability of RNA? That means they could be recruited to polysomes but not efficiently translated, because for M7G capped RNAs you need this circularization through the EIF4F, and this makes translation more efficient. And whether NAD capped RNAs are less efficiently translated, so you can just change the proportion of translability.
That's possible. Yeah, it's possible. But the NAD RNAs also are polyadenylated. So you do have the 3' end being bound by poly A binding proteins. We don't know whether you would form a circle though. Yeah.
Thank you.
Okay, so the last question for this afternoon talk, Gerardo del Toro, please.
Yes. Yeah, my question was a bit related to the last question that Ceci asked, but maybe I should switch to another one. I was wondering if how sensitive these techniques are, like SPAC techniques. How much RNA do you need in terms of how possible it is to adapt this kind of techniques when you have cells with very low content of RNA or RNA is limited?
Very good question. Yeah, so we initially used quite a bit when we were developing the technology, but since we have looked into the amount by doing a dilution series, and so far from Arabidopsis, I would say 1 microgram of poly A RNA is more than sufficient. For bacterial RNA, we're also looking into this. There, of course, you cannot isolate mRNA using oligo-DTBs. And there, we think a few microgram total RNA is sufficient.
Thank you. Very nice talk.
So thank you, Shumei.
Sure. I'm sorry for the hiccups. Yeah.
No, that's fine. It was not. Yeah. So thank you again, and see you soon, I hope.
Yeah, let me stop sharing. Yeah.
Great. Take care of yourself.
Yeah, thank you.
Bye. So we will start with the next flash session. Please stay. We will have a very interesting talks in this flash talks session. So let's start now.
Hi everyone, good afternoon. My name is Maria Isabel Cruz Lopez, and I will present my work entitled A Study of the Participation of the AIB JAM1 transcription factor in gene regulation by glucose in Arabidopsis thaliana. Sugars serve as an energy source, carbon skeletons, and signal molecules. Plants have developed the ability to send sugars and elicit the appropriate response through signaling pathway. By this, plants can control the nutrients status, sugar homeostasis, growth, and development. Diverse studies have been carried out to identify the elements that participate in sugar signaling, but the current knowledge of these elements and the molecular mechanism is still limited, such is the case of the transcription factors. In order to establish novel components in sugar signaling, we conduct a DNA protein assay using the regulatory region of the sugar-responsive gene STP1, a nuclear protein extract from plants treated with glucose. The result of this gave us the AIB JAM1 as a possible regulator of the STP1 expression. To determine the role of this transcription factor, we evaluate the phenotype of AIB JAM1 mutants in presence of different glucose concentration. It was evident that the JAM1 mutant presented an altered development in low concentration, showing a glucose oversensitive phenotype in comparison to the wild type. To further explore the role of this transcription factor in gene regulation by sugars, we conduct a global transcriptomic analysis by RNA-seq of the mutant versus the wild type. In order to determine the differentially expressed genes, we use a false discovery rate less than 0.5 and a fold change greater than 1.5. By this, we obtain a total of 1,330 genes for the wild type and 5,686 for the mutant. Also, we were able to identify the upregulated genes and the downregulated genes. Subsequently, we did the gene ontology analysis by biological process. It was evident that the upregulated genes corresponds to the GO term related to the 3-carboxylic acid cycle, responds to sucrose and cellular respiration. On the other hand, the genes that are downregulated corresponds to the process of photosynthesis, gluconeogenesis, and chloroplast organization. These results correspond to the phenotype exhibited by the mutant, Therefore, it is suggested that the transcription factor AIB-JAM1 is an important element in the sugar signaling pathways during the early days of the plant. Thank you so much for your attention.
Hello everybody, my name is Javier Raya González from Universidad Michoacana de San Nicolás de Hidalgo. I am talk about the role of mediator 16 on local and systemic responses to phosphate scarcity in Arabidopsis root. Phosphate is a critical macronutrient for biochemical and molecular functions of cells. Phosphate starvation induces several defects on plant development and modified root system configuration in plants, like inhibition of primary root and induction of lateral roots and root hairs, and activate a determinate growth program in root meristems. In this work, we describe the role of Mi16, a mediator complex subunit. Mediator complex is a macromolecular complex which interacts with transcription factors and RNA fold 2 to control gene expression. Phosphate limitation inhibits in primary roots and induces lateral roots in wild-type plants, but in Mi16 mutant, the mutants weren't resistant to these effects. These results you can see in these pictures. These results correlate with the gene expression of meristematic activity gene markers like cyclin B1 and WOX5 in wild-type and Mi16 plants, indicating that Mi16 plays a key role in phosphate starvation for root system configuration. It has been shown that the STOP1 transcription factor and its target ALMT1 plays a key role in phosphate starvation. To describe the relationship between Mi16 and STOP1 model, we analyze the expression of STOP1 and ALMT1 in root meristem of plants, and we found that phosphate starvation induced the expression of LMT1 in wild type but not in Mi16 mutant. However, the expression of STOP1 did not affect. This suggesting that malate excretion could be regulated by Mi16. To this, we analyzed the effect of malate application in the media, and interestingly, we found that malate application restored the phenotype of Mi16 like STOP1 and LMT1 under phosphate starvation on primary root growth. These effects we can see in these pictures. Finally, we found that Mi16 physically interacts with the STOP1. As conclusion of this work, these results unravel the function of critical transcription component Mi16 via STOP1 LMT module in the root adaptive responses to phosphate starvation. For more details of this work, you can find the article recently published in New Phytologist. Thank you very much.
Hello everyone. It's my pleasure to attend the National Congress again and to show you an overview of the project that I have been working on as a postdoc in the group of Milton scientists at the Max Planck Institute. We are interested in elucidating the genetic mechanisms driving the formation of simple leaves like in Arabidopsis and complex leaves like in Cardamine. The gene RCO was identified in a mutant screening in Cardamine as a necessary gene for the production of complex leaves. Conversely, when this gene is introduced in Arabidopsis, it's sufficient to increase leaf complexity. RCO arose from a tandem duplication of the gene LMI1 at the early divergence of the Brassicaceae family, but has been lost in some species like in Arabidopsis thaliana. In spite of being duplicated genes, they have acquired a very divergent expression pattern. LMI1 is expressed only at the leaf borders and at the stipules, whereas RCO is expressed only at the base of the leaflets. We want to know how they have acquired such a different expression. This is a hypothetic evolutionary scenario where after duplication, RCO was subject to restrictive regulation from the distal domain and permissive regulation from the proximal domain. The Brassicaceae family has been phylogenetically grouped in 5 evolutionary lineages. I found the LMI1 and RCO genes from 18 different species belonging to all the lineages and analyzed their gene sequences. Previously, the enhancer regions of both LMI1 and RCO genes were identified. They are the most conserved sequences between orthologs, but very divergent from their paralogs. These 500 nucleotide sequences are sufficient to drive the proper expression of their genes.
I generated marker lines in Arabidopsis for the expression of LMI1 and RCO enhancers, and for RCO genomic from cardamine. And I fused each of the enhancers to cardamine RCO genomic to test their influence on leaf shape. RCO increases leaf complexity only when it's expressed in the RCO domain. I am analyzing the same kind of lines using the genes from Euclidium syriacum and Arabis alpina, because these are early divergent species which already have both LMI1 and RCO genes, but still produce simple leaves. Finally, we want to know how conserved are the LMI1 and RCO function and their regulatory networks. We already know that introducing cardamine RCO genomic into Arabis alpina, which belongs to a different lineage, also increases leaf complexity. So thank you very much and see you soon.
Hello everyone. Today I will talk about how the chloroplast chromatin is organized. We all know that chloroplasts have an endosymbiotic origin and have co-evolved with the host cell to become a non-autonomous organelle, losing a lot of genes in the process and only keeping those required for photosynthesis. Up to date, the chloroplast nucleoid three-dimensional organization remains a mystery, but so far we know that this circular genome is not organized by nucleosomes or histone-like proteins. There are no known bacterial nucleate-associated proteins either. And it is anchored to the inner membrane, where most compact areas of the DNA appear to be, as you can see in the micrograph. We also know transcription is a very well-related process with two polymerases in charge, NEP, or the nuclear encoded polymerase, and PEP, or the plastid encoded polymerase, which transcribes mainly genes related to photosynthesis in mature chloroplasts. These features make the nucleoid chromatin a unique model to study. We first proved that protein binding is a contributing factor of DNA accessibility in the nucleus, because when we cross-linked DNA together with the associated proteins, we can see DNA shifting in MNase protection assays, as you can see in the yellow on the left. With this evidence, we analyzed the protein footprints at the genome-wide scale, and you can see the pattern on the first track of the genome browser at the right, just below the gene annotation. Interestingly, most of the highly transcribed genes in mature chloroplasts have the strongest signal of protein binding. For this reason, we performed ChIP-seq experiments with the plastid encoded polymerase and confirmed that most of the protein footprints come from this transcriptional complex, suggesting it is a determining factor of the nucleate organization. Lastly, we analyzed the other main feature of the nucleate, its association to the membrane. By splitting membrane and membrane-free DNA fractions, we found that most of the membrane-associated DNA resembles the plastid polymerase binding pattern. With all this information, we believe that transcription is a major mechanism that contributes to the nucleate organization in the chloroplast, and that transcription by PEP happens preferentially in compact areas close to the membrane, with very little activity on membrane-free fractions, as you can see in our proposed model. Thank you for your attention.
Hi, I'm Pamela Jimenez, and I'll be presenting the project titled Role of Flotillin in Root Hair Growth during the interaction Facialis vulgaris rhizobia. Flotillins are used as markers for cholesterol-rich membrane microdomains and are associated to membrane through the SPFH domain. Present in the N-terminal region, they define a clutter-independent pathway required for endocytosis, and by interacting with effectors that can bind actin, flotillins mediate membrane-shaping events, including membrane binding. In plants like Arabidopsis thaliana, as we can see here in the image, flotillin was found in the plasma membrane and detergent-resistant membrane domains. In legumes like Medicado truncatula, flotillin 2 and flotillin 4 are present also in the membrane, and both are required for early symbiotic events. In common bean genome, we identify one flotillin gene that encodes a putative protein of 474 amino acids with the SPFH flotillin domain in the N-terminal region. Flotillin 1 is related to Medicado truncatula flotillin 4 and flotillin 1 from Arabidopsis thaliana. According to the Common Bean Gene Expression Atlas, flotillin 1 expresses especially in early nodulation stages. We conducted transcriptional profiling in roots and find out that flotillin expresses in root hairs. We determined the subcellular localization of flotillin-1 in common bean during root hair growth and epidermal cell roots. Flotillin-1 accumulates at discrete spots in the cytoplasm of the mature root hair, and these spots move along the cytoplasm. In the epidermal cells, flotillin-1 accumulates also in small vesicles that move along the cytoplasm. These observations suggest that flotillin have a role in vesicular trafficking and root hair polar growth. Flotillin and the NADPH oxidase could be associated to membrane microdomains because flotillin-1 in common vein localization is similar to previous unpublished RBOH localization in young growing root hair of common bean. We also observed the localization of flotillin-1 during the early stages of nodulation and find out that flotillin-1 in common bean localized in the infection threads during rhizobia infection, in a similar way that was reported in Medicago truncatula flotillin-4 localization during bacterial infection. Using flotillin promoter and Gus reporter gene during rhizobia interactions, we found that flotillin-1 in common bean is expressed during the infection thread formation and throughout the nodule formation. These results suggest that flotillin-1 in common bean is important in the rhizobia facialis vulgaris interaction. Our features directions are to generate RNA interference-mediated knockout flotillin-1 of common bean transcript to evaluate the phenotype during mutualistic interactions.
Hello, my name is Eduardo. I am going to talk about the results that I have obtained with my research team. The title of this work is In Vitro Assessment of DCL RNase has biofungicides against pathogens of pineapple, orange, and cacao, developed at the Universidad del Papaloapan, Oaxaca, Mexico. We have identified 5 pathosystems of interest on this study, which are Cytosinensis Penicillium digitatum, Thebroma cacao Phytophthora tropicalis, Thebroma cacao lacipia thebrome, Ananacomosus Phytophthora nicotinae, and Ananacomosus fusarium oxysporium. Molecular identification of pathogens was carried out by sequencing the ATS region. Then the sequences of dicer-like genes, pathogens, and plant and pathogens were researched in the databases, and a link to select region of low homology between both sequences. Those regions were used for the dosing and production of those RNAs by in vitro RNAs synthesized of long and short dose RNAs. Those RNAs were tested for fungicide activity via gene silencing. Here, I will show the case of Cytosinensis Penicillium digitatum. Those RNAs were synthesized for the silencing of the pathogens DCL1 and DCL2 genes. A spore germination test we carry out found an inhibitory effect of 78% at 0.125 microgram of long dose RNAs. In the case of small dose RNAs, a significant effect was observed in all concentration used, specifically at the concentration 1 microgram with 82% inhibition and 0.125 microgram with 86% inhibition. This indicates that those RNAs have a negative effect on spore germination. The validation of the silencing of DCL genes was performed by qPCR custom plate with used cDNA synthesized from .RNAs extracted from mycelium growth for 3 days on 1 milliliter of PDA growth added with 1 microgram of small dose RNAs. The result shows a slightly but significant decrease for each test of both the DCL1 and DCL2 genes. This decrease in the expression per statement can also be observed with respect to the control. With this result, it can be determined that those RNAs have a strong effect on spore germination than a mycelial growth. The fungicide effects of those RNAs on other pathogens in this work remains to be explored. Thank you very much.
Hello, my name is Tania Gomez Diaz, and I am a second-year PhD student at the Universidad Veracruzana. The work that I'm going to present to you today is how the ascorbic acid plays a prominent role in the formation of cuticle and in the response to abiotic and biotic stress in Mercantia polymorpha. It is part of an article in which we are working. Ascorbic acid, or ascorbate, is an important antioxidant involved in several fundamental functions for plants regulate cell metabolism, growth, development, and maintains the homeostasis of reactive oxygen species to protect plants from environmental stress. In plants, 4 alternative pathways have been proposed for ascorbate biosynthesis. The D-mannose L-galactose pathway is highly conserved across the embryophytes, and it involves the activity of the enzyme L-galactone 1,4-lactone dehydrogenase, or GLDH, to catalyze the final reaction leading to ascorbate. Here, we characterize the role of ascorbate during the defense responses of Mercantia polymorpha to stress. We took a reverse genetic approach using the CRISPR-Cas9 gene edition system to isolate mutants in the MPGLDH gene. MPGLDH1 loss of function mutants have defects in the cuticle formation with increased epidermal permeability and developmental effects, including organ fusion, dorsal outgrowths, and accumulate significantly less ascorbate than the wild-type tally. We establish a protocol to perform botrytis cinerea infection assays, and we test the cell stress tolerance employing the MPGLDH1 mutants, and we found that these mutants are more sensitive to cell stress, and its permeable cuticle leads to a deferred infection with the necrotrophic fungus Botrytis cinerea. Taken together, our results show that in Mercantia polymorpha, ascorbate is required for normal development. It is implicated in the formation of cuticle, the control of lateral organs, and it is involved in the responses to both biotic and abiotic stress. I can only thank all the institutions and people that make this project possible.
Hi, my name is Jesus Salvador Lopez Bucio, and I'm going to talk about primary response to phosphate deprivation driven by auxins and the transcription factor sombrero. Phosphorus is assimilated by plants as inorganic phosphate, and this is one of the most limiting nutrients for plant development. The plants under phosphate deprivation increases in root branching and root age formation in order to improve phosphate uptake. But also, the primary roots of these plants stops to grow because root apical meristem is exhausted. We found that before root apical meristem exhaustion, the primary root tip increased in thickness depending on overproliferation in the cells of the root cap, including columella layers and lateral root cap layers. This cellular overproliferation correlates with diminishing auxin levels. Moreover, when auxin-related mutants are exposed to low phosphate levels, the primary root is enhanced in thickness depending on also cellular overproliferation in the distal meristem, in the root tip. It's well known that the transcription factor sombrero modulates cellular proliferation in the root cap. The mutants of sombrero have increased cellular layers in the root cap. Interestingly, when sombrero mutants are exposed to low phosphate levels compared with wild-type plants, the primary root is not inhibited by phosphate deprivation, and the root cap doesn't change in number of cells, which is the relevance of increased root thickness. It's well known that the expression of phosphate transporter is enhanced in the root tip when plants are under low phosphate levels. Interestingly, the expression of the phosphate transporter ATP1 is enhanced in both conditions in normal and also under phosphate deficiency plants. With this data, we can construct a general model in which plants experiencing phosphate deficiency have diminution of auxin signaling, which leads to cell overproliferation and thus phosphate uptake, which helps primary root growth. Thank you.
Hi everyone, my name is Kiran Jimenez. I will speak to you about my project, the plant beneficial Rizobacterium Achromobacter SP501 influencer root development through auxin signaling and redistribution. In the middle of Chihuahuan Desert in Mexico, far from civilization, lies a natural sinkhole called Poza Salada. It is a very extreme environment and scarce in nutrients and water but high in salt. We collect them from a mosquito tree bacterial isolates and one of the bacterial strains was of particular interest. The bacterium was propagated of Luria bertani medium and both gram staining and electronic microscopy analysis proved it as a gram-negative flagellate bacterium. 16 xrRNA sequences identified as Achromobacter sp501. Cultivation experiments using plant model Arabidopsis thaliana showing an increased lateral root number and a promoted primary root in seedlings inoculated with Achromobacter sp501. Also, the primary root was deviated from the gravity vector forming coils. Based on the agravitropic root phenotype and given the intimate involvement of auxin in root architecture and gravitropins, we analyze the effect of the bacterium in primary roots and its relationship with auxin and Arabidopsis endil that express GR5, GFP, and auxin-inducible gene marker. Confocal imaging of DR5GF peaks showing an increase in auxin responsiveness in lateral root primordia and enhancing auxin responsiveness and the very root type while match fluorescence to the concave side of the coil. To understand what is behind this different distribution, we study the abundance on several auxin transporter spins using transgenic sampling expressing GFP level constructs. We found that these pins proteins show an altered abundance in roots cultivated with the bacteria. Auxin transport relay mutants are 1 to 1 and AUX 1 to 7 showing an altered primary root growth and colon response, but only a minor effect on the increase of lateral root. Based on these results, we propose the next model. Achromobacter promotes pin3 expression in columella cells, while representing pin1 and pin2 and maristem as cell elongation region. Thank you so much.
In this flash talk, I will talk about Arabidopsis glycine-rich domain protein that interacts with proteins associated with RNA processing and translation. We previously described that this gene is expressed in plant development and in response to stress. Also, the overexpression led plants with accelerated growth, flower earlier, and improved salt tolerance. The opposite phenotype was observed in mutants. Our protein of interest is encoded by this gene, ATGRDP2, and we know it is constituted by 3 domains. In N-terminal, a domain with unknown function, in central region, an RNA recognition motif, and in C-terminal, a short glycine-rich domain. However, its function is unknown. So in this research, we were interested in elucidate its possible mechanism of action. For that, we found that our protein localizes in both cytosol and nucleus. So we were interested in know which domain was responsible for nuclear localization. For that, we generated truncated versions of our protein that include N-terminal and central region of the protein and found that both localize in nucleus. So when we analyze the amino acid sequence, we found that our protein has at least 5 possible nuclear sequences that are interspread between both domains. Then we identified its potential interactors by GIS2 hybrid split ubiquitin and found that 3 of them were RNA binding proteins, such as polyadenylate binding protein 3, an elongation factor, and a ribosomal protein CL15. Then we corroborate this interaction in planta and found that these interactions are carried out in nucleus, cytosol, and chloroplast. with the truncated versions, we could elucidate which domains were responsible for this interaction. For example, in the case of PABN3, both domains DUF1399 and RRM are very important. In the case of elongation factor, only domain with unknown function was key for this interaction. And finally, with chloroplastic CL15, RRM is necessary. So far, we propose, or we have a hypothesis that our protein could be a carrier for RNAs, and they will be transported to chloroplast and cytosol. Thank you.
Hi, my name is Melissa, and today I will tell you about how the function of a transcription factor family could be conserved since the most recent common ancestor of embryophytes. Comparative genomics has revealed that members of some lineages of land plants like mosses and ferns share a set of highly conserved transcription factors with derived flowering plants. It has been predicted that diversification, co-option, and reassembly of gene regulatory networks implicated in development are related to morphological innovations that led to more complex land plant bodies. Integumental-like genes are considered part of the developmental toolkit of land plants. These transcription factors, 8 in Arabidopsis thaliana, are induced by auxin and are mainly expressed in dividing tissues where they play roles such as the maintenance of stem cell niches, the correct development of the embryo, and formation of lateral organs. But what could these genes be doing in relatively simple plants like liverworts? The single integumenta ortholog MPANT encoded in the genome of the liverwort Marcantia polymorpha is very similar to that of Arabidopsis integumental-like gene family, and the sequence and length of the DNA binding region is also conserved. By searching for the conserved DNA binding motif within the genome of Marcantia, we have found that putative targets of MPANT include the same MPANT, some auxin biosynthesis genes, and some orthologs of Arabidopsis meristem development genes, such as SPL, MATS2, and LIFI. We have also made some experiments with cool transformant lines. I have found that the MPANT loss of function line is smaller and has fewer rhizoid cells in the early GMA stage, the total opposite to the overexpression line, which has a bigger number of this type of cells. We also measured the number of meristematic apical cells in Marcantia thalli from these lines and discovered that the overexpression line harbors more apical cells than the loss of function and the wild-type lines. This could mean that MPANT is regulating the number of cells formed by divisions of the central apical cell. In this picture, you can see how the thallus area between transformant Marcantia lines and the wild type is very contrasting, and we determined these differences were evident from the 7th day stage onwards. This reveals that the abundance of MPANT affects not only the number of cells, but also the overall size of the thallus. But one of my favorite experiments is this one, where we discovered that MPANT partially complemented the wild-type phenotype of the plethora 1, plethora 2 Arabidopsis mutant, which means that not only the fundamental structure of the TF is conserved between plant lineages, but that the meristem maintenance function of integumental-like genes and its interaction with hormones could be conserved since the last common ancestor of land plants. All these results and more that I hope to publish soon will help us shed light into the role of the sole integumental-like gene in the liverwort Marcantia polymorpha. Thank you.
Hi guys, my name is Jesus Leone and I'm here to talk to you about my flash talk, the evolution of the pituitary related LC-mediated interaction landscape across Brilliant plantain. As you know, the cell cycle is one of the most widely considered pathways there is. A key checkpoint is the G1 to S transition right here, regulated by E2F and the retinoblastoma protein. And besides this role, both PRB and this plant homolog retinoblastoma related have been implicated in many aspects of cell growth and differentiation, which are believed to be the result of retinoblastoma's ability to interact with many protein partners, usually with proteins containing the LC motif. In Arabidopsis, RBR interacts with many transcription factors such as SCARCO and PAMA to regulate asymmetric cell division as well as cell fate commitment in the root apical meristem and stomatal lineage. And interestingly, when these interactions are abolished, the result is aberrant and additional cell divisions in both the gland tissue and stomata right here. However, I do need to stress that these studies have been made solely in Arabidopsis, so what about the rest? So to explore this, what about the rest? We use a computational evolutivo approach to explore in silico what type of RBR interactors are conserved or have changed throughout green plant evolution. And what we found is that RBR's association with chromatin modifiers from different types seems to be conserved in all the analyzed species. as well as potential interactions with DNA repair and replication effectors, and as suspected, interactions with proteins involving cell cycle regulation, such as the dicyclines, are well conserved in all the species analyzed here. And moreover, we decided to explore potentially interacting RBR transcription-associated proteins, and we found that interactions, for example, with grass and DHLH families seem to be conserved in land plants. In addition, potential interactions with the SWSNF and PHC domain families, which are chromatin remodelers and readers, seem to be universally conserved in all the plantae. So we can basically suggest that interactions with mitotic cell cycle regulators, chromatin modifiers, and DNA replication and repair effectors seem to be conserved. However, we did found low conservation in several of the detected LC motif-containing families regulators, suggesting that if indeed these interactions actually occur in vivo, RBR was likely independently recruited to form part of transcriptional regulatory networks, most likely in a species-specific fashion. I'm sorry. So this is a lot of data to discuss in a flash talk, so I'm gonna leave you guys with these take-home messages right here, which I'm unable to read because of matters of time. And well, for my PhD, I'm working right now in disrupting and experimenting with specific RBR interactions, which I believe holds great promise in helping unravel the complexities of this multifunctional protein. So thank you so much for your attention, and wait for paper, as they say, and enjoy the rest of the symposium.
Hi everyone, this talk is titled Understanding the Tripartite Interaction Between the Plant, a Fungal Pathogen, and a Beneficial Bacterial Endosymbiont. Fusarium verticilioides can infect maize root. In this interaction, plant chitinase plays a key role, since they must degrade the fungal cell wall in order to release molecules that act as pumps and stimulate the plant defense system. However, the fungus can avoid this because of the production of proteins that can modify the plant chitinase. In this image, we show that Fusarium can infect the vascular system of roots from maize plants. Bacillus cereus B25 is an effective biological control agent against Fusarium, and we have proposed that in the tripartite interaction, the bacterial chitinase degrade the fungal cell wall and stimulate the plant defense system. And in this particular case, the fungus cannot avoid this because the proteins that it produces to modify the plant chitinase cannot modify the bacterial chitinase. In this image, we show that B25 can colonize the vascular system of roots from maize plant as well as Fusarium. To understand the biocontrol mechanism mediated by B25, we first analyze the B25-FB interaction. We evaluate all the genes from B25 that are related to fungal antagonism, and we found that they are induced by FB mainly between 6 and 24 hours during its direct interaction. In this image, we show that B25 has an inhibitory growth effect over the mycelium of FB. We perform a tripartite interaction, and we took root samples from different treatments, and we perform an RNA sequence. When we compared the different treatments against the control, we found different number of differential expressed genes, and the interesting part here is that we found chitinase activity as one of the top overrepresented categories in the tripartite interaction, as well as other categories that were not found in the other comparison.
Hello, my name is Hector Ruiz, and I would like to present my master thesis project in silico analysis and gene expression of transcription factors related to cuticle biosynthesis during the ripening of soursop fruits, Annona muricata. The soursop is a crop with a short post-harvest life and with a great economic importance to Mexico, reaching in 2019 a production value of $12 million. It has been reported that the composition and structure of the cuticle have an influence in the quality and post-harvest life in fruits. For instance, Saladi and collaborators in 2007 reported a tomato variant with differences in its cuticle properties that allows the fruit to have optimal condition of consumption much longer than other tomato variants. Cuticle biosynthesis in plants is a highly regulated process at different levels, such as transcriptional level. At this level, there are transcription factors involved in regulatory networks acting in several plant processes, such as cuticle biosynthesis. The elucidation of these regulatory networks helps us to understand complicated processes in living organisms like cuticle biosynthesis in plants. Despite the importance of the soursop for the Mexican economy, this type of study is scarce for this crop. Therefore, we ask the following question. Which transcription factors are involved in the regulation of cuticle biosynthesis during the fruit ripening? To address this question, we compared a de novo assembled transcriptome and predicted proteins in the genome of soursop fruit with transcription factor databases PlantTFDB and ITAC. Our results show a total of 1,121 predicted proteins coding for transcription factors from 75 different families. A comparison between proteins and transcripts showed a total of 961 proteins coding for transcription factors, represented by 4,174 transcripts with an average of 4.4 transcripts per protein, suggesting that we identified putative isoforms of these transcription factors. The transcription factor families most represented were these, and according to previous studies, transcription factors members of these families are involved in the regulation of cuticle biosynthesis in fruits. Therefore, it is possible that we are finding in soursop fruits transcription factors involved in cuticle biosynthesis. Thank you so much.
Hi, my name is Luis Reyes Aguilar, and I'm going to show you my master degree thesis project, which is titled Contribution of MicroRNAs MIR482, MIR2118, and MIR1510 to the regulation of early stages of nodulation between Bacillus vulgaris and Rhizobium tropici. Legumes establish a symbiotic relationship with a specific soil bacteria called Rhizobia, resulting in the formation of specialized root organelles called nodules, where the Rhizobia synthesize nitrogen compounds usable by the plant. The success of this interaction depends on the recognition of the specific partner and the exclusion of possible pathogenic bacteria. Communication between these symbionts is necessary for the molecular and physiological changes that allow the initial stages of nodulation. Recent studies have demonstrated that recognition of molecular patterns that belong to Rhizobia and other bacteria present in the rhizosphere is in many cases necessary for successful symbiotic nitrogen fixation. As part of its immune system, plants have a diverse family of proteins and function as cytoplasmic receptors to recognize strain-specific effectors. These proteins contain two main domains, a nucleotide binding site and a leucine-rich repeat domains, commonly known as MBS LRRs proteins. The regulation by microRNAs, a group of small RNAs involved in a post-transcriptional gene regulation, has gained relevance. Three families of evolutionarily related microRNAs have emerged as important regulators of MBS LRRs: MIR482, MIR2118, and MIR1510. Nevertheless, the participation during the establishment of symbiosis has not been studied. Thus, we are characterizing the accumulation of these microRNAs and confirming selected messenger RNA targets during the early stages of nodulation between Bacillus vulgaris and Rhizobium tropici. In results, we found that accumulation of the three microRNAs tend to decrease over the first hours of interaction, as you can see in the graphs. Similarly, at later time points, we also observe a decrease in accumulation levels for the three microRNAs, just this time at 48 hours post-inoculation. In addition, we use the Haiti-Roots methodology to reduce the levels of MIR482 and MIR2118. When we silence MIR482, we obtain the reduction of its levels. However,
MIR-2118 increases its levels. And likewise, this event occurs when we reduce the accumulation of MIR-2118. This time, MIR-482 is the one who increases its levels. The reason behind it is not entirely clear, but we are still working on understanding these effects and evaluating new approaches. Thanks to my thesis advisory committee, to Instituto de Biotecnología TUNAM, and to the GAPA for the money. This is my contact info, my mail, and my Twitter if you want to chat with me. Thanks.
Hello, my name is Dulce Flores. I'm PhD student at the Universidad Veracruzana. My project consists in the characterization of cell stress responses in Mercantia polymorpha. So, soil salinity is one of the major environmental stress faced by the plants. Plants grown in saline soil suffer from sodium-induced ionic stress. Salt stress in plants causes metabolic disorders, inhibit photosynthesis, affect ionic balance and cell permeability, and eventually causes death. Plants have developed different response mechanisms to counteract salt stress. The ability of plants to respond to increase in salt concentrations depends on change in gene expression. In order to understand the physiological and molecular change during salt stress response in Mercantia polymorpha, a rhyophyte that colonized landscape 470 million years ago, we characterize the physiological response to low concentration, 50 millimolar, moderate 100 millimolar, and high 150 millimolar sodium chloride. We find that the low concentration has positive effect in growth and that moderate and high concentrations affect the growth of Mercantia polymorpha. The plastogram measurement in Mercantia polymorpha indicates a developmental delay from 100 and 150 millimolar sodium chloride. At the concentration of 50 millimolar sodium chloride, the thallus of Mercantia polymorpha grow more than at control. The plants acquire higher biomass and gene production accelerates. We also observe ROS production under saline stress conditions, especially at the concentration of 150 millimolar sodium chloride. There is an explosion of ROS in 2 hours as opposed to 24 hours where the production decreased. Then we profile global gene expression and identify genes differentially expressed in Mercantia polymorpha when exposed to 100 millimolar sodium chloride for 2 and 24 hours. By means of a transcriptome analysis, we identify differentially expressed genes. We were able to identify gene pathways involved in carbohydrates, amino acid, and lipid synthesis, auxins, and ROS production, which helped us use to clarify whether the response mechanisms to saline stress of Mercantia polymorpha are similar to those of vascular plants. Finally, we conclude that saline stress in Mercantia polymorpha is redirecting its energy to maintain cellular integrity and basal functions due to osmotic stress, and that high concentrations of sodium chloride impact resource assignment, causing a decrease in area, biomass, and affecting asexual reproduction. Finally, I appreciate the support for this project to all institutions and researchers. Thank you. Hello, my name is Dulce Flores. I'm PhD student at the Universidad Veracruzana. My project consists in the characterization of salt stress responses in Mercantia polymorpha. So, soil salinity is one of the major environmental stress faced by the plants. Plants grown in saline soil suffer from sodium-induced ionic stress. Salt stress in plants causes metabolic disorders, inhibit photosynthesis, affect ionic balance and cell permeability, and eventually causes death. Plants have developed different response mechanisms to counteract salt stress. The ability of plants to respond to increase in salt concentrations depends on change in gene expression. In order to understand the physiological and molecular change during salt stress response in Mercantia polymorpha, a rhyophyte that colonized landscape 470 million years ago, we characterize the physiological response to low concentration, 50 millimolar, moderate 100 millimolar, and high 150 millimolar sodium chloride. We find that the low concentration has positive effect in growth and that moderate and high concentrations affect the growth of Mercantia polymorpha. The plastogram measurement in Mercantia polymorpha indicates a developmental delay from 100 and 150 millimolar sodium chloride. At the concentration of 50 millimolar sodium chloride, the thallus of Mercantia polymorpha grow more than at control. The plants acquire higher biomass and gene production accelerates. We also observe ROS production under saline stress conditions, especially at the concentration of 150 millimolar sodium chloride. There is an explosion of ROS in 2 hours as opposed to 24 hours where the production decreased. Then we profile global gene expression and identify genes differentially expressed in Mercantia polymorpha when exposed to 100 millimolar sodium chloride for 2 and 24 hours. By means of a transcriptome analysis, we identify differentially expressed genes. We were able to identify gene pathways involved in carbohydrates, amino acid, and lipid synthesis, auxins, and ROS production, which helped us to clarify whether the response mechanisms to saline stress of Mercantia polymorpha are similar to those of vascular plants. Finally, we conclude that saline stress in Mercantia polymorpha is redirecting its energy to maintain cellular integrity and basal functions due to osmotic stress, and that high concentrations of sodium chloride impact resource assignment, causing a decrease in area, biomass, and affecting asexual reproduction. Finally, I appreciate the support for this project to all institutions and researchers. Thank you.
Hello everyone, I'm Cintia Soberanes. Today I'm talking about host-induced gene silences of Phytophthora capsicii GIP1 glucanase inhibitor protein 1 decreases infection in Nicotiana ventamiana. Homicides from the genus Phytophthora, fungus-like plant pathogens that are devastating for agriculture and natural ecosystems. P. capsicii, which infects a large number of agriculturally important vegetables like pepper, tomato, cucurbits, and eggplant, causes huge economic losses. This homicide is able to secrete a glucanase inhibitor protein, GIP1, a protein that inhibits endobeta 1,3 glucanase produced by the plant host. The host-mediated gene silences Higgs is a technique for genetic silences in which we introduce double-stranded RNA or RPin into the host and mediate the silences genetic machinery. In this study, we developed a strategy of Higgs to silence the gene GIP1. We identified the protein homologous to ones codified by GIP1 gene and consists of 777 nucleotides and codes for a hypothetical protein of 259 amino acids. And for other hand, we performed the blast assay between protein GIP of P. capsicii and other species. We performed the structure of the protein GIP1 of P. capsicii used as a template for the modeling GIP1 protein of P. infestans. Also, we characterized the pathosystem and we observed that the Nicotiana tabacum plants were resistant in comparison with Nicotiana ventamiana plants was died in few days. We observed that transient expression P. capsicii genes showed reduced symptoms in Nicotiana ventamiana in comparison with the empty vector. In order to evaluate the GIP1 silences in plants infected with P. capsicii, we analyzed the expression of this gene by qRT-PCR, and the analysis confirmed that GIP transitory expression confers resistance to P. capsicii. In conclusions, we confirm that Higgs method could be an effective strategy to confer resistance against the homicide P. capsicii. Thank you very much.
Recording in progress.
Hi, the present project is Antibesogenic effect of Calisthemus citrinus in rats fed a high-calorie diet rich in fat and sucrose. The excessive consumption of diets with a high caloric intake and the decrease in physical activity, among other factors, together rapidly develop overweight and obesity in the human population worldwide. During these conditions, the excessive accumulation of adipose tissue is favored, an increase in the generation of reactive oxygen species and decrease of the antioxidant system, producing oxidative stress. Obesity is associated with numerous diseases: hyperlipidemia, diabetes mellitus, hypertension, inflammation, gastrointestinal disorders, and cardiovascular diseases. Calicium citrinus is a plant that produces compounds with a high pharmacological potential and antioxidant activity. In the present study, the effect anti-obesogenic of Calicium citrinus in rats fed with a high fat sucrose was determined. 15 family Wistar albino rats were used, which were randomly divided into 3 experimental groups in equal 5. Group 1, normal diet; Group 2, high-fat sucrose diet; and Group 3, high-fat sucrose diet plus daily oral Calicium citrinus extract at dose of 250 milligrams per kilogram for 50 weeks. The morphometric and biochemical parameters for the high-fat sucrose diet only group were significantly increased compared to the control group, while Group 3 showed significant suppression of weight gain. The morphometric parameters were strongly correlated with the body weight. Calicium citrinus extract decreased the parameters compared to the high-fat sucrose diet group, acting similarly to the control group. These results suggest beneficial anti-obesogenic activity of Calicium citrinus leaf extract. Thanks.
Hello everyone, it's my pleasure to attend the National Congress again and to show you an overview of the project that I have been working on as a postdoc in the group of Miltos scientists at the Max Planck Institute. We are interested in elucidating the genetic mechanisms driving the formation of simple leaves like in Arabidopsis and complex leaves like in Cardamine. The gene RCO was identified in a mutant screening in Cardamine as a necessary gene for the production of complex leaves. Conversely, when this gene is introduced in Arabidopsis, it's sufficient to increase leaf complexity. RCO arose from a tandem duplication of the gene LMI1 at the early divergence of the Brassicaceae family, but it has been lost in some species like in Arabidopsis thaliana. In spite of being duplicated genes, they have acquired a very divergent expression pattern. LMI1 is expressed only at the leaf borders and at the stipules, whereas RCO is expressed only at the base of the leaflets. We want to know how they have acquired such a different expression. This is a hypothetical evolutionary scenario where after duplication, RCO was subject to restrictive regulation from the distal domain and permissive regulation from the proximal domain. The Brassicaceae family has been phylogenetically grouped in 5 evolutionary lineages. I found the LMI1 and RCO genes from 18 different species belonging to all the lineages and analyzed their gene sequences. Previously, the enhancer regions of both LMI1 and RCO genes were identified. They are the most conserved sequences between orthologs, but very divergent from their paralogs. These 500 nucleotide sequences are sufficient to drive the proper expression of their genes. I generated marker lines in Arabidopsis for the expression of LMI1 and RCO enhancers and for RCO genomic from Cardamine. And I fused each of the enhancers to Cardamine RCO genomic to test their influence on leaf shape. RCO increases leaf complexity only when it's expressed in the RCO domain. I am analyzing the same kind of lines using the genes from Euclidium syriacum and Arabis alpina, because these are early divergent species which already have both LMI1 and RCO genes, but still produce simple leaves. Finally, we want to know how conserved are the LMI1 and RCO function and their regulatory networks. We already know that introducing Cardamine RCO genomic into Arabis alpina, which belongs to a different lineage, also increases leaf complexity. So thank you very much and see you soon.
Hello everyone, this is Eddie Mendoza Galindo, and today I'm going to talk about our work called Rose generation is decreased during host-induced gene silencing in Phytophthora capsicum G1 in Nicotiana bentamiana. Reactive oxygen species, or ROS, are produced within several cell compartments. The most common of them, hydrogen peroxide, either can act as a signal molecule or induce damage. This depends mostly on its concentration. When a pathogen attacks a plant, one of the first responses is the high production of ROS in the first minutes after the pathogen is detected. Then, the cell will either die or acquire resistance. Homicides are important pathogens of the Solanaceae family, and the way they influence is by repressing glucanases. These proteins degrade glucans in the homicide cell wall and induce other defense responses. Glucanase inhibitor proteins have been studied in many Phytophthora species. It has been demonstrated ROS production and glucanase activity are induced at the same time in infected plants, and both are compromised when ROS production is inhibited. In this way, we hypothesize ROS is an important part of plant defense response against Phytophthora capsicii GIP1, using Nicotiana benthamiana as a model to silence this gene. In order to test our hypothesis, we first analyzed GIP1 sequence, resulting in a very similar sequence to Phytophthora infestans GIP1. Thus, we decided to compare it with other Phytophthora infestans elicitors in our experiments. And with the aim of knowing its physiology, we constructed an experimental design using host-induced gene silencing and implant overexpression. First, we used PHELS-K12 Higgs vector within Agrobacterium tumefaciens to transform cell discs. Then, we inoculate agroinfiltration zones with Phytophthora capsicii zoospores, and finally, we perform diaminobencidine staining to dye hydrogen peroxide in situ. Our results indicate ROS production is decreased when GIP1 is silenced. This might be due to a compromised pathogen development when glucanases are not inhibited. This phenomena was also perceived when silencing the Phytophthora infestans effector ENF1. On the other hand, overexpression of GIP1 induced high ROS generation, and we observed background ROS production caused by agroinfiltration in the empty vector. This supports our hypothesis, and ROS is coupled to immunity response of Nicotiana benthamiana against Phytophthora capsicii GIP1. In conclusion, Phytophthora capsicii GIP1 is an important pathogen effector, and if silenced, host plant can easily stop its infestation and decrease ROS generation.
Hello everybody. This is the title of my work that I will be presenting today, and I want to thank to the organizers for the invitation. This is our model. This is the MOS Viscometrium patent, which is a very powerful model system. As we all know, reactive oxygen species play a very important role in several physiological processes. Most of the ROS can be generated by activity of the NADPH oxidases. In Viscometrium patents, our model, we have identified 4 different genes encoding for these enzymes. By using CRISPR-Cas and in collaboration with Dr. Besanilla at Dartmouth College, we have isolated single, double, triple, and quadruple mutants. By phenotyping those mutants, we found that in the triple and quadruple mutants, there is a reduced growth area as compared to the wild type. If we treat those cells with chitin oligosaccharides for mimicking a pathogen response, we can see that in the triple mutants, there is an increase, while in the quadruple mutants, there is a decrease in growth area as compared to the wild type. If we look at the intracellular hydrogen peroxide distribution, we can see that there is a very nice gradient at the tip region of the growing Caulonema cells in wild type. However, in the quadruple mutants, this gradient is highly reduced as compared to the wild type. Then we did a series of experiments in which we treated those cells in the presence of chitin oligosaccharides for mimicking the pathogen response. Under these circumstances, we observed that there is a very nice increase in hydrogen peroxide distribution in the cytoplasm. But also, this kind of fluctuations that seems to expand from the base to the tip region. This is the first time that we are describing such responses in the most modern Viscometrella patents. Again, if we look at the quadruple mutants, we didn't see that response, indicating that the presence of the NADPH oxidases is required. Therefore, we can say as a conclusion that the NADPH oxidase is involved in maintaining the apical and cytoplasmic hydrogen peroxide distributions. We can also conclude that the quadruple mutants experience the strongest effects on the intracellular hydrogen peroxide distribution, but also in growth. Finally, we can say that the induced ROS response in the presence of chitin oligomers is NADPH oxidase dependent. Therefore, the most cells present a mechanism for inducing a ROS-induced elicited response, which happen to be very similar to that described in superior plants such as Arabidopsis. Thank you very much. I appreciate it.
Hello, everyone. I'm Vincent Cervantes, and I'm going to tell you a little bit about my PhD project, which is titled Analyzing the Function of shoot meristem less related to cytokinin during Janison development of Arabidopsis thaliana. Well, cytokinin biosynthesis is a process that starts with the addition of an isoprenyl molecule to an adenine in an ATP or ATP. This process is regulated by an enzyme called isopentanil transferase. After that, that adenine needs to be released from that ATP or ATP. This process is regulated by an enzyme that is coded by lonely guide genes. We have 9 genes of each enzyme in Arabidopsis. This process has been related to a transcription factor that is important for the maintenance of the shoot apical meristem, STM. When STM is overexpressed, it induces the expression of the IPT genes, especially the IPT7. Also, when it's overexpressed, regulates the production of cytokinins. We can see here how this response regulator line increases its GFP signal when STM is overexpressed. So STM is doing something important here in the cytokinin production. STM is not limited to the shoot apical meristem. It is also expressed in gynecium and is important for the development of this tissue. We can see here how the gynecium is not well developed when it's not there in this line of RNA. But what STM doing here is inducing the cytokinin production to which of the genes is regulated in STM? And are these genes regulated directly by STM? That is something that we want to know. First, we started with the study of the expression patterns of every biosynthesis genes. We found here that some of them are very well represented, and some of them are not well represented. We have a highlight in the lonely guide genes, and we found that all of them have a different expression pattern during the Janison development, and we found that also all of them are in different places and in different intensities. So we can suggest that all of them have different functions and in different places are activating the cytokinin. We also found that STM is regulating them, and I'm going to show you here this beautiful result. When STM is overexpressed by the dexamethasone addition, the lonely guide 4 GFP marker line increases its signal. We can see here very clearly how this increased. And we also studied the direct regulation of STM in this gene, and we found that STM is binding to a specific portion of this gene. You can see here that the gene is still growing, even in the presence of aurabazidine antibiotic. We also found that STM is binding to a specific portion in this chip experiment, and we also did this experiment in luciferase assay. We are really excited to show you more about these experiments and how important is this regulation for the Arabidopsis genus development. Thank you so much.
Hello everybody, my name is Eduardo and I'm going to talk about molecular response to tomato plants treated with biostimulant Natrisar L. The global population is already 8 billion people. For 2050, we will be more than 10 billion people. Feed all people with the current habits of feeding is not easy. This increase of population is a big challenge for the actual agriculture in an age-worn world with less water, less fertile soils, and with more starving people. For this reason, the agriculture has employed strategies for improved plant nutrition through the use of agrochemical-based nitrogen and phosphate. Our stimulant is an extract obtained from organic compost that it had biological activity in plant improvement. In the case of biostimulant Natrisar L, it has been documented that stimuli foliar and root grown. Here we present the transcription profile of several key genes involved in oxidative stress, ion transport, biosynthesis, and response to auxins. In the way to understand the molecular effects of biostimulant, we propose an experiment using tomato Ohio set. We grown the plants with photoperiod of 16 light and 8 hours dark, water daily with weekly application with Hoagland solution. The first application was in the germination of the seed, second immediately to transplant into flower pot. Other applications were 7 days after transplant and third application 14 days after transplant, and then we take the sample in several times. Our molecular analysis was focused to the transcriptional response of oxidative stress. We evaluated oxidative stress with the expression of ascorbate peroxidase and glutation S-transferase, ion transport with transcription of ATPase4, signaling osmotic response by the transcription of the SNRK2, and jasmonic biosynthesis, but oleooxidase synthase. We found that Nutrisor L in plant tissues was firstly observed in plant germination. We observed that biostimulant evaluated increased the percentage of germination. We also observed that biostimulant induced gain weight, and we observed in the graphic that plants was vigorous, height, and thickness. The first effect was observed immediately in root. Here we observed that the expression of oxidative stress and ion transport was stimulated. 1 and 4 hours later, auxin biosynthesis and ABA signaling pathway, respectively, was induced. In leaf, ion transport and ABA response to osmotic stress was induced immediately. 4 hours later, we observed biosynthesis and transport of auxin. Here we shown how this biostimulant could work in roots and leaf. Immediately response in roots was related to root transportation and oxidative response. It is important to highlight that genes related to root development and auxin biosynthesis were induced 4 hours after application, and this reaction continues to the foliar tissues, and which gene related to auxin biosynthesis was induced 8 hours after the treatment application. See you, everybody.
So we have reached the end of the program today. We're sorry for the repetition of some of the flash talks, but we think we don't miss any. And if someone was missed, we will have it at the end of at the last day on Thursday. If there is one flash talk that we missed today, sorry, but we will be presented everyone in this meeting. So thank you so much for being today, and see you tomorrow. Tomorrow we'll have also very interesting talks related to plant microorganism and biotic interactions. So we will see you, hear you tomorrow. Thank you so much for everyone.